Good morning, Prof.
Everybody for wonderful participate.
Hello, everyone. A very warm welcome to one and all, Dean, distinguished professors, speakers, and fellow presenters. Welcome you all to day 2 of the International E-Conference on Biotechnology, Bioinformatics, and Biomedicine. This conference is being hosted by Faculty of Applied Sciences to celebrate AIIMS University's 20 years of research and academic excellence. The theme of the conference is Challenges and Opportunities in Biotech, Bioinfo, and Biomedicine in the Post-COVID Era. Yesterday saw various speakers from different universities share their knowledge and research with us, some amazing presentations and posters. So without much further ado, let us begin day 2 of the conference. I would like to remind all the attendees to please turn your phones to silent mode. We have participants from all over the globe, hence it's a request to be mindful of everyone's presence and refrain from using explicit language while commenting or asking questions. Today starts with Conference Technical Session 3. Each session will have a chairperson and co-chair. So for this session, I present to you Senior Associate Prof. Dr. Neeraj Floria as chairperson and Dr. Annie Jaya Christie as the co-chair. Over to you, Dr. Neeraj.
Thank you, Ms. Sarah. Thank you so much. So hereby we start a very good morning, good afternoon, good evening, and good day to all the speakers, presenters, and other participants. Welcome to our second day and third technical session of AIIMS International E-Conference on Biotechnology, Bioinformatics, and Biomedicine 2020. I am Dr. Neeraj Floria, the chair, and together with Dr. Annie, the co-chair for this technical session. Before initiating this session, let us brief you about this technical session. This session will be initiated by 2 keynote addresses, each of 45 minutes, by Senior Prof. Dr. Manikam Ravichandran, Dean Faculty of Applied Science, AIIMS University, Malaysia. Then followed by Prof. Dr. Christopher M. Austin, School of Life and Environmental Science, Deakin University, Australia. Next, there'll be 2 plenary talks, each of 30 minutes by Dr. Fong Cheng Pan from Weill Cornell Medicine Institute, New York, USA. Followed by Dr. J. Rajendran from Madurai Kamaraj University, Tamil Nadu, India. At the end of each keynote and plenary talk, there will be a question and answer session. If you have any question, you can post it on Zoom link or Facebook or YouTube link of this IECBBB 2020. After the plenary talks are over, the session will be continued with the 6 oral presentations, each of 10 minutes, followed by the lunch. So before we open this technical session, I would like to introduce our first keynote speaker, that's Senior Professor Dr. Manikam Ravichandran. To talk about him, Prof. M. Ravichandran is the Dean of Faculty of Applied Science, AIIMS University, Malaysia. He obtained his Master in Science in Medical Microbiology from Kistan Medical College, Vellore, and gained his PhD in Biotechnology degree from Anna University, Chennai, India. Currently, his research interests are genetically engineered cholera vaccine and formulation, molecular diagnostics, and phage therapy. To his credit, so far, he has 77 publications in various international journals with cumulative impact factor of 152,950 citation, officially from Web of Science, not from Google Scholar. He supervised 40 postgrad students, filed 8 patents, commercialized 3 products on diagnostics. He has been awarded with 44 national and international awards for the academic and research excellence, including Ideas, Inventions, New Products, IENA, Nuremberg, Germany, International Exhibition of Innovation, New Technology and Products, Geneva, and Anugraha Inovasi, Negara, Malaysia. He was an associate member and the committee member on Top Research Scientist Malaysia TRSM of the Academy of Science Malaysia. He was also the former Vice Chancellor of AIMS University, Malaysia. Today, Prof. Ravi will enlighten us with his keynote address titled Cold Chain Free Cholera Vaccine, VC-USM 14P, a potential live oral bacterial vector vaccine platform for emerging diseases. So with this, I request Senior Prof. Manikam Ravichandran to share his knowledge with us. Welcome, Prof. Now over to Senior Prof. Ravichandran.
Good morning, everyone. Thank you, Dr. Niraj, for a kind introduction. And thank you, Dr. Annie, the chair and co-chair. And thank you for the organizing team for making this event another successful day, second day of the conference. Welcome to all participants of this particular conference. Let me share my screen. So good morning and good day, and some of you and good evening. So good day to everyone. And today I'm going to talk about cold chain free cholera vaccine, VC-USM 14P, a live potential live oral bacterial vector vaccine platform for emerging diseases. I'm just going to share my journey of developing a cholera vaccine strain and how this strain can be used in the future vaccines. So it's going to be like a 20 years old story, but it is continuing. That is a part is sustained and the research is continuing. There are so many generations of students and so many PhD and master students have developed. But the beauty of this work is the research is still active. And we have been recognized by WHO as one of the active research going on the vaccine development for cholera. Let me tell briefly about cholera. Cholera is everyone knows it is a diarrheal disease. Basically, we lose water and electrolytes, and it is caused by a gram-negative bacterium, Vibrio cholerae. It can be O10 group or O1390 group. There are many different types there. So mostly O1 and O1390 groups are the one which causes this illness. And it's a permanent disease, and it causes around more than 224,000 deaths every year. The economic loss can be more than 2 billion per year, according to WHO. And 83% of the deaths in sub-Saharan Africa is due to cholera. And it is also the second leading cause of death among children in the world. And this disease is prevalent in more than 69 countries. And most of the countries that has the disease are Africa, India, Bangladesh, Middle East, and Central America. It's a very important disease, and we need a good control over it. But if you look at the WHO statistics, and this disease has been spiking now and then, but according to the latest report available in WHO, and it has more than 1.2 million people affected due to this disease. And though the mortality is going down, but the disease is still prevalent. So why vaccine? Vaccine is the clean drinking water and the proper sanitation are the most important way to prevent any fecal-oral transmitted diseases. Sometimes treatments are too expensive for the people, particularly bottom billion. And vaccines are supposed to be cheap alternative that can be used to protect a large population. So the best way for most of the communicable diseases is sanitation combined with the vaccine is a synergetic and most effective approach for vaccine development. In order to prevent cholera, there are many vaccines available. The vaccines can be classified into killed vaccine, which required multiple dose and it gives short-term protection. The vaccines currently available are Dukoral, Shancol, and Vivicol. Live vaccines also available, but advantage of the live vaccine is you just need to give a single dose. So immunogenicity is better. There's a vaccine called Vaxchora is available. And the cost of this vaccine can range between up to $6 US. And one of the important things we have to remember, the supply chain, the cold chain. You have to give multiple dose and you have to give cold chain. The cost of the vaccine, the cold chain alone will cost around 1/4 of the vaccine cost. Around 23% of the cost goes to the cold chain. So that's why one of the objectives of when we want to develop our vaccine, we started to have a thermostable live oral vaccine. This is a global cholera vaccine landscape analysis by WHO, and these are many different vaccines are currently available in the trial and currently being used. Some are in phase 1 trial, some are phase 2 trial, phase 3, or some are in the regulatory level. You can see that our vaccine VCSM has been listed as one of the very actively active program, and we have been constantly working step by step. We are moving forward in developing this strain. And any way forward for cholera vaccines, according to WHO, there are so many wish lists here. You can see so many wish lists here. It has to be single dose. It has to be safe. Store at room temperature. It should protect more than 80%. It should have a long shelf life. Inexpensive. You want to reach the bottom billion. The vaccine should cost less than $1 a dose. So these are the wish list which WHO is outlining, and we took 5 important cardinal wish lists from this particular specifications. So we wanted our vaccine to be live. It should be able to colonize good. It should be cold chain free, 100% protection, and non-reactogenic. So we developed series of strain when in USM. It's a live oral attenuated vaccine strain. Basically, it's a virulent 0139 strain, and they are metabolic oxotrop. And later, some of the series are P series, which is a prototropic strain. So the journey started in the year, it's not 2000, we started in, before 2000, we wanted to isolate a gene, important metabolic gene in Vibrio. At that time, the Vibrio genome was not available. So we, based on this Heme and M gene that was available in E. coli and other gram-negative enteric bacteria, we aligned the sequence, and then we pulled out this first time this sequence was pulled out from Vibrio cholerae in the year 2000. So almost 20 years ago, we started this journey, and then we constructed an oxotrop of the Heme gene, means ALA oxotrop, and then that we named the strain at VCSM2. That's the first part of my talk. We have filed a patent on it, and then we developed so many strains, and one of the landmark strain was VCSM14P, where we have mutated the cholera toxin gene and also removed the ACE and ZOD gene. It was published in Microbial Pathogenesis. And followed by that, we developed another strain, VCSM14P. It's a prototropic strain. And last year, we published the formulation toxicity evaluation of this particular thing in vaccine. And this January, our team has formulated this vaccine, and it can survive in room temperature without cold chain for 140 days. So this is a journey I'm going to share with you. The last part of the talk will be how this vaccine can be used as a bacterial vector for the emerging diseases. I'm just going to tell the future perspective of this particular vaccine. So this is a journey again. We cloned the gene. We constructed the VCSM2 strain in 2006, and followed by 14 in 2010 and 14P. And we evaluated toxicity of this particular, we formulated and did the toxicity test. And then in January 2020, we published on that this formulation can stay at room temperature for 140 days. So our future plan, achieving toxicology studies are done, and we are going into for looking for a potential company which we can technology transfer and develop the dossier and go for the clinical trials. So the first part of the talk is the oxotropic strain. So safe and effective cholera vaccine can be developed by including mutation in the housekeeping gene of Vibrio cholerae. So this will minimize the vaccine shedding in the environment and diarrhegenic effect. And so we focused on mutating a housekeeping gene called heme gene that code for glutamyl tRNA reductase. So this is our first paper. I just briefly summarize the outcome. The main person who started this work was Prof. Zainul, F. Zainuddin, and it was inspired by him, and we started with cloning the heme gene and created the mutation. Let's look at the metabolic pathway of this heme gene that is that encodes the glutamyl tRNA reductase. So when you look at this pathway of synthesis of 5-aminolevulinic acid pathway, that's a C5 pathway which is present in prokaryote. So this particular enzyme is an important rate-limiting step, glutamyl tRNA reductase, for the 5-aminolevulinic acid ALA synthesis, which is one of the important precursor of cytochrome and many other compounds. So we target this rate-limiting step so that we can stop the synthesis of ALA. If you don't provide ALA, the organism won't grow. So it becomes the ALA oxotrope. This is the gene which we cloned. There are two genes there, heme A and heme M gene. Actually, we did a lot of work on these two genes. It's the first time we isolated. Heme A gene we use for developing the vaccine strain, and heme M gene, it's now currently labeled as a LOL-B gene. It's an outer membrane gene, and it is apparently very specific to Vibrio cholerae. We published a few papers on diagnostics on, you can search PubMed, LOL-B gene PCR and octoplex PCR, and many diagnostics has been developed to detect Vibrio cholerae using this heme gene. It's a very unique gene. So how we did this is actually almost like 3-4 years work. I put it in one slide. The strategy, how we created the mutant. And nowadays you call this method as genome engineering, okay? And those days we used to call it as a site-specific recombination. So what it did was, before I tell anything about Vibrio cholerae is a very nice organism. It's a gram-negative bacilli. But one challenge with the Vibrio cholerae is it's very, very difficult to transform a plasmid into Vibrio cholerae. You can transform a plasmid in E. coli by calcium chloride method. It goes inside very nicely. But however, Vibrio cholerae has membrane DNAs. It has a DNAs on its membrane. Any foreign DNA comes, it chews up. It's very, very difficult to transform any DNA into Vibrio cholerae. So how to transform or put a gene, or how to make do a genetic engineering of Vibrio cholerae. So what we did was we cloned the gene, heme M gene, in a vector, plasmid vector, and we mutated the heme gene with the kanamycin-resistant cassette. So we have the can cassette there, and the heme M gene is there. Then we transferred to a suicide plasmid. Actually, it's called PWM91. So this plasmid will not replicate in normal E. coli, normal cells. Once it goes there, it gets integrated. But this plasmid can multiply in E. coli. So this was in a special E. coli, it will multiply, and it has that the TGORT. So it will be able to conjugatable into the Vibrio cholerae. So through this E. coli host, special E. coli host, the mutated heme gene construct was transferred via conjugation into Vibrio cholerae. So the gene goes inside to Vibrio cholerae, but it will recombine into the genome, and by two selections, so we got a mirodiploid, and then we did look for the recombinants, and we finally obtained the heme mutated kanamycin resistant, that is VCSM1. Later, we do another recombination to remove the antibiotic resistant gene from the heme mutant of Vibrio cholerae. So this is a very interesting side, and that was our dream when we started our work. We wanted to, this is a wild type on the left side, and the right side is a VCSM1. You know that bacteria divide by binary division. Every time it divides, the cytoplasm and the whole content is separated into two. So what we do is, what the theory is that when you grow this bacteria, like wild type, the ALA concentration is there. When it divides, the ALA concentration will be 100% because it's synthesized, the gene is there, it will produce. But this VCSM1 mutant, the heme gene is mutated. So when it divides, you can grow them in the presence of ALA. When you remove the ALA, the ALA content becomes 50%, and then it will become 1/4 of it, and then we go on. And further, by few divisions, the bacteria won't have that supply of ALA, and it will die by itself. That was our strategy. So we wanted to use the full repertoire of all the virulent factor in the Vibrio cholerae, but we want to program the Vibrio to die in few generations so that we can elicit a good immune response, and it can be a very good vaccine candidate. So that was our strategy of safety for the biological containment of VC USM1. So every multiplication, the cellular ALA is depleted, and consequently, VCUSM auxotrop fails to multiply, but the wild type, as it has this gene, it will grow. So we check the protective efficacy of the VCSM1 and 2 in rabbit model. Is retard means rabbit intestinal tied adult rabbit model, removable intestinal tie adult rabbit model. So so basically the rabbits are orally vaccinated with this strain on day 0 and day 14, and were challenged with by the retard method with the wild type strain. Vaccinated strain survived, but unvaccinated strain died. And this is our first trial when we did that, and we found that all the vaccinated rabbits with our strain survived. And we also checked the environmental stability of this strain, because when we developed this vaccine, so we wanted to die in few generations. So we tested, we put this, because Vibrio is a strain that present in river and sea water. So we tested this strain on various waters, and we found that in seawater, and the 3rd or 4th day, they are not able to stay. And even in river water also around 3 to 4 days. However, the wild type strains were able to survive in the environment very well. So the overview of the results, it colonizes and multiplies in the small intestine of infant mouse very well. And it also elicits a good humoral immune response, like vibriocidal antibody, anti-cholera toxin IgG, IgA antibodies, and it had a little bit of reactogenicity in the ileal loop assay, and it was 100% protective and did not survive in river and seawater, and it was environmentally safe. So in conclusion, this strain was good, auxotropic strain, and it colonized well, 100% protection, but you can see the reactogenicity is still there. The reason is the cholera toxin is still inside there. We thought that attenuation by auxotropy will prevent the toxigenicity of the strain, but it is still there. So we filed a patent in US and Europe and so on. But the main problem was this strain was mildly reactogenic in animal model, but higher dose due to the presence of the CTXA gene. Cholera toxin is coded by 2 genes. One is A and B. A is the active gene which causes the toxigenicity. B is the binding factor. B is a very potential, CTB is a potential adjuvant. So the summary of our strain is it is reactogenic and cold chain free, but it fit the criteria of live attenuated, good colonizer, and give good protection. So this factor we sought. So we went the auxotrop, the CTX operon is still intact. So we went back to our boardroom, and then we started another study. So we mutate the CTX operon. So we don't want to delete it, because if you see most of the cholera vaccine, they remove the CTX A and B from the thing, or they screen for a naturally available strain which lacks the cholera toxin. Then they use it as a strain. But we did not want to do that. We want to keep both A and B there, CTX cholera toxin A and B there, but we want to mutate the active sites that is responsible for the toxigenicity. So we mutated by side-directed mutagenesis, and we developed a strain called VCSM14P. It was developed by one of my postgraduate students, Ms. Melissa Chan. And so this we wanted to construct a live O139 strain, but it should mimic a natural infection, but it should not cause even a mild diarrhea. So, so we mutated the 2 sites of the CTXA gene, and we also deleted the ACE. So these are the other accessory virulent factors of Vibrio cholerae we deleted. And then we transferred into the VCSM2 by allele replacement. And in particular, you want to know specifically, we mutated the 7th and 112th amino acids of the CTXA gene. So we mutated, and then we checked it, and it did not cause any diarrhea. And over this line, and we developed a single CTX mutant, VCSM11, and a double copy mutant, VCSM12, 13, and 14. Several strains were produced during this process. And these are the plasmids we have used to mutate the gene. One of the first things we wanted to check after we constructed, we took the rabbit intestine and we injected the wild type. You can see a lot of fluid accumulation inside that. But when we injected VCSM14, which is mutated, and it was almost same like normal saline. So there was no toxicity at all in that strain. And the fluid accumulation ratio was 0. In 14, when compared to 2 and wild type, it had a fluid accumulation ratio, and fluid accumulation ratio at the higher level also 10 to the power 8, that is actually 10 to the power 8, it still had no fluid accumulation. And so this is not it is producing cholera toxin, but it is not functional, but it is antigenic. So the CT ELISA experiments were performed, and we found that this strain did produce cholera toxin that could be detected by ELISA, 1 to 1.2 nanogram, or could be detected from every 100 microliter of the LB we used. Relatively same, but a little bit less than the wild type strain. And the colonization by the mouse, because if you want any vaccine to work, the bacteria has to colonize onto the intestinal epithelial cell. So we use a mouse model, infant mouse model, and we found that they were able to colonize very well in the infant mouse colonization assay. And the good news is it is not reactogenic. It seems to produce a little bit less cholera toxin compared to VCSM2 because it's still an oxotrope. And it has also the holotoxin, the A and B has not formed well because there's a mutation there in the 7th and 112th amino acid position. It was a little bit poor colonizer. So coming back here, you can see that this particular strain become poor colonizer, but it's non-reactogenic. So the safety is there. It gives protection. It's live attenuated, but colonizing capability and the cold chain property is still not there. So we understood that oxotropy, our first dream of creating an oxotrope, was not logical. So the oxotropy led the strain into a poor colonizer. So the mutagenicity or mutation of the CTX operon was actually a good idea. We did that. So both CTX operon have been replaced by mutated operon, and 14 was not reactogenic, but it's also poor colonizer. So the colonizing ability can be improved by putting back the heme gene. So this is what we did. We reverted the first experiment we did in 2006. So we constructed the 14P candidate wherein we put back the heme gene into this strain. So this is actually also a long work I'm summarizing in one slide. This time we took the wild type. It's actually, we did the gene therapy for the bacteria. So there's a mutation of heme gene in bacteria. So we use this heme and M gene and put it back into PCVD, and then we transform. By conjugation, it was transferred into 14P. We obtained mirodiploid and then finally got the 14P that has the intact heme and M gene. And allele replacement was conducted, and the strain now is prototype. No longer needs aminolevinic acid for its growth. It's not an oxotrope again. And it found to be very good. And 14P was able to colonize relatively better when compared to the wild type. It was able to colonize. And the cholera-toxin production also is almost same. 14 and 14P and wild type, almost same amount of cholera-toxin was produced when we determined by polyclonal antibodies. And fluid accumulation is almost nil when you compare in the rabbit intestinal model, and we found that the 14P was very good, and there was no fluid accumulation in the rabbit intestine. And we also checked the antibody profile, the serological profile, whether it produced a good antibody or not. So it produced a good anti-LPS antibody in the vaccinated animal. And we also able to find a neutralizing anti-CTB and IgA, IgG antibodies. And fluid accumulation, also we checked at the higher doses. So higher doses there was there, but up to 10 to the 6, there is no fluid accumulation. Vibrio-cholera antibodies are basically the antibody, when it gives the complement, it kills the Vibrio cholera. It is one of the important hallmark of the cholera immunity. So if you have Vibrio-cholera antibody, that vaccine definitely protects the particular serogroup. So we got a titer of more than 20,000 when we used this 14P as immunize the rabbits, and we got a very good titer of Vibrio-cholera antibodies after 2 weeks. And we also did a challenge studies in the retired model, and we found that vaccinated animals were protected when challenged with wild type Vibrio cholerae. No diarrhea, no death was noticed when we did in a rabbit model. So basically, the prototype carrying the CTXA operon was successfully constructed. We named as VCSM14P, and it's a prototypic strain. And there is no, it can grow without aminolevinic acid. It colonized very well in the infant mouse model, and there's no pathological changes. Actually, we also published few paper on the pathological changes of the wild type strain and the vaccine strain, and it does not cause any damage in the rabbit ileum. And it was able to elicit a good amount of antibody titer, and it gave a good protective efficacy. So finally, we have this strain which is very good colonizer, live attenuated, 100% protection, non-reactogenic. But still the cold chain free part was not done. It's a very good colonizer. We are very happy with that. We went on developing the formulation. And so we also did the toxicology test. We did a repeated dose toxicity evaluation of this cold chain free formulation in a rat model, and it was led by Dr. Gurusamy Prabhakaran and his team. And he came up with a wonderful formulation, and the toxicological test was very successful. No toxicity was detected, and he developed a very unique formulation. And this formulation can be kept at room temperature for 140 days, and he has filed a patent on it. And it is also, we also filed a PCT patent very recently in few countries. It was published in BMC Immunology in last January. So currently our vaccine formulation can stay at room temperature for 140 days. I think it is, you can, as I mentioned to you before, the vaccine cost, the 1/4 of the vaccine cost goes to the production cold chain. So cold chain can be removed, and it is live. You don't have to have 2 doses. It's a very good candidate vaccine. So with this, we broke the cold chain. So no need to have a cold chain. So you can, I think he has estimated, as I mentioned you, 23%. We can reduce the cost by 42% because it's going to be single oral dose. And it will, this formulation will be it's low cost, cold chain free, non-reactogenic, and it can protect. And it also won Falling Wall Innovation Challenge in Berlin. It was won by Dr. Prabha. And finally, we have achieved all the 5 goals which we started with. It is live attenuated, good colonizer, cold chain free, non-reactogenic, and 100% protection. So this is our journey, long journey of 20 years journey, and we cloned the gene, developed the first ALA oxotrope, and then we create a oxotropic CTX mutant, and then we made the mutant into prototrope, and we evaluated the toxicity, and we developed the cold chain free formulation. And currently we are using this strain for cross protection because there is a 86% similarity of the LT of ETEC, enterotoxigenic E. coli, with the cholera toxin. So we have one project going on by Dr. Prabha and his team to develop this as one of the vaccine for traveler's diarrhea. The trademark of this vaccine has been registered as a MyCall. And currently we are going through this process, the GLP, preclinical toxicology studies of GLP and so on. And these are the various strains we have developed. You can look at the genotype later. And so many of them are currently going on. So now I want to switch gear a little bit. Maybe next 10 minutes, I'm going to talk. My talk should finish in 10:15. So I have another 10 minutes. I want to spend time with you. We all know DNA vaccines. Even in the current SARS-CoV-2, we have RNA vaccines currently in the final stage of the trial, and DNA vaccines, the people are using it and so on. So what are DNA vaccines? Basically, DNA vaccines are basically a vehicle, basically a plasmid, the prokaryotic promoter and replication origin, and it also has a eukaryotic promoter and eukaryotic replication origin, and it has a gene of interest. Let's say you want to develop a vaccine for COVID, we put a spike gene inside here, or if you want to develop a vaccine for another viral disease, you can put the gene there. So the plasmid that has an antigen encoding antigen with the mammalian promoter and bacterial promoter can be cloned into this, and this plasmid can be given into the system and in the mammalian cells, and this can express the antigen and give an antibody response. So very easy to develop. You just need to have a vector, just clone the gene and you can do it. It can be delivered by gene gun, and it can be delivered by injection. It can be delivered by many other different ways. So one of the disadvantage of this particular vaccine, which has been developed for TB, diabetes, and cancer and influenza, it's the lower efficiency in large animal. You need to give multiple dose, and that has a major barrier. But advantage is very easy to develop. We can no cold chain is required, because to transport DNA, I think you don't need a cold chain. And delivery, in terms of delivery, you can use gene gun to deliver DNA vaccine, or you can use the injection. You can do electroporation, you can use nebulizer. However, one of the best way to deliver is the oral way of vaccine that will work very well. So I'm just switching here. I just want to tell that bacterial vectored vaccine that deliver the DNA vaccine as a carrier is one of the best possibility to develop a vaccine. In our case, we have developed the VCSM14P that is a very good bacterial vector. So that can use as a carrier to the carrier as a DNA vaccine vector, and that can be transferred into the mammalian cell. Examples of this DNA vaccine, bacterial vector DNA vaccine, has been developed in the literature, like Lactococcus lactis, Salmonella species, Listeria monocytogenes, Shigella species. All has been successfully used to deliver foreign antigen into the mammalian cell, using that as a carrier for the DNA vaccine. So what is the model we are trying to propose now is that, so we have done a wonderful model 14P, which works very well for O139 cholera. That's fine, but it is a very good carrier. The reason is it has a built-in CTB, choletoxin B. Choletoxin B is known as an important mucosal adjuvant, very important mucosal adjuvant, which is already built in there. So the thing is, you just need to transform the DNA vaccine plasmid into this 14P as a carrier, and it can be done in a very short time. You don't even have to clone it. You can order a company that will construct a plasmid for you that can be delivered into this VCUSM 14P, and you can do the challenge studies and whatever studies can be done and move on. So the oral delivery of the DNA vaccine is simple and efficient, and it is such a way of developing a live bacterial vector. To my knowledge, to the thorough search of literature for a COVID-19 SARS-CoV-2, there are a lot of viral vector, adenoviral vector, or other viral vectors has been developed, but there's no live bacterial vector has been utilized for the development of vaccine for SARS-CoV-2. I believe this kind of bacterial system would be a good system to develop a vaccine. And the reason is it targets the mucosal immune system, is low cost and effective antigen delivery, low reactogenicity. Most importantly, it will lead to a mucosal immune response, because that is the viral entry. It enters our mucosal system. IgA will neutralize it. And one more thing about it is there won't be any ADE. Antibody dependent enhancement of viral infection will be minimum, because the immunity elicited by these vaccines will be mostly IgA mediated. So what happens in this vaccine, a bacterial vector vaccine is, so this is a bacteria that has a plasmid. So bacteria are recognized by the immune system, and it is phagocytized by the mammalian cell. And inside the cell, the bacteria fuse with the lysozyme and form phagolysosome, and lies, and the plasmids are released into the cell. That the DNA plasmids are engulfed by the dendritic cells, and inside the dendritic cells, the antigen gene is expressed as a protein, and the MHC, in the presence of MHC class 1 or class 2, and antigen can be efficiently induce both. This one can induce a cellular immune response and also the humoral immune response. Any bacterial DNA vaccine, it can be a plasmid encoded or chromosomally integrated. So it can be, you can use it at a both way. Easy to develop a plasmid encoder, just transform. You just have to send the plasmid into the bacterial system. So once it goes inside, the protein can express inside the cell, or it can be displayed on the cell surface, or it can secrete the cells. Once they are done, then can elicit both the humoral and cell-mediated immune response, and it can give a good protective immunity, whether the antigen could be a viral or a bacterial or anything. Very easy to, it's a kind of plug-and-play system. So you already have the host ready. You just have to put the gene inside and then move on. So the fast-track DNA vaccine development strategy, actually, this is one of the slides we proposed to one of the funding agency. So we have this 14P, and the formulation is ready. You just need to put the plasmid inside, and you can go for doing the testing of protective efficacy of this particular strain. And this is a summary we propose in that. So the unique feature of 14P as a bacterial vector prototype, it's a oral, very easy to give. It's cold chain free, 6 months you can keep. So it's easy, and we are targeting, I think, all the vaccines which you have produced, and we have to think those poor bottom billion can afford or not, whatever vaccine we are developing it. But this vaccine, I believe, it will be affordable, live. It has a built-in adjuvant. I mentioned it has a CTXB. It gives a better mucosal immunity, less cost, and no additional adjuvant required. And it gives a mucosal immunity, IgA mediated, less antibody dependent enhancement. That is my gut feeling. That's my personal opinion. And bacterial vector 14P platform is already almost 20 years, and it has protected by many patents, and it has a great market potential. The whole population is there. So I think this 14P, I think, will be a game changer. It's a very good potential bacterial vector candidate that can carry any future antigens for any of the emerging diseases. I'm one of the person who started from the beginning. I'm still alive. And Prof. Zainul was the person who started this work. Lots and lots. I could not put, this one slide is not enough to put all the images of students who worked on it. It's a lot of people's hard work. And anyway, the guru who started is Prof. Zainul, and I think some of us passed away, some of us, I'm still alive. And the current generation currently working on is Dr. Prabhakaran and Viro. They are the one who really are doing the formulation, bringing this vaccine to the next level. I'd like to acknowledge all my students. This whole talk is dedicated to all of them who made this vaccine a great success. So hopefully this thermostable cholera vaccine will reach the global immunization program for the bottom billion. That's our dream. And the team is working. I'm fully confident with my team. And I hope this particular strain will make will make some change in this planet Earth. With this, I think I finished my talk on time, I guess, and I will leave it to the chair and co-chair to handle the session. Thank you very much for your attention.
Thank you, Prof. Thank you so much. Thanks for enlightening us with the various statistical, economical, genetic, physiological, microbiological, toxicological, and other pharmacological and other aspects of cold chain free cholera vaccine. So I'm sure your talk will definitely benefit the budding and other scientific community also. So Prof, I believe, actually there's no question, but I have a question for you, sir. Because the way you have explained, it's complete actually. So within how much time we can expect this vaccine to come in?
It has to go through all these phase 1. We have completed only the preclinical stages.
Okay.
And we have developed the dossier. And the vaccine has to be produced in GMP, good CGMP facility. So once it is done, it has to go through phase 1, phase 2, and phase 3 clinical trials. Basically, we are looking for a potential pharmaceutical partner. I think it's a gold mine. They can take it up. They can take it up, and it's an omnipotent vaccine. It can be used to deliver other antigens.
So it's a wonderful message for all of us, sir. It's very nice. I think it will be benefiting the whole scientific community. So far, Prof, there is no question. I think everybody has understood. I think for everyone, it's a novel concept also. With this, thank you, Prof, because as there is no question. So still, if at all somebody has got any question, maybe they can post it on Zoom link, Facebook, or YouTube link of this IECBBB conference. So with this, thank you, Prof. Thank you so much. I'm sure your words will definitely be useful for community. Thank you. Thank you so much again, sir.
Yeah, thank you so much for the organizing team, Dr. Suresh and Dr. Annie and Dr. Niraj and Dr. Subhash and everyone I could see. I think there are many of our friends and all the people from all over the world. Thank you so much for listening to my talk. Have a great day.
Thank you, Prof. Thank you so much. So now with this, we go to our next keynote address. So I request Dr. Annie to take up the chair now.
Yeah, thank you, Dr. Neeraj. Thank you, Senior Professor Dr. M. Ravichandran. It was rather informative and also it was enlightening taking through your travel in your research field. And now we would like to invite Professor Christopher Austin from Deakin University. Professor Christopher Austin, actually his CV is almost more than 40 pages. So we'll just summarize his achievements in his career. Professor Christopher M. Austin is presently working as head of School of Life and Environmental Sciences in Deakin University, Australia. He has completed his undergraduate in 1979 and his PhD in 1986 in University of Western Australia. He has almost 35 years of research and academic experience. He has headed various departments in different organizations like Deakin Genomic Research Center, Charles Darwin University, Australia, Auburn University, Alabama, USA, Tropical Medicine and Biology Multidisciplinary Research Platform, and Genomic Center, Monash University, Malaysia. He has also established various molecular genetic, and genomic laboratories in different universities like Charles Darwin University, Australia, Queensland University of Technology, Australia, Deakin University, Australia, and Monash University. To his credit, he has received various research grants of more than $8.5 million in the last 10 years. And he has published more than 150 publications with high impact factor. The special focus of his research are in the areas of development of integrated wet lab bioinformatics for genomic and population genetic applications, long and short read hybrid assemblies for animal genomics, metagenomics, genomics, transcriptomics, and molecular marker development, molecular taxonomy, population genetics, and phylogenetics and phylogenomics, evolution, biodiversity, biogeography, and conservation of inland, aquatic, and marine species, molecular ecology, phylogeography, and conservation genetics, sustainable aquaculture, production, and nutrition, and the list goes on. Under his supervision, he has about 5 scholars who have completed their masters and 21 scholars who have completed their doctoral degrees. Apart from the academics, he is also an eminent leader with extensive administrative experience in contrasting and multicultural academic environments. So he has a very good experience both in academics and administrative. We welcome you, Prof. Austin. Over to your session now.
Thank you very much. Look, I feel exhausted just listening to that. Firstly, I'll try and share my screen so you can see my presentation. Okay. So how's that for everybody? Can everybody see my first slide?
Yes, Prof.
Yes, Prof.
Thank you very much. So firstly, I'd just like to thank the organising committee. It's a great honour to be able to talk at this meeting. And especially because I have a strong connection with Malaysia and great fondness for the country and the people, and I certainly hope to be able to continue that. Of course, at the moment, I'm based in southern Australia, in the state of Victoria, where we're having our own battles with COVID-19 at the moment. So we're still all working from home. So from the introduction, you've got an understanding that I've been working in the field of molecular genetics for quite some time. And today I just want to focus on one thread of my career, if you like, which is focused on mitochondrial genetics or mitochondrial genomics. Now, obviously, there's a growing interest in and capacity to undertake big-scale genomic studies. But over the years, probably mitochondrial genes have been used excessively to support a range of studies in the environmental sciences, ranging from population genetics through to molecular genetic identification of species, molecular taxonomy, evolutionary biology, and phylogenetics. So they're all areas that I'm familiar with. And of course, there are not too many advantages in growing old, but one of those, as long as you can keep your memory, is that you can draw on experiences over many years where there have been changes for better or worse, which can give you a good guide to the future and put many of the more recent developments into context, which is obviously a bit more difficult, particularly for young academics and students, because they tend to be introduced mostly to the latest and most interesting techniques, but often found it really helps a depth of understanding to understand the past, and also some of the scientists and characters who've contributed to major developments in the field. So today I'll be reflecting on nearly 30 years of experience working on mitochondrial genetics in the fields that I've mentioned. I will apologize up front that my bias will be towards animal mitogenomics, and even more specifically around fish and crustaceans. You can see one of my favorite animals. Looks like it's about to drag me into its burrow behind me, which is a very well-known Australian crustacean called the yabby, which will feature quite often in this talk. So what I want to do today is just give a bit of a refresher on the mitochondrial molecule. So I know there'll be some people here from biology and biotech backgrounds who will know a lot of the details about why the mitochondria is special, but just in case there's a few people with computer science or chemistry backgrounds or mathematics, then I'll give a bit of a biology 101 just to make sure that they understand the context. I'll then look at a bit of a history of the molecular genetics, particularly with reference to how our understanding of mitochondrial genetics and genomics has evolved over the years. And then I'll also go over a number of different applications for mitochondrial information in the environmental sciences. And mainly because I'm familiar with it, is that I'll use examples from my own research group to illustrate these points, which I hope will be of interest to the audience here. And then I'll speculate a little bit about the role of metagenomics going into the future, particularly as many of us will be interested in the amazing things we can now do in terms of whole genome assemblies, which of course is about a lot more information. But I think for smaller laboratories and research groups, I think metagenomics is still an important field. And I also see that it has great prospects for supporting other disciplines, particularly new areas that are quite exciting around eDNA and rapid biodiversity assessment, particularly in aquatic environments and to some degree soil environments. And also I've been recently brought into the field of food fraud, where there's a lot of interest in what the real composition is of the food that we're eating. And again, here, mitogenomic markers can be important. And also in other fields like ecology, a lot of ecologists work on particular species or need to know what their species are eating. So again, mitogenomics can facilitate analysis of diet at the molecular level. Okay, where am I? So advance that. So, okay, what are mitochondria? So they're small membrane-bound organelles that are found in the cytoplasm of all eukaryote species. They're the site of respiration, that is where the energy for cells and organisms are generated, ultimately producing ATP from glucose. So they're a key component of how cells work for eukaryotes, and for the biologists and biochemists, then of course you'll know the mitochondria as the site of the citric acid cycle, or sometimes called the Krebs cycle as well. So the mitochondria is an incredibly important organelle within cells in terms of maintaining homeostasis and the functioning point of view. What's really interesting about mitochondria is that, I guess, if we go back 50 or so years, while they were known to be present in cells, nobody really knew what their origin was. So there's an interesting story here that intersects with my own journey through academia. And so way back in 1975, when I was a first year, I actually remember being introduced to a very controversial theory at the time, which was the endosymbiosis theory for the origin of mitochondria and chloroplasts. So it was speculated that about 1.5 billion years ago, when only prokaryotes existed, a larger prokaryote and a smaller prokaryote basically entered into a mutually agreeable relationship, which then ended up as the basis for obviously the very highly successful eukaryote cell. So basically mitochondria were thought to be once an independent free-living prokaryote that basically took up residence inside a larger cell. a little later on, one of the cell lineages, if you like, or the species lineages, this particular endosymbiosis got a little bit more complicated when a different kind of prokaryote was also absorbed, and that is the one that eventually evolved into the chloroplasts and of course gave rise to the very successful plant lineages. So while most students would know this as gospel, as accepted truth, in the mid-70s it was far from simple. And the idea was introduced by Lynn Margulis, who's a very famous female scientist, and her paper, while she was still a young member of staff, less than 30 years old at Boston University, she had published a paper on the origin of mitosing cells in the Journal of Theoretical Biology. Now, like a lot of, I guess, big ideas, this was rejected at the early stages. In fact, I think she had to submit her paper to I think 15 times before it was finally accepted. And one of the reviewers of her grant applications wrote back, "This is rubbish, do not bother to apply again." So again, this is an interesting lesson in persistence and self-belief, and don't always take reviewers' comments to heart, and just keep on persisting. For those people who are interested in just a little bit of trivia, then Lynn Margulis was married to Carl Sagan, the famous astronomer, in 1957, which happens to be the year I was born. So for those of you who don't know Lynn Margulis, then certainly that's a connection that you will probably know about. Now, her idea was experimentally demonstrated 10 years later by Robert Schwartz and Margaret Dayhoff, and the mathematicians here today will recognize that name. Another very smart female scientist who was a pioneer in bioinformatics. And then by the early 80s, basically the idea became widely accepted. And the key point here was, by that time, it had been discovered that both mitochondria and chloroplasts had their own DNA, which was circular, and showed consistent differences with nuclear DNA. So that basically was the point at which her theory became widely accepted. So that was my initial introduction to mitochondria. I then completed my undergraduate degree and commenced a PhD using a different kind of molecular technique that was very popular at the time. And I didn't come across mitochondria again seriously for about another 10 years, but I'll come back to that story in a little while. Okay, so just move on. So the features of the mitochondria, which I've already referred to in passing, is that it's a very abundant organelle. So it's highly abundant in cells. There are many, many copies of the mitochondria. It has a separate genome. It's clonally and maternally inherited, so it doesn't is not subject to recombination, which is important. And then while it's fairly consistent in size in the animals, so can people see my cursor on the screen here? Maybe not, I'm not quite sure.
Yes.
Yep.
Yes, sir,
okay,
we can
so
see.
these little rings here are the relative size of different mitochondrial genomes. I don't know how well you can read this on your screens, but oops, we've got humans down the bottom, mice, fruit flies are all very similar in size at about 16,000 base pairs plus or minus. The amoeba is a lot larger, and then yeast is quite enormous compared to the animal mitochondria. And then plants, the mitochondrial genome does something completely different. So at the top here, we've got tobacco with over 430,000 base pairs. So, and The other thing about mitochondria is that it evolves about 10 times faster than the nuclear DNA in animals. It's more conserved in plants than that. So that's another important feature of the genome. So a key and important element here is that the number of mitochondria in a cell can be in the hundreds to thousands, and even an individual mitochondrion can have over 20 copies of its genome. So that means you're potentially looking at tens of thousands of copies of mitochondria, even just in one cell, let alone a sample of tissue. And if you compare that with just the 2 copies of the nuclear, each nuclear gene, then of course it means that mitochondrial DNA is going to be a lot easier to isolate from tissue, and particularly if that tissue is degraded or preserved. And so, as well as some other special features of the mitochondria, this is one of the key features that has made it so attractive to study over the years, because it's easy to work with, particularly once the PCR revolution got underway. The other aspects to the mitochondria is that unlike the nuclear genome, there's very few intergenic regions or repeats. As I mentioned, it's fast evolving. And even though individual genes and parts of those genes may evolve quite quickly, generally the order of genes within the genome is relatively conserved, and certainly the number of genes are conserved. So you'll see the publication I put up there is, I believe, the first animal mitochondria to be sequenced, which not surprisingly was the human. And you can see in the list of authors there, one F. Sanger. So I think we can assume that Fred Sanger lent his expertise to the sequencing activities. And the quote there is that the sequences show extreme economy in that the genes have none or only few non-coding bases between them. So this is what has, as said, made it very useful to work with, is the fact that it's a very consistent organization with the 13 protein coding genes, the 22 transfer RNAs, 2 ribosomal RNAs, and a large non-coding region. Now, as we see, as with almost everything in biological sciences, exceptions will apply. But if we go to the next slide, then this was, I think, probably the next mitochondrial genome to be sequenced, which was by the same research group the following year. So I don't know how many thousands or tens of thousands or hundreds of thousands of dollars it would have cost. But in those days, before PCR and before the Sanger sequencing that we know now, it would have been quite tedious and labor intensive. So the outcome here was that the next genome was the bovine genome. And surprisingly, the sequence homology was quite close, particularly with regard to the organization of genes. So in fact, the genes were almost, well, they were identical in terms of their gene order. So the number of mitogenomes from this time accumulated rather slowly and really didn't get much momentum for another 10 or 15 years. But now we have almost 8,000 mitogenomes from an NCBI, and that has been because of a very great surge in activity over the last few years. Pretty much all major animal groups are now represented, but sampling is still quite biased, particularly towards mammals and the vertebrates. So if you're working in invertebrates that are not insects, then you'll find that there are major groups that are poorly represented on data banks. But as the number of mitogenomes build up, then of course that's a very rich source for comparative studies. And even if you don't have a wet lab yourself, quite often now, mitogenomes are submitted to databases without any higher level analysis. So if you're short on funds and you've got a good student at bioinformatics, really you just need to mine NCBI to assemble data sets that can be of great value in terms of higher level analysis. As I said before, generally, and particularly the early findings, was that the gene order, the 37 genes, were quite conservative in major groups. And you might be disturbed to know that your mitochondrial gene order in your cells is identical to that found in fish, or at least fish-like, the trout. So that gives you a sense of how conservative gene order can be, but luckily the individual genes do vary quite a lot. In 1999, Bohr published a review of the state of animal mitogenomics, and at that stage, less than 90 mitogenomes were available, and the majority of those were from mammals and from chordates. And from my perspective, because my interests are in crustaceans, at that time, there were no crustaceans represented on the international databases. So I'm going to change track a little bit here. So that was a little bit on the history of mitogenomics, on what mitochondria are, the nature of the mitogenome, and the state of mitochondrial genome sequencing as it was about 20 years ago. In parallel to these sequencing activities, which were a bit slow and tedious, mitochondrial information became of great interest to environmental scientists who were interested in population genetics, molecular taxonomy, and evolution. And so as this information accumulated, knowledge of the levels of variation in mitochondria, people realized that this could be a very valuable genetic maker for particularly population and species level studies. Up to that time, and certainly probably initially started in the 60s and particularly the 70s and 80s and some of the 90s, the predominant technique used was something called allozyme electrophoresis or gel electrophoresis. And some of the audience who are closer to my age might remember that. It was actually a technique that I used for my PhD, and I still miss it because you could actually generate lots of good nuclear data inexpensively in a short period of time. And you can see on the slide there an allozyme gel where you can clearly see some different alleles, some homozygotes and some heterozygotes. But it had its limitations, and there was a great emphasis now on using mitochondrial information to contribute to those fields. The early methods were pretty horrendous. You had to be a very skilled molecular biologist and biochemist. And I did try to use some of these techniques back in the mid-90s, and I was very glad that PCR turned up very soon after that, together with Sanger sequencing. So I'd be interested. And so before I leave John Avise, so he was firstly a pioneer of allozymes, and then he became a pioneer of the use of mitochondrial information in, as I said, population genetics and systematics. And he also introduced us to a field which goes hand in hand with mitochondrial DNA analysis called phylogeography. And it's a special field because it combines both what you might call the information we can get from molecular markers about the degree of difference within and between populations and the broad divergence between populations and between species. But it also introduces to a concept of phylogeography and genealogy as you might apply it within the population context. So phylogeography is really a bit of a hybrid between geographic-based genetic variation and genealogy and phylogenetics. But this was a major revolution at the time. And again, Some of you may recognize that diagram in the bottom right-hand corner, where we have a map with the various circles with other circles embedded within those circles. And basically those circles are encompassing samples of similar genetic background. And then if you've got good eyesight, there'll be some bars connecting the bigger circles with a number of hashes in them, and those hashes refer to the number of possible mutational steps between those groups. So again, that's a bit of history that's still with us. This is just the point I'm making, is that we should appreciate some of the historical developments that makes our life in the lab a lot easier. In the early days, mitochondria had to be separated by high ultracentrifugation, which took about 10 hours using cesium chloride gradients, which is quite expensive and it may or may not work. Then if you got separation of your mitochondria, you would then subject that mitochondria from that particular sample to a restriction digest. And if you then had mutational differences between your different samples in the mitochondria, you would get different bands, different number of bands in different positions based on the size of the fragment, would allow you to genotype that particular sample. So as you can gather, that was rather tedious and time-consuming, hazardous, expensive, and frustrating. I remember looking at my autoradiogram, and I think I might have been able to see some bands there if I held it up to the light in the right angle. But I just could not imagine doing that for 20, 30, 50, 100 samples. But you can also see on the right-hand side there, there's this diagram with circles and arrows. And again, that's a way of representing the haplotype differences between the samples, which again was a precursor of the basis for analyses later on that became a lot simpler once we had PCR and some robust primers to use. So this is where, for me, I started to get seriously involved in mitochondrial genetics with my lab at Deakin University. So once we had some good primers, we used PCR rather than whole mitogenome isolation to amplify up fragments and then use restriction enzymes like in the top reddish diagram to identify different haplotypes. Or we used a technique called SSCPs that relied on different conformation based on different sequences, which are run out on an acrylamide gel. Then, of course, once Sanger sequencing became readily available, which it did through various companies in the mid-90s in Australia, then for certain application, obviously we used Sanger sequencing, but Sanger sequencing was still pretty expensive, so we'd only use that on a restricted number of samples. But for population genetics, where you might have usually up to 50 individuals per sample, and samples might be between 20 and 50, then you had to use either restriction fragments or SSCPs to estimate gene frequencies within population, because otherwise it just, you couldn't do it because of the high rate of costs. But all these techniques were based on Sanger sequencing. So what I've always tried to do is try to be at the forefront of the use of these newer techniques, and where possible to collaborate with people who are smarter than I am and who know more than I do. So one of my early successful collaborations was with Keith Crandall. Some of you who do phylogenetics may recognize his name. At one stage in about 2000, he had the world's most highly cited paper. Well, he was the most highly cited author in both bioinformatics and conservation biology at the same time, and was a pioneer in the development of phylogenetic techniques. And so, fortunately for me, he did his PhD on American crayfish, and I did my PhD on Australian crayfish. So we got together and generated one of the first comprehensive 16S based phylogenies for crayfish, which was published in 1999. So, and that was then the start of a very significant focus on the use of mitochondrial markers in phylogenetics. And certainly in the early days, you could get published a paper with one marker. A few years later, you wouldn't get it published unless you had at least 3 markers, or maybe even 5. And then a few years later, then you couldn't get published unless you had both mitochondrial and nuclear markers. So again, that was just like the arms race at the time. But we certainly made great use of sequencing of mitochondrial genes for phylogenetic and taxonomic applications. Another application was in population genetics. So this is one where we used SSCPs. This time, this is actually a fish example using the mitochondrial control region, where we're able to develop a range of haplotypes. So in that first table, you can see haplotypes A, B, C, D, E, F, G, H. So we then estimated the frequency of those haplotypes in each of the samples, which in this case was common carp, study whether they were from an experimental population, from hatcheries, or from the wild. So that then allowed us to estimate diversity, and the diversity in the hatchery populations tended to be low. And then we also were able to look to the extent to which the experimental populations were mixed in the hatchery or were affecting the wild population. So the data was subjected to your traditional population genetic analysis using analysis of molecular variation. And then we also then looked at the relationship between samples on genetic distance and summarized by principal coordinates analysis. So that was just a classic case of how you could use mitochondrial information in population genetics. Now, at that time, we also did a parallel study using microsatellites, which are, of course, are a nuclear marker as well, and the outcome was quite complementary. But what we're also able to do with the mitochondrial information, we're also able to construct genealogies through sequences of these different gene regions. And then, as I talked about earlier on with John Avise, he essentially created the new discipline of phylogeography. So once we'd gone as far as being able to use PCR, we then used that technique along with sequencing to look at genetic variation and relationship between populations, again, of my favorite crayfish species here. And here's a map of the distribution of haplotypes across Eastern Australia, which generated some very unexpected results. And basically the freshwater crayfish across the desert inland regions were much more similar to each other than they were in some of the major river systems like the Murray and Darling River, which is the major rivers in Eastern Australia, where 2 very distinct haplotypes occur. And then in the bottom, you can also see there's quite significant variation in the part of the world I come from. And that also includes, again, cross-catchment migration, which is picked up by this particular method. So again, as you can see, that the combinations of using mitochondrial markers, PCR sequencing, and the new analytical methods of phylogeography were a major step forward. So next, I just want to talk a little bit more about how mitochondrial genetic information has been used. So as I said, after we got beyond having to use whole isolated mitogenomes, we used PCR. And this is where the existing mitochondrial genome information became very valuable. So even though there weren't a lot of species sequenced, by aligning the species that we did have, allowed us to identify conserved regions for building primers, if you like, identifying universal primers. So, of course, that's always what we call the catch-22 with PCR, that it's a great technique, but you've got to have pre-existing genetic information to be able to make it work. And then what happened is that different research groups, depending on the history of the laboratory and the species they were working on, often focused on different gene regions. So I followed Keith Crandall and used 16S, but if you were studying fish, you might have used 12S or cytochrome B. If you were studying reptiles, you might have used NADH. If you were studying insects, you might have used CO1 or COI. And if you're studying mammals, particularly population genetics, you might have used control regions. So that's fine for particular focus studies, but the great value of mitochondrial genetics and sequencing is that it doesn't just contribute to your study. Once you put it on NCBI or GenBank, then that becomes a really important resource that other scientists can use. And this is what led to a concept that many of you will be familiar with now, because the idea goes back 20 years. And that's the idea of biological identification through the use of a universal mitochondrial barcode. So this idea came about, essentially the realization that the differences within and between species can be quite consistently detected through the use of mitochondrial barcodes. And so this idea was introduced by Hibbert. Now, I suspect he just happened to have a preference for COI in terms of his research. If this had been my idea, because I was doing this at the same time, then I might have used 16S. If a fish person had done it, then they might have suggested cytochrome B or 12S. So I guess suggesting that it wasn't necessarily, it was a bit by chance, if you like, that we've now pretty much locked into the use of this one gene region for barcoding. And it's now part of a huge initiative. So it's sometimes called IBOL, the Initiative for the Barcode of Life. There's also a very large data system called BOLD, which sits alongside GenBank. It utilizes information from GenBank, and sometimes it has its own sequences that are deposited exclusively there. So there are now 6 million COI sequences, which are thought to represent over 500,000 species. Now, of course, those species are not representative of all groups, with mostly a preponderance on vertebrates. But what barcoding has done is very much raise the importance of taxonomy identification and biodiversity assessments, having good taxonomic information, and then being able to contribute to ecology, evolutionary biology, food regulatory compliance, forensics, and biosecurity. And the reason you can contribute to all of those fields is because of this existing database. Down bottom there's one of my own studies where I used DNA barcoding as a method to validate the field-based identifications by ecologists, in this case, tagging stingrays in a very remote part of Australia, where it would be impossible to get a taxonomist to get to, and then how do you actually verify your identifications? And you can't, because stingrays are very large, you can't basically put a whole stingray in a small tube and send it to a museum. So this was a case where DNA barcoding was of great assistance in a related discipline. So I don't think this approach to taxonomic identification is going to go away very soon. And as I said, the two key things here is that COI is useful for identifying species, and as the database grows, and of course it has an even greater importance. However, like all initiatives in science, DNA barcoding has its critics, and there are people who have pointed out potential flaws or issues that can come up, which are either major or minor. And some of those, again, is it's very hard to fix on one gene region. Even if it's useful for one group, it may not always be useful for all groups. It also assumes that your universal primers are stable, which is not always the case. Then we also have this annoying thing called NUMPs, which are nuclear mitochondrial-like sequences and sometimes called pseudogenes. So every now and again, parts of the fragments of the mitochondrial genome get translated to the nucleus. And every now and again, PCR being what it is, it'll actually amplify up one of these nuclear located sequences. And of course, for all intents and purposes, we think we're actually amplifying up a mitochondrial fragment. And of course, this will give us different kinds of divergence levels. And in fact, in our study at the top there, when we tried to use cytochrome B for freshwater crayfish, we gave it up pretty soon because in 2 different studies, we found that we're actually amplifying 2 different regions of cytochrome B that were giving us different sequences and were making no sense when it came to its analysis. So this point was also raised by Jennifer Buhay in her publication COI-like sequences, but also the issue, and also in our more recent study of the genome of the freshwater crayfish, where we looked at the occurrence of COX-1 pseudogenes, which we found up to 19 different versions of COX-1 pseudogenes in the crayfish and another decapod lineages, which gives us a great problem potentially. So it just means you've got to be very, very careful in authenticating and curating your sequences. And then of course, people will put sequences on databases and the identification of the species and the reliability of those sequences may be brought into question. And some people have claimed that up to 50% of sequences for some groups are unreliable on the databases. So that gives us great concern. And if you're concerned about this, then I recommend Kelly Micklejohn's paper down below. So I'll keep moving. So with the development of better molecular methods, then the number of mitogenomes is increasing steadily on public databases. And obviously, this has been through the use of techniques like long-range PCR cloning and primer walking, which used to be the most common approach. But now NGS has changed this so that we can now access mitogenomes through the use of next generation sequencing. My experience with metagenomics started with freshwater crayfish nearly 20 years ago. And at that time, there were very few crustaceans sequenced. And so we decided that was a good field to develop. And we were extremely surprised when we found that the freshwater crayfish had a radically different mitogenome order to what we expected. So this may be peculiar to freshwater crayfish, but it certainly gave us some insights into both the challenges and the benefits of mitogenome sequencing, because what we realized that these different rearrangements, not only are they important to understand if you're looking at finer scale genetics, but it also gives you a different kind of molecular or phylogenetic marker. So this was the start of our journey on our own mitogenomic sequencing. At the time, we did 4 crustacean mitogenomes, which was one of the largest studies of its kind. We tried to do a phylogeny, which was a bit sad because we had so few samples that we could access, but generally the phylogeny was what was expected. But at that stage in the early 2000s, we had sequenced 3 out of the 7 decapoda genomes available and 4 out of the 9 Malacostraca. And so hold those numbers, because this gives you a nice marker on how rapidly the field has developed over the last 15 years. So for my research group, we were introduced to NGS in about 2006 using I think what was called pyro sequencing using the Roche 454 at the time, which is no longer in existence. But that gave us these random or shotgun sequences, and we were interested in doing that mainly because we wanted to discover microsatellites for population genetic studies. But with discussions with my colleagues, I said, well, knowing that we know there are so many copies of the mitochondrial genome in cells, why wouldn't we look to see if the mitochondrial, even though this is a very small data set, let's see if there are any mitochondrial gene fragments there. And in fact, what we realized is that you could not only achieve your microsatellite loci discovery, but at the same time, you can often recover the entire mitochondrial genome from these same random sequences. So that was really the start of a whole new way of using these shallow genome skimming techniques to recover mitogenomes. So we then extended that research when I moved to Monash, Malaysia. And by 2014, we'd developed a very fast wet lab pipeline for generating mitogenomes. And at the same time, we realized that as we were becoming so efficient at generating mitogenomes, that then created another problem for us, namely the bioinformatics. So we also developed a specialized pipeline. And so in the second study, we ended up looking at 89 decapod crustacean genomes, 20 of which had come from our lab. So you might remember that 10 years ago, we only had 5. So by another 10 years, we now had 90, and we had contributed 4 or 3 decapods. 10 years later, it was 20. So again, this gives you an idea of how quickly things are developing. So, so this was a major breakthrough in terms of both lab efficiency and also bioinformatics. So a few days to sequence 20 mitogenomes, a few hours to analyze 90 mitogenomes. So we continued to study crayfish mitogenomes and realized that other southern hemisphere crayfish were the same as our earlier results using long-range PCR, so that was encouraging. Northern hemisphere crayfish were completely different, again with a major genome rearrangement and an inversion of a very large block, as you can see down below there. So where are we now? Yeah.
Yeah,
yes.
Just a reminder, you have a few minutes, sir. Thank you.
Sure. Okay, I'll start winding up pretty soon. So we have all these new techniques that gives us new tools for mitogenomic recovery, from the big machines to the smaller machines and long reads and short reads. So as I mentioned before, the NGS revolution has made things a lot easier for us. So again, I won't go through all the details there. There are still challenges. Using NGS doesn't solve all your problems. And sometimes, particularly for Illumina sequencing with high AT-rich genomes, then the levels of coverage can be quite low. And also different methods, wet lab methods and different bioinformatic pipelines can give different results based on the same samples or the same data sets. So because I'm running out of time, I won't go into this study in any detail, but it's one that illustrates the power of combining long reads, nanopore sequencing with short reads when you have a different assembly. And also it illustrates the fact that you shouldn't always believe all the data that's on GenBank. So Shen et al. 2013 suggested the European lobster was missing some genes. We redid it and found that quite the opposite. It had a much longer genome than normal, and there was a major duplication embedded within the European lobster genome, which is quite ironic because the American lobster has your traditional structure, but the very close related European lobster is radically different. And again, I won't say a lot here, but over here you can see that the coverage of the Illumina is much lower once the AT drops. And by comparison, the nanopore short reads gives you much more even coverage. And again, this is just another indication of how fast we're moving. So our most recent study on the decapod crustaceans utilizes 256 mitogenomics, 90 that have been sequenced by our group. So we've gone from about 2015 to about 2019. And again, we've ended up almost trebling the amount of mitogenomes that we have available. So just to finish off, there is some concern that we are sequencing too many mitogenomes, and David Roy Smith has even gone as far as saying that there are so many published that we no longer need them. So I would argue against that because, yes, I don't think there's much value in just another mitogenome announcement, but I think we need to continue building up the databases because I believe that mitogenomics will eventually replace COI barcoding. And people might say, well, that's a big call, but I've seen the changes that occur in over 5 to 10 years. And I think with more focus on biodiversity assessments, the analysis of food products, then quite clearly the most common DNA you'll extract from environmental samples and complex food samples will be mitochondria. And that allows us to forensically identify species, but we can only do that reliably if we have sufficient large databases to allow us to do those identifications. So I think at this point, I'll say thank you. And of course, there are many people that have contributed to my research over the years, and I've had strong support from my universities, but particularly Dr. Gang, Dr. Tan, and Dr. Miller have been the students and staff that have made outstanding contributions to this research. So thank you very much.
Thank you. Thank you, Professor Christopher Austin. It was an excellent, insightful presentation on the mitochondrial use of mitochondrial information in genetics. It was rather elaborate presentation too on various applications of mitogenomics for environmental sciences. So we enjoyed traveling through your research process. We felt like we were through your research flow. I think there are no questions from the audience. So thank you very much, Professor Christopher Austin.
Yep.
Can I ask one question, Dr.?
Yes, sir.
Okay, Prof. Chris, thank you so much for your enlightenment. So I have one question. So when you are sequencing the mitogenomics using the NGS now at present MiSeq or HiSeq, so how many folds of the coverage of the mitogenome sequencing is actually recommended?
I would say between 50 and 100 is your sort of lower benchmark.
Okay.
And if you go back to that sort of slide, I emphasized the AT content. That
okay.
The thing is that can vary across the whole mitogenome. So whereas your coverage might be over 100 in some places, it will be down around 5 to 10 where there's high AT content. So I would actually aim for probably more than.
So when sequencing, is there any way we can exclude the main nuclear genome, or is there anything you are doing, or you're sequencing everything?
Not really. The nuclear fragments are just noise. But I mean, you might find just a shallow genome skim. You might find that 1 to 2% from mitochondria, which is enough to for a mitochondrial genome assembly. So as long as your nuclear genome is not too big, I mean, 1 GB or less is usually enough data for you to recover the mitogenome.
Okay.
And some people try to publish with coverage around 8 or 10, which is just not going to work. So, but Jenny, I mean, if you're interested in doing some of this work, then come and talk to me or my colleagues, and we can give you some advice.
Sure, sir. So one more fundamental when we are thinking the evolution of the mitochondria, which is included in the cell during the evolution. When you see the recent findings, the discovery of microRNA and the long non-coding RNA in mitochondria, which is more resembling the normal cell rather than the prokaryotic cell, where the small RNAs are there, absence of microRNA and long non-coding RNA. So how we can actually answer this, the evolution, our prediction of inclusive?
I'm not sure. I mean, the relationship between the mitochondrial genome and the nuclear genome is complex, because in the process of evolution, a large number of the genes that actually contribute to the respiration process are actually now being translocated to the nucleus and as functioning genes. So I think and maybe that exchange has been partly two-way, I'm not sure. I certainly don't have a lot of expertise on the non-coding side and the small RNAs, but I think it's just another intriguing question that is probably ripe for further investigation. I mean, I must say that my expertise is not so much around the function, it's mainly around the use of comparative information. But again, that's a really interesting fundamental question that I'm sure will be answered by increasing contributions to mitogenome sequencing and functional genomics.
Okay, thank you so much, Prof. Austin.
Thank you,
Okay.
Prof. Christopher Austin. Thank you, Dr. Suresh. So we'll
Dr.
move on to, yeah.
I have one question, can I ask?
Yes, sir. Yes, sir.
Yeah, Professor Christopher, it was a wonderful talk. I just want to ask one question because, I mean, as far as the gene therapy is concerned, so since you are working on mitochondria, I was just thinking, what do you think, how long it will take to use the gene therapy to treat the diseases, those are associated with the mitochondrial genes, you know?
I'm not sure. I really haven't been engaged in sort of human mitochondrial genetics and the disease associated with that. So I really would be talking outside my area of expertise. So as I said, most of my work has been comparative studies on animals, on different animal groups. So yes, I really can't help you with that question. I'm sorry.
Okay. Thank you.
Thank you, Dr. Subhash. Thank you, Prof. Christopher Austin, once again. So we'll move on to the next plenary talk. So over to Dr. Neeraj to introduce the next speaker.
Thank you, Dr. Annie. So as we are moving to the next plenary session, let me brief about this plenary session. This plenary session includes 2 plenary talks, each of 30 minutes, each followed by question and answer session at the end. So let me introduce you to our first plenary speaker, Dr. Fong Cheng Pen. It's my pleasure to introduce Dr. Pen. Dr. Pen is presently working as a research assistant professor in the Department of Surgery of Weill Cornell Medicine Institute, New York. Dr. Pen received her honors bachelor in molecular biology from University of Kebangsaan, Malaysia in 2000. She was accepted to join the International Masters and PhD program in molecular biology at George August University, Göttingen. Her graduate studies centered on the role of retinoic acid and BMP signaling in xenopus endoderm patterning and pancreas development. She joined Chris Wright Laboratory at Vanderbilt University in 2007 as postdoc fellow. She worked as senior research scientist for 2 years in Memorial Sloan Kettering Cancer Center in Stephen Leach Lab, wherein Dr. Pen generated a collection of pancreatic cancer organoid lines harboring common mutations found in human using the CRISPR technology. Her current research focuses on how metastasis is initiated in pancreatic cancer using the organoids and in vivo orthotopic transplantation models. In addition, Dr. Pen also established some protocols for directed differentiation of human pluripotent stem cells towards colon fate. and generated a panel of colon organoid that contained common mutation found in colorectal cancer patient for modeling and colorectal cancer. Apart from it, she has published numerous high-impact research papers in journals of international repute. Today, Dr. Pan will enlighten us with her plenary talk titled Human Embryonic Stem Cell Derived Colon Organoid as a Platform to Model Colorectal Cancer and COVID-19. Now it's over to you, Dr. Pan. Please initiate the session. Dr. Pan?
Can you hear me?
Yeah, madam.
Okay. Thank you for the kind introduction, and thank you for allowing me. It's my pleasure to talk in this conference. And I would like to express my gratitude, especially towards Professor Tang, who invited me to give a talk in this conference. And today I will talk to you about some of the research that is going on in our lab. So our research major focus is to use stem cell to manipulate stem cell fate using chemical and biological approaches to generate a variety of functional tissue and organs in vitro that can be used for translational studies such as diabetes, cancers, and the recent one, COVID-19 research. And today, I'm going to tell you how we combine the knowledge that we gain from developmental biology studies together with human pluripotent stem cell technology and 3D organoid culturing system to model colorectal cancer and COVID-19 and to find drug targets for these diseases. So just to keep everybody in the same place, same space, there are two types of human pluripotent stem cell. The first type is the human embryonic stem cell, which is derived from the inner cell mass of a blastocyst. So this blastocyst we normally obtain from in vitro fertilizations, and the inner cell mass will be extracted to generate human embryonic stem cell. There are other types of pluripotent stem cell called the induced pluripotent stem cell, in which they are derived from somatic stem cell, somatic cells such as skin cells or blood cells, which are reprogrammed by adding some reprogramming factor, mainly transcription factor. We call this the reprogramming factors, and they can reprogram these somatic cells into induced pluripotent stem cells. And this stem cell can give rise to any cell types of the bodies, as long as we know how these cells are developed in vivo. So this can be generate a variety of cell types. So the pluripotent stem cell has served as a very good platform to study the embryonic development of various human organs in a dish. And this wasn't possible because this study wasn't possible in human because there's ethical with obtaining the healthy human embryos. So human pluripotent stem cell will serve as an alternative to study those embryonic development. So apart from that, pluripotent stem cell can be used to study various genetic diseases. For example, patient-specific pluripotent stem cell can be generated where they carry a specific mutation, can be generated by reprogramming the skin or the blood cell of the patient, and then directed differentiation into the tissue or organ of interest that this genetic mutation is affected. And then from here, ideally, we could actually correct the mutation and to transplant this cell back to the body. But this is far from ideal because of the risk of the genetic engineering method, such as CRISPR-Cas9 that we use right now, which is not suitable for in vivo transplantation of this cell back to the body. But we can use this tissue or organ that is generated from this patient-specific pluripotent stem cells for studying of disease mechanisms, or we can also use this cell for drug discovery that can use to treat this treat the genetic disease of this patient. So we call this the precision medicine. So the organ of interest for me is a colon. So colon is part of the large intestine, which also contain the rectum and anus. And I'm interested in colon because there are several diseases that are associated with colon, such as intestinal bowel disease, colorectal cancer, as well as several pathogenic bacterial infections, such as those mentioned by Dr. Ravi earlier, like Vibrio cholera, Salmonella typhi, as well as several viruses that are affecting the colon that will cause diarrhea. So the function of colon is to reabsorb water and to process waste product of the body and prepare it for elimination. And the morphology of the colon is slightly different from the small intestine, in small intestine you have villi that's protruded to the intestinal lumen for absorption of nutrient during digestion. But for large intestine or colon, the villi is embedded in the tissue itself, and this is forming a crypt. So a single unit of the villi is called crypt in colon. So this is the schematic of crypt, which is embedded into the intestine tissue, and the crypt consists of 5 different cell types. The main cell type is the colonocyte, which is responsible for absorbing water. And there are also enteroendocrine cells, which secrete hormone and peptide in response to various stimulus, and is required for maintaining the physiological homeostasis of the body. And at the base of the crypt, there are colon stem cells, which will give rise to the transit amplifying progenital cells. And this cell can actually give rise to goblet cell, colonocytes, and enteroendocrine cells. And so in order to make colon in the dish, we have to understand how colon develop during development. So this has been studied quite extensively by developmental biologists using mouse model. And since mouse and human intestinal development are quite conserved, we can use the knowledge that we gained from the study in mouse to apply onto the directed differentiation of human pluripotent stem cell towards colon phase. So based on those study in the mouse, we and James File group in Cincinnati has actually generated a protocol to make matured colon organoids through a stepwise differentiation of pluripotent stem cell towards definitive endoderm, hindgut endoderm, colon progenitors, and at the end, the matured colon organoids. And this whole process takes about 48 days and use a combination of growth factors and chemicals for each differentiation steps. And as you can see, the human pluripotent stem cell is initially in the monolayer 2D form. And during hindgut endoderm differentiation, the 2D monolayer cells start to undergo morphogenesis and start to bud out to form small organoid, as you can see here. So when we connect this hindgut endoderm and culture them into a 3D matrix gels, this hindgut endoderm will eventually form colon organoids that you can see here over time. So these are one single colon organoid that contain the villus structure that I just described to you. This is one single crypt that I just explained to you. And we show that these are definitely colon organoid by showing that this colon organoid actually express intestinal marker CDX2 as well as colon specific marker SATB2. So this confirms that these organoids are of colon origin and not small intestine origin. So we also performed some single cell RNA-seq to see what kind of cell type that is inside this colon organoids. And it showed that majority of the cells in this colon organoid are stem cells, but we also found a lot of enterocytes, transient amplifying cell, goblet cell, as well as enteroendocrine cells in the colons. And this is further confirmed by the immunostaining of MUC2, which mark the goblet cell, as well as enteroendocrine cells. So this colon organoid actually has all the major colonic cell types. And so we can use this platform now to study to model various diseases such as colorectal cancer, as well as COVID-19. So the first part I will talk about my study when I first joined Cornell is to establish this colon organoid to model colorectal cancer. So colorectal cancer is the second most common cause of cancer death worldwide. And as in many cancer types, colon cancer is a genetic disease in which there's a progressive accumulation of genetic mutations over a long period of time. So mutation in this specific genes called APC, which is adenomatous polyposis coli, is the main driver of the colon cancer. About 85% of the patient who has colon cancer has 1 or 2 mutations in this gene. So when the colon cells start to accumulate APC mutation, they start to hyperproliferate and forming a small adenoma, which we call the intestinal polyps. As time passes, and these polyps accumulate more mutations such as KRAS mutation or loss of p53 or loss of chromosome 18q arm, they will start forming large adenoma and then progress to full-blown colon cancer at the end. So at this stage, the cancer cell is able to spread to other tissues. So because of this understanding of each mutation, the effect and contribution of individual driver gene mutation to the initiation and progression, as well as metastasis of colorectal cancer, is actually warranted in order to find a better treatment for this type of disease or cancer. So in order to model colorectal cancer, we use the human pluripotent stem cell derived colon organoid to model. So we generated 4 human pluripotent stem cell lines that carry a mutation for APC. So the first one is control, which do not have any mutation. And we also generated one with APC knockout, which is the main driver of colon cancer. And the third line is the APC knockout with the activating KRAS mutation, which is the second mutation that is found, that's most commonly found in colon cancer, as well as a 3 mutation in combination of APC, KRAS, as well as loss of p53 mutation. So we generated this mutation using CRISPR-Cas9 technology in the pluripotent stem cell line, not on the organoid line. And the KRAS and p53 mutation are actually inducible by dogs. So when we differentiate the pluripotent stem cell, this cell line only have APC mutation, which do not alter their capability of differentiating into colon organoid cells. And then once they reach the colon organoid stage, we will induce the expression of KRAS mutation as well as knock out p53 at that stage. And all the cell lines express a green fluorescent protein upon Dock's treatment. So this allows us to track the cells when we put this cell in the mice. So what we do is to differentiate this pluripotent stem cell carrying this mutation into colon organoid using a 48 days protocol that we developed. And then after that, we induce mutation of KRAS and p53 with doxycycline. And we first transplanted this colon organoid into the kidney capsule of a no-skid mice, which do not have T or B cell to avoid rejection of the graft. So we want to know whether this colon organoid actually are the colon cancer organoid. So to confirm that, we performed the kidney capsule transplantation. We also performed some RNA-seq to find out what regulator or pathway are specifically upregulated in each of the mutation to see if we can find drug target that will target those signaling pathway or regulators. So this is to show you that the organoid that we generated from each of these mutations doesn't differ in morphology. They look almost the same. And these are the human colorectal cancer organoid that we isolated from the patient. They look a little bit different because these are cultured in the different medias. But in general, the morphology for this colon organoid that contain the mutation are pretty similar with each other. So as I say, we first performed the transplantation to make sure that the colon organoid with cancer mutation are in fact a colon tuber organoid. So this is, so the transplantation will tell you that, so this is the control colon organoid transplanted under the kidney capsules. After 7.5 weeks and 10 weeks, they do not grow much, but then they maintain their expression of intestinal markers such as CDX2. And one good thing is they do not form teratoma. So the caveat of the differentiation is if you have cells that do not completely form differentiated into colon, they have a chances to form a teratoma, which is a type of cancer that contain all cell types. So we do not see any teratoma formation in the control colon organoid, which suggests that all the colon organoid are actually fully terminally differentiated. And when we transplant, the organoid contain the APC knockout mutation. You can see there's a huge tumor forming in this in the kidney of this transplanted mice. And this is similar with the morphology of this tumor is quite similar to the colon organoid that we isolated from the colorectal patient carrying the APC mutation. And also the morphology of this APC tumor is suggesting of a benign adenoma. And they express CDX2 and FP2, which is the marker that, as well as CK20, which is a marker of intestine, suggesting that they are intestine origin and they are colon tumor. And then we also transplanted the one with 2 mutations and with 3 mutations. And these are more aggressive than the APC alone, as most of the mice die between 5 to 7 weeks, whereas the one transplanted with APC can live up to 14 weeks. So these two mutation and 3 mutation actually are more aggressive and they cause death of the mice. But the tumor is slightly smaller than the APC mutation alone. And as you can see, they are more aggressive than, from the morphology, they are more aggressive compared to the adenoma that is seen in the APC mutation alone. And again, they express intestinal markers such as CDX2 and FB2, confirming that they are actually a bona fide colon cancer. So we perform RNA-seq just to see what pathway are upregulated in each of the genotype or in combination genotype. And we found that fatty acid metabolism is one of the top gene lists that is being upregulated. And it has been shown that in many cancer types, fatty acid metabolism are altered. And this is quite normal. And we also see that there are, the one with 2 mutation and 3 mutation has a different fatty acid, have different gene set being upregulated compared to the control as well as APC. And several genes that is involved in fatty acid oxidation are also upregulated in these 2 genotypes. So what we would like to do is to find a target that will target the fatty drug or inhibitors that can target the fatty acid oxidation pathway to see whether this inhibitor can reduce the survival of the cancer cells. So just a brief introduction to fatty acid oxidation. So fatty acid oxidation is a breakdown of fatty acid into acetyl-CoA, which can be used in the TCA cycle to generate ATP. So for fatty acid, which is normally generated in the cytoplasm, it needs to be transported into the mitochondria as fatty acid oxidation occurs in mitochondria. And so first, the fatty acid has to be converted into acylcarnitine by this enzyme called CPT1. And after converting into acylcarnitine, it will be transported across the mitochondrial membrane and then convert back to fatty acid acyl-CoA. And this is the rate limiting enzyme, and many inhibitor has been developed to target this when they want to target the fatty acid oxidation pathway. So CPT1, there are several CPT1 inhibitor available, such as paraxylene, ectomosel, and oxphenicin. And these are all FDA approved drugs. And there are not many CPT2 inhibitors available. One of them is the R-aminocarnitine. And apart from inhibiting CPT1 or CPT2, we can also target the fatty acid oxidation at the downstream, in which the fatty acid acyl-CoA is breaking down into acetyl-CoA by targeting the 3-ketoacyl-CoA thiolase inhibitor, 3-ketoacyl-CoA thiolase enzyme. This is trimetazidine is the only one that we can find that inhibits these enzymes. So because we have been using the paraxylene in the lab for pancreatic cancer research, and it works pretty well, so we started treating this colon organoid with this drug, the CPT1 inhibitor. So treatment of the control colon organoid, we do not see any cell death or minimal cell death with the paraxylene treatment. But when we treat the colon organoid with 2 mutation and 3 mutation with the paraxylene, as you can see here, there's a dramatic cell death in this colon organoid, suggesting that targeting the fatty acid oxidation in these two genotypes can actually reduce the survival of the colon cancer. And now we are actually trying to dissect what is the downstream mechanism of this paraxylene. Is it via reduction of ATP production, or is there an increase in accumulation of reactive oxygen species because there's a reduced production of the reducing power NADPH? So we are still studying that. And we are now switching gear to part 2, where we use this panel of colon organoid to model COVID-19 and also to find drug target that can target this virus infection. And this is done in a big group in collaboration with various groups from Weill Cornell Medicine, Columbia University, and Mount Sinai Hospital. So the first thing, the reason why we want to study SARS-CoV-2 infection in colon is that a lot of patients that is coming in, either they have a very severe respiratory symptoms. Other than that, they also have about 50 to 60% of the patients who came into the clinic will have gastrointestinal problems. So we asked if SARS-CoV-2 will in fact colon. So we isolate colon tissue from some of this COVID-19 patient. As you can see, this is a normal colon. And in the COVID-19 patient, we see that some of this colon tissue are being lysed. And we can find the SARS-CoV-2 RNA in the anterior site. And this is confirmed by the electron microscopy showing that the viral particles are found in the anterior sites, suggesting that SARS-CoV-2 can infect colon. So in order to use the colon organoid to model SARS-CoV-2 infection, we need to know if the receptor for SARS-CoV-2 is expressed or the processing enzyme of the spike protein is also present in the colon organoid that we use. So this is the single cell RNA-seq that I showed you before. So we look for ACE2, which is the receptor for SARS-CoV-2, and TMPRSS2 is the processing enzyme for spike protein, whereas furin is the pro-protein convertase that pre-activate the SARS-CoV-2 before it enters the cells. So we found that ACE2 is actually expressed in anterior site, highly expressed in anterior site, and some of the stem cell population. And TMPRSS2 is also expressed in the anterior site, as you can see here, and also found in other cell types of the colon organoid. And furin is also expressed in majority of these cell types. And this is confirmed by immunofluorescence staining of the ACE2 receptor, which is co-localized with the keratin 20 expressing anterior site or colonal site. So now we show that the receptor and the processing enzyme are being expressed in the colon organoids. These are SARS-CoV-2 able to infect this colon organoid? And the answer is yes. So we infect the colon organoid with SARS entry virus, pseudo-entry virus, and then we found that they are able to infect this colon organoid. As you can see here, the red is the virus and CDX2 is the colon cells. And the same for here is that SARS-CoV-2 is also detected, infection is also detected in keratin 20 expressing colonal sites. So now we show that SARS-CoV-2 are able to infect colon organoids. Can we use this colon organoid to screen for drugs that can prevent the SARS-CoV-2 infections? So we use this panel of organoid to screen for drugs, FDA-approved drugs that will prevent the entry of the virus into the cells. And the result came, we got 3 hits. One of them is called imatinib. Imatinib is actually an anti-cancer drugs that are used to treat myeloleukemia. And it has been shown it can also prevent the infection of other viruses such as MERS and SARS. Mycophenolic acid is actually immunosuppressive drug, and it has been used before to treat MERS and dengue virus as well as HIV virus. Quinacrine is an anti-malaria drug, which also is anti-helminth, anti-parasite, and anti-neoplastic. And it has been shown to also bind to MERS and SARS-CoV-1. And
Dr. Pong?
So, yes?
Dr. Pong, you have last 2 minutes to wrap up.
Okay. Yes. I'll be that soon. So when we use this drug to treat the cells that are infected, either we treat this organoid with this drug, either pre-infection with the virus or post-infection of the virus, we show that it can actually dramatically reduce the viral RNA expression in this colon organoid. And you can see that there's a reduced infection of this SARS-CoV-2 upon treatment with this drug, either pre-infection or post-infection treatments. and suggesting that they are working very well. We also confirmed this in the in vivo setting in which the colon organoid is transplanted in the kidney capsules and then infected with the virus and treat them with this drug, and it showed the same result and it's very promising. And we also generated lung organoid and tested the 3 drugs that we use, and it also works on lung organoids. So we also generated a panel of cells to study the tropism of this SARS-CoV-2 to see which cell type that SARS-CoV-2 is infecting. And as you can see, it is infecting cardiomyocyte, pancreatic endocrine cell, livers, and also dopaminergic neuron. So this human pluripotent stem cell can serve as a very good platform to model SARS-CoV infections. So what I've just tell you that we have used this human pluripotent stem cell derived colon organoid to as an isogenic platform to model colorectal cancer, and also to discover genotype-specific drug targets. And also this serves as a good platform to model SARS-CoV-2 infection and finding drug target that can prevent or reduce the infections. And to this, I would like to thank my mentor, Todd Evans and Shibing Chen, and members from both of the lab, and our collaborator in the Memorial Sloan Kettering Cancer Center, Columbia, Mount Sinai, and Weill Cornell Medicine. And the project from the colorectal cancer are funded by the Weill Cornell Medicine Surgery Department and the New York Stem Cell Foundation, and the COVID-19 project are funded by the Bill and Melinda Gates Foundation. Thank you very much.
Thank you, Dr. Pan. Thanks for enlightening us about embryonic stem cell derived colon organoids as platform to model the colorectal cancer and COVID-19. So Dr. Pan, we have received a lot of appreciation for this presentation. And there's a question, it's been raised by one of our member, Dr. Suresh. He appreciated your protocol for standardization for the production of organoid from pluripotent stem cells. So there's a query whether the pluripotent stem cells were taken from embryonic or induced pluripotent.
So in this case, they are human embryonic stem cell, which has been established for quite a while. And the line that we use has been approved by the US government. Has been used in many studies. And we have also used we have generated the IPS induced pluripotent stem cell from patient that has the APC mutation and use that to find drug target that can reduce the APC mutation or the proliferative effect of APC mutation.
There's one more question actually we received now. Have your research team made any comparison in terms of expression of the ACE2 and that of the spike protein processing enzyme between colon organoid and the other organoids such as lung organoid?
So ACE2 and TMPRSS2 and furin are expressed in the colon organoid and lung organoid, as well as in some of the endothelial cells that we generated in that last study that I showed you. And expression-wise, I would say it is highly expressed in lung organoid compared to colon organoid. But other than that, the virus can infect the colon organoid as well as the lung organoid equally efficient.
Okay, there's one more question. Is the effect of CPT1 or 2 inhibitors checked on normal cells or tissues?
So we have treated the normal cells or the control organoid, which has no mutation. Those are the normal organoid, and it doesn't actually cause any cell death, so which is very good.
Okay, with this, Dr. Pan, there is, I think, no other questions. So thank you very much for sharing your knowledge with us and giving your valuable time. So thank you, Dr. Pan. Thank you very much.
Thank you.
Thanks a lot. So now over to Dr. Annie to take up the next plenary session.
Thank you, Dr. Neeraj. We'll start with the next plenary talk by Dr. Rajendran from Madurai Kamaraj University. To summarize about his achievements, Dr. Rajendran is an assistant professor of microbiology at the School of Biological Sciences, Madurai Kamaraj University in Madurai, India. He has more than 15 years of teaching and research experience in microbial genomics. His research team has sequenced more than 200 bacterial genomes of industrial and clinical importance and have annotated. He has sequenced the whole genomes of several plant growth promoting rhizobacteria and identified the genes responsible for the plant growth promotion. He has also sequenced the whole genomes of more than 30 Brucella species and more than 100 Staphylococcus species. He has also created a dedicated genome repository of Brucella, namely Brucella base. He identified several genes coding for industrial enzymes and antibiotics from various metagenome sources. He profiled the circulating microbiome in cardiovascular diseases. To his credit, Dr. Rajendran has published more than 90 research papers in leading international journals. He is a recipient of ASM Indo-US Research Professorship from American Society for Microbiology. He has received Young Scientist Award from Association of Microbiologists of India, UGC Raman Postdoctoral Fellowship to work in USA. SBS MKU Genomics Award from Biotech Research Society, India, and Dr. Wilson Aruni Best Research Mentor and Teacher Award from Indian Association of Applied Microbiologists. So he has a very good record of academics and research. We would like to welcome Dr. Rajendran to deliver his speech on the prospects and applications of metagenomics. Welcome you, Prof.
Yeah. So thank you, Annie, for your nice introduction.
Thank you.
So, yeah, thank you. So today I'm going to share some of my research in the area of metagenomics. So my talk is scheduled in the area of prospecting from metagenomics, prospects and applications of metagenomics. So now shall I share my screen?
Yes, sir.
Yeah. So my title of today's talk will be Prospects and Applications of Metagenomics. So bioprospecting, so it's a general terminology used widely to understand the process of discovery and commercialization of new products from biological resources. In simple terms, investigation of a living organism in search of how they can be commercially useful, made it as commercially useful to humans. So conventional bioprospecting starts like this. You have a microbial community like soil microbial community or marine sediments or marine water or aquatic samples or something like that, or even gut samples from other insects or other animals. So you take some sample, community sample, maybe some soil sample, and then you isolate organism and then you cultivate, maybe growing the bacterial culture and then extraction of metabolites or extraction of enzymes, and then you do bioassays like looking for antimicrobial activities or antifungals or some enzyme, something like that. And then once everything is successful, and then you go for compound purification using PLC, proprietary HPLC or FPLC or something. And then after doing purified compound, having purified compounds or molecules, you go for structural elucidations using NMR or other sources, and then finally you develop some compounds of molecules with prospects for commercialization. But today we are in post-genomics era or omics era. So omics-based bioprospecting. So on the one hand, people are doing single strain isolation, but they couple with whole genome sequencing instead of going for conventional screening. They go for whole genome sequencing also, and then they will go for RNA-seq or metabolic analysis, differential gene expression, everything. And then they come for bioinformatics, cheminformatics, gene mining, visual screening, all those things. And then go for proteomics, glycomics, lipidomics, metabolomics, metabolic profiling, structural activity studies, refinement, and then functional elucidation. On the other hand, they take the entire microbial community and they go for community analysis, and they go for marker gene analysis like 16S, 18S, 18S typing, and then metagenomics, diversity analysis, and then from the metagenome they go for metatranscriptome or metabolome, everything. They go for bioinformatics, and then they come to once again for novel compounds. So there was a revolution in 1977. It's called the Woosian Revolution, done by Carl Woos, a chemist working in relative isolation and comparison of 16S rRNA sequences. So earlier people have tried to classify microorganisms, and of course they wanted to have a tree of life which could connect everything from bacteria to human being. So many people have attempted using phenotypic and biochemical characteristics. They could not achieve that. And in the 1977, Carl Woos made that. And then based on the analysis of 16S rRNA, or in general small unit ribosomal rRNA, he could make a beautiful tree of life where every organism, living organisms, could be connected with each other through the 16S rRNA sequences. And then he discovered a new kingdom called archaeobacteria. So now this is the accepted three kingdom concept, where we have bacteria and archaea and another kingdom in between eukarya, and then we have eukarya where animals and plants are there. So an universal and quantitative phylogeny is possible with 16S rRNA. So that's why 16S rRNA sequence analysis has made a huge advancements in microbial community structure. And in 1985, so there was a paradigm shift where a paper was published in Applied and Environmental Microbiology in 1985. First time they have shown enormous novel kinds of microorganisms are there in environmental samples. So they took some sample, soil sample or hot spring sample, and then they directly analyzed 16S and 5S RNA gene. So they have seen large number of unknown samples, and then that was the first time people could realize that in the environment there are a large number of newer kinds of organisms. And then that was a time in 1983 and 88, a PCR was marketed, developed and then commercialized. And then in 1977 itself, Sanger sequencing was available, and then it was the automated version were also introduced in the market. And then after the discovery of PCR and automated sequences, so we have got a large number of sequences, and then people have identified large number of unknown sequences from the environment also. And then the concept of metagenomics. The term metagenomics was coined by Professor Joe Handelsman from University of Wisconsin. So this was a first paper to frame the concept of metagenomics. We know about meta-analysis, like something like that, analysis of large number of research data and then combining data in the form of metadata analysis. Likewise, genomics of all the living organisms of a community are called as metagenomics. So metagenomics is a new area of phenomenal genomics. It gives unmined richness of microorganisms. So using various analysis, so people found that when they analyze see the microorganisms directly under the microscope, say for example, after staining with a stain like DAPI under fluorescence microscopy, they could count the cells. Similarly, when they have used culture-based technique using series of media, and then they have estimated the CFU, colony forming units. So the people identified there was a big plate anomaly, means estimating the bacterial diversity or microbial diversity directly under the microscope, and after culturing there was a huge gap. So most of the studies reveal that only 1% of the microorganisms are culturable. So people say that large number of microorganisms are unculturable, and then there was a debate. So we cannot say that unculturable. We do not know how to culture those organisms. So then people say that these organisms are not yet cultured, or not yet uncultured organisms. So metagenomics is direct access to, it gives direct access to all the microorganisms. So it gives a way of finding novel genes from directly from metagenome sample. Just to take sample and then extract DNA based on your, the fund you have, and then the ability of understanding, you can get large number of data out of metagenomic samples without knowing what is the source of organisms. So that is the power of metagenomics. Yeah, this is a recent version of a new view of tree of life, which includes 92 bacterial phyla and 26 archaeal phyla and 5 of eukaryotic subgroups, which is in the bottom. So this is archaeal group, and then this is a bacterial domain. So in majority of the groups, you can see a red dot. So red dots represents phyla without any culture representatives. All these informations are derived from metagenomic analysis. No organisms, that is more than 50 or 50 and 10 archaeal, or more than 10 archaeal, more than out of some 120, approximately 80 phyla are there without any culture representatives. So imagine the power of metagenomics. We do not have any organisms from these sources. Whereas there are other well-characterized organisms like firmicutes, bacteroidetes, where all these the well-studied organisms like E. coli and other pathogenic organisms are there. So functional metagenomics. So one aspect is diversity. Who is there in a microbial community or a microbial in an environment? So 16S rRNA or 18S rRNA, which can reveal eukaryotes, that will give you information on what organisms are there. And then using functional screening, functional metagenomics studies, one can see what are they doing in the environment. And then from the information derived from both who is and what are they doing, we can infer how they are doing it using some functional genomics and functional proteomics studies. Now, this is the functional metagenomics. So, which is widely used for the bioprospecting. So you take environmental sample, you take DNA, directly extract DNA from the environmental sample, you isolate metagenomic DNA, and then similarly you prepare restriction digested vectors like PUC or PBSK+, and then you ligate it and transform it in E. coli like E. coli TH10B, and then transform it. That will represent your metagenomic library. Based on your transformation efficiency, you may get up to millions of clones. And then those millions of clones can be screened for two ways. One is functional driven analysis. Say, for example, if you are interested in an antibiotic, you can do an antimicrobial screening. Likewise, if you are interested in some enzymes, you can go for enzyme-based functional enzyme assays. On the other hand, you have sequence-driven analysis, where if you know something about the conserved sequences, that can be used as a probe for either colony hybridization or PCR-based screening, and then you can identify clones that carries your target genes, and then you can, from the library, you can score the clones and then identify what genes are there. And interestingly, most often, more than 90% of the functional metagenomic studies are end up with identification of novel genes. So there may be a related genes, but it will have novel phenotypes, novel sequences, because, as I said in the previous slide, 90% of the organisms are unreported. So if you are tapping untapped microbial community, say for example, novel kind of organisms, you will end up with identification of very many novel organisms and very many novel genes. So we did a functional genomic study on marine metagenomics from near Andaman Nicobar Island. So we have collected this. So this is a water sampler. So we have collected from each sampler, we have collected 300 liters of deep sea water, say for example 50 meter to 300 meter deep sea water, and then we have concentrated, means we have filtered and then removed the water and then enriched the biomass. I mean, enrichment means we didn't culture it, so just we have concentrated the biomass. So on top of it, we will get the concentrated water with the rich biomass. So 300 water samples were made into 300 ml of water. From this, so we have extracted metagenomic DNA. So this is isolation of metagenomic DNA from the water. And then this is restriction digestion with partial restriction digestion with an enzyme called SOT3A1, which will cleave, it will form a smear like this. And then after this, we have extracted or sized only 2 to 10 kb fragments were extracted from the gel, and then we have extracted from the gel, and then this was used as a insert for making a metagenomic library. Yeah, we made a library in PBSK plus vector and PUC 19 vector, and then each white colony represent a recombinant clone, and then we have scored on a plate like this, and then likewise we have made some 50,000 clones out of metagenomic marine metagenomic library that represented metagenomic library. And this is a colony plasmid extraction and analysis, a rapid screening of the quality of your library. So if you found a varied insert size in your plasmid prep, then it means that your clones are carrying different kinds of inserts. So randomly we took some 15 clones, and then we analyzed the plasmid size, and then it reveals that the plasmid, I mean the library is good because it showed different sized inserts. So then we have screened all the 15 clones for a variety of enzymes and antimicrobial compounds or antibiotic activity. So we have obtained several enzymes like lipases, amylases, several enzymes, cellulases, various enzymes, and each of them were characterized separately. And then, however, we couldn't get any antibiotic, I mean antibacterial clone. There could be several reasons, because though the clones or genes might be there, so there are several reasons for not being expressed. So due to codon biases, or even the expressed antibiotic might be toxic to the E. coli host. That could be another reason. So therefore, we didn't identify any antibacterial agent, but however, we could find out one particular antifungal compound, I mean fungal clone, MMG, the library 74th plate E1 well. So this well showed some inhibitory activity against Candida albicans. We have screened all the library, all the clones against Candida albicans. Out of 50,000, we have identified one particular clone inhibited the growth of Candida albicans, and then we have scored this clone, and then we checked in a plate also, which inhibited Candida albicans in plate. This is control and this is test. Similarly, it also inhibited the growth of Aspergillus niger, and then which confirms the clones, E. coli clone carrying a fragment from marine metagenome have the antifungal activity. Yeah, and then we have identified the insert, so which had a 1.6 kb region, and then we have cloned we have sequenced the entire insert, and then we analyzed based on ORF finder. So here you can see a number of some 10 different ORFs and some of the partial ORFs. And then subsequently, individually, we have cloned and expressed individual possible ORFs in PET expression vector, where one out of these clones, a smaller clone from the +3 frame, I mean it is 111 base ORF, means 108 coding sequence, 36 amino acid coding peptide, was responsible for the inhibition of fungal activity. So we have identified a peptide from marine metagenome which could inhibit fungi. So we made a series of publications from this peptide, identification of a novel antifungal peptide with chitin binding property from marine metagenome. So we found that this peptide could bind to chitin and then enter into the cells. So through various biological activities, it inhibits the growth of fungus, both unicellular organisms like Candida or yeast and the filamentous fungi like Aspergillus niger. And then we found that this cell could directly penetrate into the Candida albicans using a series of experiments. We found that this peptide can directly enter into the cell, and then we have characterized the antifungal action, various mechanisms like inhibition of transcription, translation, and then it also had something like proteolytic activity in both in vivo and in vitro. So put together, so we have started with 300 liters of marine water, and then we end up with some library, and then after through a systematic fungal screening, we end up with 136 amino acids, which is having antifungal activity. So you can imagine the power of metagenomic studies, where from the huge genetic resources, you can identify several functional genes. So when we have blast search this peptide, so no related sequences were exist in databases. So some sequences, some minor aligned regions were there, but it's a novel region. We do not know the sources of organism of this peptide, but yet we have identified a potent antifungal peptide from metagenome. So as I said, there are several reasons for not being expressed a particular gene. A clone may be there, but the gene will not express. So only when it is functionally expressed, you can screen using functional assays like inhibitory assays or enzyme-based assays. But people have developed something like substrate-induced gene expression, where you have a promoterless GFP, and then you make library and then look for expression of the GFP under some substrate. So therefore, we can identify the promoter which is inducible by promoter. Therefore, the substrate. Thereby, you can identify several genes associated with the metabolism of the substrate. Say, for example, assume that there is some xenobiotic compounds like phenolic compounds. If you use phenol to induce the library, whatever the genes, promoters which are response to the phenolic compound, can be sorted out using FACS, fluorescent activated sorter, based on GFP expression. And you can analyze only those clones for analyzing some metabolic genes associated with your degradation of some phenolic compounds, something like that. Similarly, so there is product-induced gene expression. It's called PyGex, to identify genes induced by specific products. They, for example, so certain amylase genes are induced by amylose, that is starch, whereas certain amylose genes can be induced by maltose, which is a product of starch after the digestion with alpha amylase. So we made a construct with GFP, with lac promoter, with the cloning sites, PGX1, where we made a vector. We have confirmed that clones are inducible by GFP. So when this expression reveals that IPTG can induce expression of GFP, whereas if you make a library, metagenomic library, if the inserts are there, lac promoter will not work, but only when your promoter is induced with your substrate or product here, then you will have expression of GFP. So here, these are the FACS analysis. The elimination of clones. In the first step, we made a library. The entire library was passed through a FACS, fluorescence activated cell sorter, and then induced with IPTG. So these are the self-ligated clones, or with smaller inserts. These inserts are, GFP is responding to the IPTG. So these are self-ligated or self-induced clones. So these were eliminated. We have collected these clones and then induced these clones with 0.2% maltose, and then we again subjected into FACS. We can see 0.2% of these clones are showing signal. So we have collected these cells and then we have analyzed using routine plating assay. Within some 100 clones, we have identified the expression of GFP with induction of GFP with maltose. Here this is without maltose, this is with maltose, the same clone. And then we have identified the expression of GFP using fluorescence and FACS assay also. And then we have identified a novel alpha amylase, and then we have expressed and characterized, which was published in some journal. So we have identified a novel alpha amylase. So this is another way of screening your clone. So this is like innovative screening strategies like substrate-induced gene expression or product-induced gene expression. So likewise, we could identify several novel genes from various sources. And another interesting example is, say for example, microbes enables cows to eat grass. So cows or other ruminants, they eat, feed on grasses, but their genome, cow genome or other ruminant genomes, sheep and goats, they do not code for any genes for cellulases. So only the microorganisms, that is the rumen microbiome, play a major role in extracting energy from the diet from the grass. So likewise, we have a termite gut microbiome. So the termite assets, they do not code for any cellulases. They cannot feed on wood material or soil celluloses or plant material, which has cellulose rich or lignocellulosic rich diet. So the microorganisms in the gut, so they helps the termites to extract energy from the wood or the other soil sample. So termite gut microbiome were well explored by several institutes in US and other countries. Whereas on the other hand, you can see highly microbially rich sources is called termite mound or termitarium. No studies have reported except few on the microbial diversity of termitarium or termite mound sample. We did some study, one of my students has done some excellent study on termitarium microbiome. So we made microbial diversity analysis in termite nest. So here you can see the culture independent studies, that is direct metagenomic studies. So at the phylum level, we have identified several phylum. So whereas we studied the cultured study also, means we have cultured the bacteria from various sources. Yes, sir.
You have last 5 minutes, Dr. Rajendra. Thank you.
So culture dependent studies, where you can see large number of unknown isolates. So this picture tells you about, so here the each blue dot represents one 16S ribosomal RNA from the metagenomic sample, whereas you can see some red sample, red dots. So these are the representatives from the cultured studies, and then gray represents both culture, the sequence represented in culture as well as uncultured samples. So imagine the power of metagenomics. So each sample represents some living organisms from the sources, that is here the termitarium. So here you can see more than 99. Now we can accept my statement from some of the early studies. 99% of the organisms are yet uncultured. And then interestingly, the circle represents the similarity with existing sequences. So this circle represents 80, this is 85, this is 90, this is 95. Only these circles, these dots in the outermost ring represents the species associated with known organisms. So these are 80 means they are even, we do not know whether they belong to bacteria or archaea, but there are huge number of samples. So similarly, we compared the samples of termite nest and termite gut. So termite nest has a unique microbial population. And then we did some functional metagenomics also, means we have directly sequenced everything. And then we have identified several carbohydrate GH families, I mean cellulolytic carbohydrate active enzymes. Several. So all these are acting on cellulolytic organisms. And then this is the biosynthetic clusters associated with polysaccharide utilization loci. All these were, majority of them were novel sequences. There were no homologs are there in, of course, homologs are there. The sequence similarity are very less. And then we know about the human microbiome also. So there were several studies focusing on human microbiome. So this was an excellent paper where people have correlated obesity and the microbial composition. Similarly, there was another interesting question like whether the blood of healthy individual contained bacterial ribosomal DNA. That was a question asked in even 2001 itself, and the answer was yes. So this is a control sample. This is a healthy blood sample. There are 16S RNA sample. So likewise, we have characterized some circulating microbiome from cell-free DNA from the peripheral blood. So we have extracted blood based on other sources. You have microbial sources. So we have identified, we have profiled the microbiome of plasma, that is circulating microbiome of cardiovascular disease patients and the control. So we have identified that this is 3 groups of cardiovascular diseases and then control. If you see firmicutes and proteobacteria, there is a huge difference, statistically significant differences are there in the microbial diversity of cardiovascular disease patients and control. Similarly, we have seen elevated levels of circulating DNA, and then this could be developed as a biomarker for earlier diagnosis of cardiovascular diseases. So with this, I thank the organizer for giving this opportunity. I thank my research scholars who did various works on this, and then various funding agencies, CSIR, DST, DBT, SERB and UGC, and the central facilities of our school in MKU for providing all these opportunities. I'm happy to answer if you have any questions. Thank you all.
Thank you, Dr. Rajendran. It was a wonderful presentation, informative on metagenomics. We have 2 questions from the audience. The question 1, what is the current phylogeny cutoff for new or novel bacterial strain on the basis of 16S RNA?
Yeah, 16S RNA based on it is 97% sequence similarity, but that will not be sufficient. We have to go for other studies like DNA hybridization. It should give less than 70%, something like that. With respect to 16S RNA alone, it is 97%. We call them as operational taxonomic units. So if it is showing 97% sequence similarity, we can say that this is species level OTU. But we cannot claim as new species, but it has to be evidence with, it is one of the criteria, it has to be supported with other evidences.
The next question is, how can I go for a metagenomic analysis for newly isolated anaerobic bacterial strain from bioreactor?
Yeah, if you isolated organism, it is not metagenomics. But you can do some microbial, total microbial studies from the bioreactor itself. If it is a microbial community, there you can go for metatranscriptome analysis, or even metabolomics are possible. You can study the entire metabolic products. Similarly, if you are doing the same with individual organisms, it is functional transcriptomics or proteomics.
Okay, thank you. And one more question, which is the best method for analyzing microbial community composition?
Yeah, 16S RNA sequence analysis based microbial composition analysis are widely accepted, but there are bias. Even from my studies, when you go for 16S RNA analysis or shotgun approach, total metagenome shotgun approach, don't do anything, just take DNA and then share it and then make a library and then do shotgun sequence analysis. From the shotgun sequence, you can go for 16S based microbial diversity analysis, because whole metagenome analysis avoids the biases associated with PCR or the PCR amplification bias. So 16S rRNA based analysis is acceptable, but to have total microbial composition without any bias, now people are recommending whole metagenome shotgun sequencing.
Thank you, Dr. Rajendran. It was really nice presentation from you. So over to Ms. Sarah.
Yes, doctor. Thank you so much, Dr. Annie and Dr. Neeraj, for chairing the session. And thank you to all the keynote and plenary speakers for sharing your work with us. It is now time to move on to our first oral presentation session of the day. The chairperson is Dr. Annie Jaya Christie and co-chair will be Dr. Neeraj Floria. They will also be serving as the panel of evaluators. Over to you, Dr. Annie.
Yeah, over to actually Dr. Neeraj will take up the first part of the session.
Sure, doctor. Okay. Over to you, Dr. Neeraj. Are you there?
Yeah, I'm there. Okay.
Sure. Okay.
Thank you, Ms. Sarah. So let's start with this oral session now. To brief about this oral presentation session, there will be 6 oral presentations, each of 10 minutes, where at the end of each presentation, there will be a question and answer session. So with this, we are going to invite our first speaker, Mr. Aisamuddin Ardi Jainal Abidin, with presentation code 017. Mr. Aisamuddin is from Department of Biochemistry, University Putra, Malaysia. Today, his presentation title is Stability of Transgene in Nanochloropsis Species, Potential Vaccine Delivery System to Fish Against Fibrosis Disease. So over to Mr. Aisamuddin now. Mr.
Please
Aisamuddin.
unmute yourself.
Hello, can you hear me?
Yeah, we
Yes,
can hear you. Please.
Thank you.
Yeah.
Good afternoon to everyone. So my name is Aisamuddin. So I'll be presenting a simple part of my master project entitled Stability of Transgene in Nanochloropsis Species, Potential Vaccine Delivery System to Fish Against Fibrosis Disease. So, so as we all know, aquaculture feed the world. So in 2030, it is estimated that 62% of the fish of the world will be provided by aquaculture. So aquaculture mainly are cultivation of marine organism. So in Malaysia, aquaculture is not about contributing to the economy, but it also produce commercial products from them, restore aquatic habitat and rebuild populations of endangered species. However, aquaculture, the major challenges is the disease, which decrease the production and the economic value. So it is over the past decade, about 45 billion were lost due to shrimp disease, and 20 billion were lost in the salmon industry. So one of the major diseases is fibrosis, which is involved in fish species. So it is caused by gram-negative bacteria, by the Vibrio genus species bacteria. It occurs to more than 14 countries and have been infected up to more than 48 species of marine fishes. So in Malaysia, about 7.4 million were lost in 1990s, and in 2015, an accumulation of 1 billion were lost in Asia and Central America. So the symptoms of Vibrio we can see here. There are skin ulcerations, rotting of fins, pigmentation on the body of the fish, and hemorrhage on the liver, kidney, and spleen of the fish. So actions have been taken to control the disease, such as biocontrol by strict quarantine measures, sanitation, and disinfections. We also have antibiotics. The antibiotics is, however, will not be a primary treatment option because it may develop in antibiotic resistance bacteria. So studies have found that vaccination is the most effective method. It reduces the application of antibiotics. However, most vaccines are injected to fish, which is costly. It is stressful to the fish, which reduces the efficiency of the vaccine. It's labor intensive, and you cannot introduce the vaccine to fish at larvae stage where their metabolism is at the peak. So in this study, we propose to utilize microalgae, which we can found in freshwater and marine system, to be a vaccine carrier, which we could introduce the vaccine through oral vaccination by feeding. So the key features that we chose microalgae is that they have a rigid cell wall, which can protect the antigen by the acid of the fish gut and deliver the antigen to the organs that induce the immune system of the fish. It is unseasonal, so we can grow microalgae throughout the year. And we can studies have found that we can preserve recombinant protein in microalgae up to 6 months. So in this study, we will be utilizing Nanocropsis species due to its small size and as I mentioned before, but the main key point is that it can be fed to fish at larvae stage due to small size. So transgenic Nanocropsis species have been developed in our university. So we currently have 6 transgenic lines, which varies based on the vector used. So it carries an OMPK protein, which is an outer membrane protein kinase, which I have found to develop immune system in the fish against Brevio species. It also contains bleomycin resistance gene as a biomarker, and it deficits gene nitrate reductase. So there are 3 vectors, main vectors, which is VCP OMPK ZX1, VCP OMPK ZX2, and CRISPR nitrate reductase gene vector. So the vector varies based on their way of transforming into the genome. So here, these 2 vectors are inserted into the genome via homologous recombination, as we shown here. So the homologous recombination is the way of transforming him. And the second part is the CRISPR vector. So normally CRISPR alone, it nick out some sequence to develop knockout of a specific gene in the genome. So, but in our study here, we combine CRISPR and the homologous recombination vector so that when the CRISPR knock out a specific gene, our homologous recombination vector will act as a template for repairment and introduce the expression vector into the Nanocropsis genome. So as I mentioned before, so we have problem with we have fish diseases. So vaccination is the effective way, however expensive, time consuming. So we propose transgenic microalgae. However, how stable is the foreign gene inside our transgenic Nanocropsis? And we want to see how long that will it be stable inside our Nanocropsis. So the objective is to verify the stability in the genome and to see whether it expresses the gene transcript. So an overview of the methodology is that first we cultivate it at small volume, and we start harvest them at the 4th generation and upwards. So from the harvesting cultivation, so we'll be extracting DNA and RNA, and we will perform PCR to verify the presence of the gene. So for the results and discussion, so firstly we extract the DNA, and for the 4th generation we get all transgenic lines were presence of OMPK gene. At generation 5 we have found that only 4 transgenic lines were present, and at 6th generation we can see that all generation have not doesn't have the OMPK gene. And so for the transcript, we extracted the RNA, and we can see here at 4th generation, only 1 transgenic line did not express the OMPK gene. And at the 5th generation, we can see only 4 transgenic line express the OMPK gene. So we can say here that the stability of the foreign gene inside the DNA of the genome of the Nanocropsis were detected up to the 5th generation, which is approximately 12 months. And however, CPV1 and CPV2 were unstable and only stable up to F4 generation. For the expression, we can see that at generation 4, it was expressed except CPV2, and at generation F5, only 4 generations were expressing the OMPK transcript. So in conclusion, so it is the nanocopsis transgenic lines can be maintained up to 5 generations after the formation of these colonies, and nanocopsis were able to express the OMPK gene. So for future recommendation, we should look into the expression of the protein to see whether the antigen is being produced and folded appropriately, similar to the Vibrio species. So here is my references, and I would like to acknowledge my supervisor and co-supervisors, and that's all from me. Thank you.
Thank you, Mr. Asamuddin. It was a really informative presentation. So may I ask the participant to ask the question?
All right.
So one question for you, Mr. Asamuddin. In your opinion, can we retain the transgenic line by including the selection antibiotic?
Yes, I think we can also screen out whether it is still transgenic by screening antibiotics using bleomycin. However, we also want to see, probably because the antibiotics are run by different promoters, so we want to see whether OMPK is also being expressed or not.
And it's from my side. How did you optimize your this system?
Actually, the vectors and everything are still being filed for patent. So the information on that.
Okay, no worries. It's one more question. If it only retained for 5 generations, how do you apply this in real life? Is it sufficient for effective treatment?
Yes, we can. The generation is based on, we can culture them in a bioreactor. So in 5 generation, so we can get a lot of biomass by up to 12 months in a year. So I think, yeah, it's applicable.
Okay, I think, I think with this, there is no other question. So thank you, Mr. Asamuddin. Thank you so much.
Thank you.
So with
Thank you
this,
so much,
we'll move on to the next presenter. So thank you.
Thank you.
So moving on to the next presenter, Ms. Bharati Chaudhary, with presentation code 018. Ms. Bharati is from Mandsaur University, India. So today she'll be presenting her talk over a validated UPLC method for simultaneous estimation of diacerin and aciclofenac in bulk and pharmaceutical formulation. So over to you, Mr. Bharati. So please continue the session.
Yes. Thank you, sir.
Yes, please.
Yes, sir.
Yeah, Mr. Bharati, please share your screen.
Yes, sir. I have already shared.
Yeah. Please continue. Yeah. Thank you.
Okay. Thank you, sir. Hello. Namaste to all. I am Ms. Bharati Chaudhary from Mandsaur University. I am one of the PhD scholars from Mandsaur University, Mandsaur, India. First, I congratulate to AIMST University for the successful organization of this international conference. And I am thankful to the organizing committee for giving me such a wonderful platform to present our work. And here today, I am going to deliver my topic, that is a validated UPLC method for simultaneous estimation of diacerin and aciclofenac in bulk and pharmaceutical formulation under the guidance of Ms. Dr. Kritika Daniel. So coming over to our next slide, that is in my presentation, my contents are introduction, experimental work, result, discussion, conclusion, acknowledgement, and references. So turn over to the introduction. Here in this work, I use two drugs, that is diacerin and aceclofenac. Both are anti-inflammatory drug. Diacerin, it is also called as diacetyl-rain. Chemically, it is called as 1,8-diacetoxy 3-carboxy-anthraquinone, which is one of the novel anti-inflammatory drug used in the treatment of osteoarthritis. And aceclofenac, the another drug aceclofenac, which is chemically 2,2,6-dichlorophenyl amino phenyl acetoxy acetic acid, which is one of the phenyl acetic acid derivative, and it is also an analgesic drug which is used in a variety of painful condition. These two drugs are available in market as a single dosage form and even in combination, but in combination it is very useful. So in commercially it is available as a tablet in a dose of 50 milligram diacerin and 100 milligram acetofenac, which is used in osteoarthritis. But from the literature survey, it was found that for the determination of diacerin and acetofenac, only UV spectroscopic method, some HPLC method and HPTLC methods and colorimetric methods are available, which are already reported. Here I have given that reference for that. So from this literature study, after performing this study, I note that no one UPLC method was there. That's why here the aim of my work is a simple, accurate, precise and highly reproducible analytical method, that is a UPLC technique, was developed and validated according to the ICH guideline for simultaneous estimation of diacerin and aceclofenac in bulk and pharmaceutical formulation. So coming over to the experimental work, here in experimental work, first two solution of these two separate drugs, that is diacerin and aceclofenac, the working standard solution, that is 5 microgram per ml for diacerin and 10 microgram per ml for aceclofenac, was prepared, and the separate solution for the marketed preparation, that is a separate sample tablet solution, was prepared. Different trials have been taken for this study, and for that determination, the chromatographic condition, that is isocratic method, is applied for this technique. And the column means that UPLC instrument, where column is UPLC BH shield C18 having dimension 50 and 2.1 mm and 1.7 micro mm of particle size. Detector used for that technique, UV PDA detector. The software used for this technique, Empower software, and the mobile phase. By performing different trials, I got that water and these two combination of mobile phase is used. The detection wavelength 268 nanometer and flow rate 0.4 ml per minute, 40 degree centigrade temperature, and sample size was maintained. And after performing this work, I got the separate standard curve and sample curve. After performing that successful, I went for validation. In validation, different parameters have been checked for accuracy, precision, LOD and LOQ, linearity, robustness. For accuracy, 3 different concentration, 80%, 100% and 120% was taken. Under precision, interday precision and intraday, both were performed. LOD and LOQ was find out. Linearity, the proper concentration, that is from 2 to 17.5 different concentration were prepared and linearity was found. Robustness was also very good. So with this experiment, now the result, that is this one is the UV overlay spectra for diacerin and acetofenac. From these overlay spectra, I got the proper lambda max value, that is wavelength was selected at 268 nanometer, and these are the two chromatogram obtained for standard preparation and test preparation. And from these chromatogram, the result obtained was, that is The retention time for diacerin, it was obtained 1.762 minutes, and for aceclofenac 2.892 minutes. These two retention time I obtained. That is the successful result obtained from my work. And after completing this result, the validation parameters which I was obtained, the accuracy from this chart, you will get idea that after performing this accuracy study, that is at the three different levels, that is 80%, 100%, and 120% sample were run. So the RSD value, that is the percent RSD value, whatever I got, it is less than 2%. That means it is the acceptance criteria for my technique. And for precision, the interday and intraday precision result was obtained. Here again, the percent RSD value obtained was less than 2%, 0.2%. And the LOD and LOQ obtained for diacerin and aceclofenac, that is 2.98 microgram per ml and 5.9041 microgram per ml, and LOD, that is 0.9841 microgram per ml and 1.9467 microgram per ml I obtained. So this is the result for accuracy, precision, and LOD LOQ parameters. Now come over to the linearity study. Here, after performing the linearity study, I got result. That is, from this graph, we can observe that the straight line is obtained. That is, while concentration is increases, the area under curve also increases. That means this method was linear for diacerin as well as for aceclofenac. And robustness. For robustness, here I perform this technique under robustness by changing flow rate, mobile phase, and wavelength. For flow rate, I have changed, that is addition of 0.1 and 0.1 less. For that means flow rate and mobile phase and wavelength. From all these things, I got the result means in percent RSD value, it is less than 2%. That is appreciable for this study. So from this study, as the discussion here, in the present study, our aim was to develop a simple, accurate, precise, and highly reproducible analytical method. Yes, sir.
Ms. Bharti, you have last 2 minutes to wrap up. Thank you.
Okay. Literature survey reveals that some of the method, that is HPLC and HPTLC methods, where the result was found to be more than 5 minutes. But in this proposed method, the retention time was found to be less than 2 minutes. And this result obtained was a satisfied one. And all the acceptance criteria as per the guideline were obtained from this method. Now conclusion. For conclusion, an isocratic UPLC method was developed and validated for diacerin and aceclofenac in bulk and formulation. This is found capable for giving quick, precise, and accurate retention time and good resolution as compared to the conventional method, and validation parameter also satisfied, and that's why it is easy to adopt for routine analysis of diacerin and aceclofenac. So the importance for my work is that here this developed method is the first UPLC method where simultaneously we can determine diacerin and aceclofenac, and this is the first fastest method forever. And I'm so here my acknowledgement, that is I am thankful to the management and Dean of Mansoor University for providing me all the facilities, my guide Dr. Katika Daniel, and all the supporting staff, my family members and friends. Here are the references for my work. Thank you, sir.
Thank you, Ms. Bharti. It was a really wonderful research what you have done, I could see, because in this you have taken a time is within 2 minutes you are analyzing the sample. So is there any other technique apart from this UPLC which claims like the time taken is less than 2 minutes?
No, sir. No one method I found for that. All the methods of that timing is more than 5 minutes. Means
so what is the significance of your study?
That here within less time we got our result, and the accuracy is also very perfect, and all the parameters which I got from validation that are in acceptance criteria, and that are all as per the guidelines.
Okay, good. That's very nice. Thank you. Thank you so much. So I believe there is no more question from the audience side. So if at all any question, maybe you will be receiving later
Okay.
through your Zoom or YouTube or Facebook. Thank you so much. So thank you so much, Ms. Bharti. Thanks a lot. So with this, we move on to our next presenter, Ms. Fatma Diana Binti Muhammad Bukhari, with presentation code 019. Ms. Fatma is from Faculty of Medicine, University of Malaya, Malaysia. Ms. Fatma will deliver a talk titled Binding Activity of Plasmodium knowlesi Duffy Binding Protein Alpha Region 2 corresponding to high and low pericythemia isolates. Over to you, Ms. Fatma. Ms. Fatma, over to you.
Hi, everyone.
Yeah. Please continue the presentation.
Okay, thank you, sir. So everyone can hear me clearly?
Can hear.
Yeah.
Yes, we can hear.
Okay, all right. A very good day to everyone. I'm Fatma Diana from Department of Parasitology, University of Malaya, Kuala Lumpur, Malaysia. So today I will be presenting our study entitled Binding Activity of Plasmodium knowlesi Duffy Binding Protein Alpha Region 2 corresponding to high and low parastemia isolates. Okay. So over the past decades, malaria has become a life-threatening disease which affects millions of people around the world, especially Africa, South America, and almost all countries in Southeast Asia. According to Malaria Report 2019 by World Health Organization, 2 to 8 million of malaria cases were estimated in 2018, causing about 405,000 that were reported. Malaria caused by a single cell organism parasite of genus Plasmodium. So far, there are 5 Plasmodium species that are known to infect humans, which are Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, and also Plasmodium knowlesi. Plasmodium knowlesi, which previously known as simian parasites, now has been reported among humans in all Southeast Asian countries. Statistics in 2004 till 2016 showed Malaysia presented the highest number of knowlesi cases with 85.78% cases reported in Malaysia, Borneo. Even though Malaysia reported 0 indigenous human malaria cases for the first time in 2018, Malaysia currently facing a dramatic increase in zoonotic knowlesi malaria cases. In general, malaria causes infection in humans through proliferation of a sexual blood stage parasite called merozoite in red blood cell. One of the crucial parasite ligand of Plasmodium knowlesi is known as PKDBP alpha 2, and it binds to Duffy antigen receptor for chemokine or DARC on erythrocytes. PKDBP, which located on merozoites, can be found in 3 distinct forms of large protein, which are alpha, beta, and gamma. PKDBP beta and gamma are only selective to bind to macaque erythrocytes, whereas PKDBP alpha can be bind to both macaque and human erythrocytes. For each form of this large protein, it contains it can be divided into 7 different regions, and region 2 is the one that contains critical binding motif for binding to DARC. Plasmodium knowlesi has a short erythrocytic cycle, which is 24 hours cycle, and this quick replicating pattern can contribute to hyperemia and possibly severe malaria. Previous literature suggested rosetting as a marker for progression of severe malaria, and this phenomenon usually associated with severe falciparum and somehow can be observed in vivax cases. Erythrocyte binding assay or EBA is usually performed to characterize the binding ability of malaria parasite ligand toward erythrocytes. Positive binding is evaluated by the detection of rosette, which is the aggregation of erythrocytes surrounding the mammalian cells expressing the protein ligand. So previously, distinct geographical distribution of PKDP alpha 2 was found between Peninsular Malaysia and Malaysia Borneo. Even though high number of Plasmodium knowlesi cases were found in Malaysia Borneo, but then the binding activity of PKDP alpha 2 was higher in Peninsular Malaysia compared to Borneo isolate. Lim et al. 2017 suggested that binding ability could be associated with virulence level and multiplication rates of Plasmodium parasite instead of geographical factor. So this study will determine whether the differences in parastemia level of Plasmodium knowlesi malaria associated with binding activity of PKDP alpha 2 towards erythrocytes. And in this study, we hypothesized that high parastemia isolates will demonstrate better binding activity with erythrocyte compared to low parastemia isolates. So moving on to methodology, 4 samples, 2 high and 2 low parastemia isolates were chosen from our archive. Samples with 27% and 0.4% were classified as high parastemia group, whereas 0.04% and 0.001% were classed together as low parastemia group. So DNA from each sample was extracted using extraction kit. So prior to cloning, PKDP alpha 2 gene was amplified using specific design primers. And then this gene was cloned into 2 different vectors. The first one is PGMT vector. After that, the nucleotide sequence
Miss Padma, we cannot hear you. Miss Padma, we are not able to hear you. Miss Padma, are you there? I think some connectivity problem.
I think it might be a network issue.
Miss Padma, are you there?
I think there's some connectivity problem is there.
Yes, sir. So shall we go to next one?
I
sir?
think we'll go for the next presentation, 20. So now it is
Okay,
over
sir.
to Dr. Annie. So Dr. Annie, you please take up the next session now.
Thank you, Dr. Neeraj. So the next presenter is Mr. Lee. Mr. Lee.
I think we cannot find the next speaker.
Okay.
We need to skip that too.
Next speaker is also not in the
Ms.
virtual room.
Maheshwari. Next is oral presentation 21, Ms. Maheshwari.
Hello.
Yeah, Ms. Maheshwari.
Hello.
Yeah, is it
Yes,
Ms.
ma'am.
Maheshwari? Okay, okay. So we'll
audible
move
to
on
you?
to, yeah, you're audible. We'll move on to Ms. Maheshwari. Ms. Maheshwari, your presentation number is 021. Ms. Maheshwari is from the Department of Biotechnology, Jain University, Bangalore.
I'm
Hello?
audible. Am I
Yeah, there
audible,
is
ma'am?
some second voice in your.
Now, ma'am.
Yeah.
Hello.
Still. There's an echo from your side. Are you using 2 devices?
No, ma'am. No.
Okay. Okay. Okay.
Am I audible, ma'am?
Yeah, you're audible.
Okay. Can I start, ma'am?
Yeah, thank you. You can start, Ms. Maheshwari. Your time starts now.
Yeah, thank you. Thank you. I'm Maheshwari from Jain University Department of Biotechnology from Bangalore, India. So the topic which I would like to discuss here is anti-diabetic effect and antioxidant potential of green marine seaweeds, which is collected from Gulf of Mannar. 2/3 of world with ocean. whose upper layers are inhabited with the algae. So India has got almost 8,000 kilometers of clusters of marine habitats like intertidal region, muddy shores, mangrove forests. But 70% of this is, yeah. Hello.
Please can you adjust your camera? We see only your half page. So maybe you can just adjust your camera, please.
Now, sir?
Little bit down. You can push it down a little bit. Now better. Still some
Fine,
face
sir?
is missing. No, the upper part of face is missing.
Yeah.
Little bit down. You can see in front of you, so you can adjust.
I can see, but is that
Upper
fine,
part
sir?
is missing. Upper part of your, I think if you bend down, now it's better. Yeah, please go ahead. Sorry. Please go ahead.
Okay.
Yeah, thank you.
Yeah, thank you.
Thank you.
So the potential of Indian marine habitat has remained largely unexplored for its potential new drugs. So our concern is about the marine seaweeds, marine macroalgae. So algae generally they are classified into marine microalgae and macroalgae. So our concern is about macroalgae, which is scientifically called as marine seaweeds. So here, these are the plant-like organisms which get attached to the rock or pebbles or even tires, and they grow in the intertidal, shallow, and deep region. So based upon the depth of the seawater, different division of algae will grow. So this is classified into red algae, brown algae, and green algae, like rhodophyceae, phaeophyceae, and chlorophyceae. So this is classification is mainly dependent on the pigmentation. So the pigmentations which gives not only the color, but it also plays an important role in the biological activity. Like the red seaweed, it gives a bright color because of the presence of phycobilins, phycoerythrins, and these phycoerythrins are also having an antioxidant potential, anti-diabetic, and anti-cancer. So many effects are there. So I have listed here some of the active compounds which are of greater importance in the pharmaceutical industry, like the polyphenols, like the PUFAs, polyunsaturated fatty acids. So this is extensively used for anti-obesity activity, and like flavonoids, phenols, terpenes. Terpenes especially, there are a lot of compounds which are derived from the terpenes, and it is of greater importance for anti-cancer property in the pharmaceutical industries. So here, coming to my topic, so what is the role of these marine seaweeds in managing the oxidative stress related diabetes? So oxidative stress is the term which refers to the balance or the imbalance or the disturbance between the oxidants and the antioxidants. So here, which means that the reactive oxygen species or reactive nitrogen intermediates like your superoxide, hydroxyl, hydrogen peroxide, hydroxyl radical. So when this is upon accumulating in your system, obviously it leads to the cellular breakdown of your critical macromolecules like your protein, fats, DNA. So this balance should be maintained, and this is being maintained by this antioxidants like so-called this flavonoids, carotenoids, terpenes, whatever I have mentioned here. So how this is going to minimize this how this is going to react with the diabetes patient is, so diabetes, it is already known that it is a metabolic disorder which depicts the high the high level of blood glucose, and this is leading to the glucose intolerance, or it is or it is a deficient in using up the glucose or the insulin which is present in the system. So here we can see because of the two enzymes like the alpha amylase and alpha glucosidase, this functions as it catalyzes in the hydrolysis of alpha 1,4 glycosidic linkages and degrades this, and further whatever is degraded or hydrolyzed by the alpha amylase is being degraded by the alpha glucosidase into from oligosaccharides or polysaccharides into simple sugars. So obviously what happens is whatever the insulin which is present or the glucose which is present is, and the system is unable to utilize it, and also on top of that it is getting accumulated because of these two enzymes. So our objective is to inhibit these two enzymes. So whether the marine algae which we have choose is inhibiting this or not, that is the concept. So we have designed our research in such way that we have collected the seaweeds, and it has been authenticated, and it is the qualitative and the phytochemical profiling we have done, and antioxidant activity we have done total antioxidant activity by phosphomolybdenum assay, ABTS and metal chelation, and for anti-diabetic we have done alpha amylase inhibition assay and alpha glucosidase inhibition assay. So the results shown here is
these are the 4 different algal species, green marine seaweeds we have collected: Chaetomorpha crassa, Cholera racemosa, Cholera verticillata, and Cholera scalpeliformis. So among all these 4 algae, Cholera verticillata has shown a high yield of flavonoid content, and most of the scientific community have accepted that the flavonoid content which is produced by the marine seaweed is more unique and more strong, showing a defense mechanism against when compared with the plant, terrestrial plants. So this antioxidant assay we have done ABTS, TAC, and metal chelation. So among all these assays, what we have, and these 3 assays I have done with 5 different concentrations, and from the results, what we can observe is the one which have given a high content of total flavonoid, that is the Cholera verticillata. Again, it has reflected in the antioxidant property. Like Cholera verticillata here, it has given a maximum of 96% in ABTS scavenging activity, metal chelation, more than 70%, and then also it is a very good antioxidant, total antioxidant potential in the TAC assay. And here I have summarized the total phenolic content and the metal chelation with metal chelation, ABTS, and phosphomolybdenum with the IC50 value. So here it clearly states that this Cholera verticillata is a very good choice for antioxidant property. So how is it going to interact with the so-called 2 enzymes, alpha amylase, and whether this Cholera verticillata is enough stable enough to inhibit these enzymes? Yes, obviously. So alpha amylase inhibition assay and alpha glucosidase inhibitory assay shows that the Cholera verticillata is inhibiting almost more than 70% of alpha amylase, and also almost nearly 70% of alpha glucosidase inhibition is also observed. So here I have summarized with the IC50 value. So comparatively, all the 4 algae are showing a very good antioxidant and anti-diabetic potential. This Cholera verticillata, especially, it is having 70%, more than 70%, and in alpha amylase and also alpha glucosidase inhibition is nearly with the standard. It is to the standard. So what I would like to conclude here is the phytochemical analysis. We have got the presence of bioactive compound, and then this Cholera verticillata could be a very good choice for a natural antioxidant, and also which has anti-diabetic value in addition. So it will be an effective alternate approach for reducing this oxidative stress related diabetes. And here this is a preliminary result, whatever I have shown, but still we have done further analysis we are doing for in vivo studies also. Thank you.
Okay, so thank you, Ms. Maheshwari. So there's a question for you. How will you compare? Oh, I'm sorry, this is not for you. Okay, Ms. Maheshwari, I have a question. Like, only flavonoids was done in your study?
No, ma'am. Actually, phenols also we have done, but okay. I have given it somewhere, so I cannot give it here. I have done phenols also,
You have done phenols. What about tannins?
Tannins we have not done, ma'am. Phenol and flavonoid we have done.
Okay.
Tannins we have not, yes.
Okay, so among the assays like ABTS, DPPH, FRAP, why did you choose ABTS for your study? Was it like...
yeah. ABTS assay, if we want to study antioxidant assay, there is 2 mechanisms like hydrogen atom transfer system and also a single electron transfer system. So I think ABTS comes under, it has both the capacity to capture both the single electron transfer system as well as the hydrogen atom transfer system. So it is best than the DPPH, because DPPH also I have done. More than DPPH, ABTS was giving a collective thing you can see in ABTS. So we have selected ABTS, ma'am.
Okay. Okay. So thank you, Ms. Maheshwari. That was an excellent presentation from your side.
Thank you.
Thank you. So we'll move on to the next presenter, oral presentation 20. Mr. Lee. Yes. Okay, so next presenter, Mr. Lee. Mr. Lee is from the department. Yeah, Mr. Lee. Mr. Lee is from the Department of Parasitology from University of Malaya to present on the topic development of point of care loop mediated isothermal amplification lateral flow assay for detection of Plasmodium species. Over to you, Mr. Lee.
Okay, hi. Thank you for the opportunity to present on IECBBB 2020. I'm from the Department of Parasitology, University of Malaya, and my name is Lee Fun Youth, but you can call me Zan Lee. My supervisor is Professor Dr. Lau Yiling, and I'll be presenting on the development of point-of-care loop-mediated isothermal amplification lateral flow assay for the detection of Plasmodium species. So, malaria. Malaria is caused by blood protozoan parasite of the genus Plasmodium that belongs to the phylum Epicomplexa of kingdom Protista. So, according to Malaria Report 2019, an estimated of 228 million cases of malaria occurred worldwide with 405,000 deaths. So, to treat malaria, diagnosis of the species is required for the treatment. So this is where point of care come in. Point of care or bedside testing is defined as medical diagnosis testing at or near the point of care that is at the time and place of the patient. So basically what this means is that when patient come in, we want to test them on the spot. So the ideal characteristics for a point of care test in resource limited settings is the assured. So A standing for affordable, sensitive, S standing for sensitive, so on and so forth. Specific, user-friendly, rapid, equipment-free, and delivered to those who need it. Okay, so to diagnose Plasmodium species, the gold standard is to use microscopy. So, but however, there are limitations to this as it needs or requires trained personnel, and depending on the stages of the species, sometimes it's hard to distinguish between the difference in species. For example, predominant circulating parasite stage, it is not possible to identify the robust morphological differences in P. knowlesi and P. malaria or P. falciparum using routine microscopy. And to actually confirm this, nested PCR will be used to actually determine the species. So rapid diagnostic test kits. So in the market already, there are actually rapid diagnostic test kits. But however, there are actually 5 species of Plasmodium and they only cover 4 species. So this is considered as a point of care as well because it is rapid and it's cost effective as said in our assured tagline. So why is this not as good, or what is the limitation of this? It has a very low sensitivity and the it can only detect parasitemia of more than 100 parasites. So molecular methods, as stated before, PCR is used to confirm the microscopy, right? And another molecular method would be loop-mediated isothermal amplification. So comparing both of these, PCR is actually takes a longer time compared to LAM. It only takes 30 minutes for LAM. And PCR is less sensitive compared to LAM. And I'll elaborate on this later on. And PCR actually requires multitudes of temperatures, whereas LAM only requires isothermal amplification, which is at only 65 degrees, depending on the polymerase that you use. So detection of malaria. So this is the ranges of the diagnostic tests and the limit of detection rates and the ranges. So as you can see, loop-mediated LAM is actually at the lowest limit of detection compared to the others. And the diagnostic test, the time taken is as stated in the table. So loop-mediated isothermal amplification. Okay, what is this? What is this process? So loop-mediated isothermal amplification requires 4 to 6 primers, making it more specific and sensitive. So the principle behind it is that the double-stranded DNA that repeats due to the multiple binding sites of the primers forming concatenamers. So how does this work? So mechanism starts off with the replication of a dumbbell-like structure that acts as a metaphorical seed that allows multiple primers to bind and continue to replicate. As the replication continues, more sites are available for the primers to bind, and it forms the concatenamers. So, discussed previously, why loop-mediated isotope amplification is very suited for POC. One, it's rapid, less than an hour, sensitive, it's specific, it's easy to use because it only requires one isotomal temperature. It's affordable, and it's ideal for resource-limited environment. So previously, already there's a paper published on this 5 species of Plasmodium. However, there are 5 limitation or things that is not optimized for POC. So one, it needs a DNA extraction method because LAM still requires double-stranded DNA, as I said before. It's sensitive and therefore it is easily contaminated. And thirdly, for this detection method or endpoint result interpretation, it needs a turbidity meter. So how do we circumvent all this, right? So one, the DNA problem. So commercial kits require you to have pipettes and stuff like that to actually facilitate this process, and it takes 20 to 40 minutes. Therefore, an alternative DNA extraction method is proposed. So an alternative DNA extraction. So previously published by this Zhou et al. Was able to extract DNA from plants in under 30 seconds using only filter papers. They hypothesized that the mechanism underlying the rapid purification method is based on the differences in the kinetic of nucleic acid binding to and released from the cellulose, which is the filter paper itself. And in another paper, Choi et al. Was able to use the filter paper coupling with high temperature to extract the DNA. So, next, the second problem we want to tackle is the contamination part. So we know that LAM is highly sensitive and makes it more vulnerable to false positive amplification caused by contamination. So there are mainly 2 types of contamination. One, carryover contamination. 2 is cross-contamination. So how do we circumvent this? Carryover contamination, mainly there have been modifications to the assay to reduce or prevent this from happening. One would be mineral oil, and the second one would be deoxyuridine triphosphate coupled with uracil DNA glycosylase. So I would like to elaborate more on this DUTP and UDG. So when LAM forms this concatemer with DUTP, and you move on to your next amplification, and you have UDG in place, the uracil DNA glycosylase actually breaks down the DUTP so that the concateners, the LAM product that's formed would not be carried over to the next amplification, making it less likely to be contaminated by your previous amplicons. So tackling the third problem, detection without machine. So lateral flow is a simple device that detects the presence of a target substance in a liquid sample without the need for specialized or costly equipment. So coupling with LAM, which is highly sensitive.
Mr.
Highly
Lee?
sensitive. Yes.
Yeah, you have 1 minute to wrap up, 1 or 2 minutes.
Okay.
Maximum.
So it allows the direct visual detection of end product able to be used in an endpoint setting. So my aim is to develop a LAM lateral flow assay for detection of Plasmodium species. The objectives would be, as stated previously, to develop an alternative DNA extraction, to modify the LAM assay, to reduce contamination, and combine LAM and lateral flow for the direct visual detection. So this is my preliminary results, and the buffers that I use for the alternative DNA extraction has mostly been successful, and the carryover contamination, it is the DUTP and UDG works as intended. So this is the lateral flow, and the big picture would be an encompassing protocol for the diagnosis of 5 species of Plasmodium. Okay, I would like to state my thanks to my PI, my lab mates, and the team, and also IECBBB giving me this opportunity to present. And these are my reference material. Thank you.
Thank you, Mr. Lee. That was a wonderful presentation. Okay, how did you validate your method?
Validate my method, which part?
I mean, you have found the lateral flow LAMP method, isn't it? So how do you say that yours is a valid method to detect Plasmodium?
So to validate it,
Yeah, there are already existing methods, isn't it? So how
Yes,
will you
correct.
compare your method with the existing methods?
So LAM has already been LAM for Plasmodium is already there, and we just want to use an endpoint interpretation result to actually show it and use it in a point of care setting. So everything is actually already on the market. We just want to make a one encompassing diagnostic tool so that we can do it in a timely manner where it's fast and effective. Because lateral flow itself is not as said in the rapid diagnostic kits in the market already. It's not as sensitive, whereas LAM is sensitive. So we want to couple them together and make it so that it's both sensitive and very time efficient as well. If it answers your question.
Okay, actually, in LAMP, there is a limitation that it requires 4 to 6 primers for the test, isn't it?
Yes.
So was there any, any effort from your team to overcome those limitations also?
It's actually not a limitation per se, because having multiple primers is fine, and it makes the lamp to be more sensitive as well.
Okay. So that doesn't interfere much with the results.
No, it doesn't.
Okay. Okay. Is there any other way, like this uses DNA, isn't it? Any other
Yes.
method in LAMP method where we can use RNA?
RNA.
Yes.
To my knowledge, I feel like it's possible.
Mm. Yeah, I think RT LAMP method is there for RNA detecting RNA.
Okay.
Okay, so thank you, Mr. Lee. That was a wonderful presentation.
Okay, thank you.
Yeah, wonderful work done by you. So we'll move on to the next presenter. Ms. Fatma is back. So can we have Ms. Fatma here? Ms. Fatma? Presenter number 19. Yeah, Ms. Fatma. Hello.
Oh, hi everyone.
Yeah, Ms. Fatma.
Ms. Padma,
Yep.
you have 5 minutes to finish your presentation, so you can start where you stopped during last session.
Okay, so hi everyone. Sorry for the inconvenient cause. So I will continue my presentation. Prior to cloning of PKD2 alpha 2, it will be amplified using specific design primers. Then this gene will be cloned into PGMT vector, and then the next one will be P-display vector. So the sequencing analysis will be performed using bioedit sequence alignment editor. And then prior to that, in EDA, COS7 cells were transfected with the recombinant plasmid containing our gene of interest. Human erythrocytes were added to the cells, and positive binding was shown by the formation of rosettes, which were aggregates of erythrocytes surrounding cells, which express the PKD2 alpha 2. So for the determination of rosette number, 1,500 transfected cells were observed at 20 times magnification. For determining rosette size, 20 rosettes were randomly chosen and their size were measured according an imaging software. Statistical analysis were carried out using SPSS statistics, and rosette number and rosette size difference were analyzed using independent sample t-test with p-value less than 0.05 were considered as statistically significant. And then moving on to the result, in EDA, the transfected COS7 cells were tested with human erythrocytes. So figure 3 show the expression of PKD2 alpha 2 on COS7 cells and adherence of human erythrocytes to the COS7 cells to form a rosette. So the results from the assay show that the mean number of rosettes formed between PKDT alpha 2 of low parastemia isolates with human erythrocytes was significantly higher compared to high parastemia isolates. Meanwhile, for data in table 2, the size of rosettes in high parastemia isolates was larger compared to low parastemia isolates. All results were considered as statistically significant with all p-value reported less than 0.05. So malaria rosetting is the spontaneous binding of Plasmodium infected erythrocytes to uninfected erythrocytes to form cluster of cells. According to previous studies, the aggregates resulting from rosetting may block capillary blood flow and previously shown to be associated with severe malaria. In this in vitro erythrocyte binding assay, the rosette refer to aggregation of erythrocytes that surround the mammalian cells expressing the protein ligand. Through this assay, we investigate the binding ability of malaria parasite ligands to bind to erythrocyte receptor. The binding activity of parasite ligand and its receptor was quantitated by counting the number of rosettes. The result from this study revealed high binding activity of PKD alpha 2 of low parastemia isolates with human erythrocytes compared to high parastemia cases. These findings contradict with those previous studies as high parastemia, a criterion of severe malaria, most likely resulting in high number of rosettes. Unfortunately, in our study, information of severity of malaria was not recorded at the time of sample collection due to limited access to the data. But then interestingly, the size of rosettes in our findings showed high parastemia isolates for human erythrocytes were relatively larger compared to low parastemia isolates. Large rosettes usually seen in patients with severe cerebral malaria or severe and complicated malaria. It is also possible that the formation of rosette is a crucial factor in determining clinical outcome, with large rosette of those which are particularly resistant to physiological shear being more likely to result in severe disease. So as a conclusion, the association between rosetting and parastemia levels still remains to be clarified, and there may be unknown factors mediating the relationship between these two variables. More in-depth studies involving larger sample size should be carried out to improve this study and to clarify the association between rosetting, parastemia level, and severity of the disease. And these are my references. And then I would like to thank to my research team, University of Malaya, of the Department of Parasitology, and also IECBBB 2020 for this opportunity to present this slide. Thank you.
Thank you, Ms. Padma. Just a question like, how it is going to be clinically significant, like finding out parasitemia when compared to the rosette formation. So how would you say that this is more efficient than the clinical significance of finding out parasitemia that you have done in your study?
Sorry, can you repeat your...
Ms. Padma, actually you have studied about the association between rosetting and parasitemia levels, isn't it? So how it is going to be useful in the clinical field, studying this rosetting and parasitemia levels?
Okay, so because usually rosette will block the blood flow and it will cause several adverse effects. So through this study, we will know the mechanism of host interaction of the parasite ligand, and then we can manage for example, like we will have a greater understanding of the host parasite interaction, which will assist in the development of new treatment.
What kind of receptor does it bind to?
Duffy antigen receptor for chemokine on erythrocyte, on the erythrocyte surface.
On erythrocytes?
Yep.
Oh, okay, so there is another question. Why did you use recombinant technology?
Why I use recombinant technology?
Yes.
Okay, so in this study, we clone our sample into mammalian expression host to postulate the mechanism that has been occur in our body.
Okay. Thank you, Ms. Fatma. That was a nice presentation from your side. So thank you. We would like to move on to the last but not the least presenter, Mr. Naveen. Presentation by Mr. Naveen. Mr. Naveen is from AMD, USM, presenting on the in silico selection of RNA aptamer against progesterone receptor using computational docking, employing progesterone response elements derived RNA. Over to you, Mr. Naveen. Hello.
Good afternoon, everyone.
Yeah.
My research title is in silico selection of RNA aptamer against progesterone receptor using computation. Yes. Yes.
Please, can you please share your PowerPoint presentation?
Oh, we need. Is it visible?
Yeah, you can put it on presentation mode. Yeah, thank you.
Yes. Is it visible?
Yes, now.
Okay,
please go
okay.
ahead. Thank you.
Yeah, sorry for the inconvenience. Good afternoon, everyone. My research title is in silico selection of RNA aptamer against progesterone receptor using computational docking employing progesterone response element derived RNA. Progesterone receptor is most commonly associated with the occurrence of breast cancer, the leading cause of death in women worldwide regardless in developed nor undeveloped countries. PR is a member of the nuclear receptor family. It consists of three main domains: the amino-terminal domain, the highly conserved and centrally located, and the C-terminal ligand-binding domain. Human PR is expressed as two main isoforms from a single gene under two different promoters, PRA and PRB. There is also another truncated isoform known as PRC. Both the isoforms PRA and PRB have an identical DNA binding domain and ligand binding domain, which is parted by a hinge, while PRC only has ligand binding domain at the C terminal. Besides that, PR is a ligand-inducible transcription factor which carries out the transcription activity through genomic action via interaction with the genome. The genomic action of PR begins upon the binding of progesterone on the ligand binding domain of PR, which confers conformational changes and subsequent dimerization of the receptor. This is followed by the direct binding of the DNA binding domain to a unique consensus sequence on generally referred to as the progesterone response element on the promoter of the target genes, eventually leading towards the transcription of the genes. The PRE comprises an inverted repeat hexanucleotide sequence separated by a 3-nucleotide spacer which is distinctively specific for PR. Thus, PR is a promising diagnostic and therapeutic biomarker for breast cancer. Detection of the altered expression of PR in breast cancer is carried out using PR antibodies. However, the antibody-based applications are usually fraught with high cost and complexity of production, batch-to-batch variability, cross-reactivity, contamination, and short shelf life. The substitute to conventional antibodies could be met by next-generation affinity molecules that overcome the limitations of cost, synthesis, stability, and specificity of target binding. Here comes the role of aptamers. Aptamers are defined as single-stranded DNA or RNA oligonucleotides capable of binding to target molecules with high affinity and specificity based on their unique tertiary structural conformation. Today, thousands of aptamers have been selected against a wide range of targets such as metal ions, hormones, antibiotics, vitamins, toxins, proteins, cells, tissues, bacteria, and viruses. Aptamers have gained considerable importance, superior to antibodies due to various advantages such as smaller size, low molecular weight, more structural flexibility, low degree of immunogenicity, higher thermal stability, prolonged shelf life, the possibility for reusability, no batch-to-batch variation, lower production costs, and easier to be synthesized. Moreover, aptamers can selectively distinguish between very similarly structured biomolecules like caffeine and theophylline, which differs only by a methyl group. Owing to their superior properties, aptamers have received a great deal of attention in numerous applications ranging from diagnostics to therapeutics, targeted drug delivery, and biosensing. Aptamers are generally selected in vitro from a random oligonucleotide library through an iterative process called systematic evolution of ligands by exponential enrichment, which is simply known as selects. Selects consists of three major steps, which includes incubation of the randomized oligonucleotide library with the target, followed by separation of the unbound sequences from the target bound sequences, and amplification of the bound sequences. These steps are repeated for at least 8 to 15 cycles before a pool of nucleic acids having high binding affinities obtained. The enriched pool of sequences is cloned and analyzed in order to reveal the putative aptamer. The entire process is tedious, time-consuming, and often fails to enrich high-affinity aptamers. Additionally, the requirement of fixed priming sites in the sequences of a library imposes a length criterion on random region, thereby restrict the diversity of the synthesized aptamer library. Even the lengthy aptamers, usually not more than 40 nucleotide long, require prior trimming and shortening for their efficient scale-up production. Recently, PR targeting aptamers have been identified using classical selects screening employing multiple rounds of protein aptamer binding, selection, amplification, and enrichment. However, these aptamers were too lengthy and required truncations to obtain smaller aptamers. The drawbacks associated with certain steps of the conventional selects intensify the efforts to adopt in silico approaches in developing RNA aptamers. In silico approach is a strategy that involves the usage of computer-based simulations to model nucleic acid target interactions to compensate for the drawbacks of in vitro selects procedures in order to generate aptamers. That said, we report an in silico selection of RNA aptamer to the progesterone receptor using RNA sequences derived from human progesterone response elements. My methodology. Firstly, 64 different RNA analogs were derived from human progesterone response elements. Then, those sequences were subjected to secondary and tertiary structure determination. AMFOLD was applied with default settings to determine the secondary structure of the sequences. The most thermodynamically stable structure with the lowest free energy was opted, whereas RNA composer was used to determine the tertiary structure of the sequences. The secondary structures of the RNA in the Vienna format obtained from the AMFOLD analysis were used as the input for the RNA composer. However, the resulting tertiary structures of the RNA are downloaded in protein data bank file format. In the meanwhile, the PR structure was also retrieved from the protein data bank. After that, the likelihood of the generated sequences to emerge as PR aptamer was examined using molecular docking. Molecular docking is a computational method to predict the formation of aptamer target complex based on the lowest binding energy. The docking tool that we utilized was PatchDock. PatchDock is a geometry-based web server which employs high-tech data structures and special pattern detection techniques such as geometric hashing and poses clustering to accelerate the computational processing time, allowing large-scale docking experiments to be carried out. Once the 3D structures of RNA and target are submitted to the server in the PDB format, the PatchDock starts the docking process. Subsequently, each aptamer target complex is evaluated by a scoring function based on both the geometric fit and atomic dissolution energy. Finally, the root mean square deviation clustering is applied to determine the top scoring aptamer target complexes. The sequence with the highest docking score was selected as the candidate for binding validation. These in silico predictions need further confirmation by methods such as gel sheaf assay and ELISA. In its entirety, out of the 64 sequences of RNA analogs of aptamer-like progesterone response elements, PR aptamer 31 showed the highest docking score of 10,806. As you can see, the secondary structure of PR aptamer 31 shows the formation of a hairpin loop due to the complementary base pairs to hydrogen bond with each other. Hairpin loop is the most common contact point that anchor the aptamers to the epitopes on the surface of the target protein. The coalescence of the variety of the secondary structures of the aptamer eventually result in a unique three-dimensional structure that is capable of recognizing and binding to the respective target. The interaction of the aptamer target complex is established via non-covalent bonds, including hydrogen bonds, van der Waals forces, base stacking, as well as hydrophobic, and electrostatic interactions. The PR aptamer 31 PR DBD complex is shown in different kind of models. In conclusion, PR aptamer 31 is identified as the potential aptamer using the in silico based aptamer selection. The selected aptamer will be validated by in vitro binding assay. These are my references. Thank you.
Mr. Naveen, good presentation from your side.
Thank you, Miss.
Okay. Let me see for any questions from the floor. There are no questions. So what was the method you used for docking?
Patch dock.
Patch dock. Why did you choose patch dock?
Because it was user friendly and it is a rigid based docking software, and it is freely available in the online software.
Are there other methods also for studying these apps?
They are available, but they were studied. We were also studied using head dock and even auto dock, but the processing of the data is still in process.
Oh, okay. So you haven't done the comparison yet with the other methods?
Yes.
Okay. Okay. Good. Okay. Thank you.
Thank you.
Thank you, Mr. Naveen. So it was a wonderful session, a platform to share our knowledge between the researchers. I hope everybody would have benefited with the presentations here. So thank you to the presenters and best wishes to them. Thank you for all the organizing committee members. And good work to all. Thank you. Over to Ms. Sarah.
Thank you, Dr. Annie, and thank you to all the presenters. I'm not sure if you're supposed to go for break. Dr. Subhash?
Yes, Ms. Sarah. We'll have 15 minutes break. 2 o'clock we join.
Okay, sure. Okay, so we'll be taking a break for 15 minutes and we'll be back in 15 minutes. See you all on the other side of the break. Thank you.
Thank you.
Welcome. Thank you, Sarah.
Dr. Subhash. You are on mute. Dr. Subhash.
Dr. Suresh?
Yes. Yeah, Dr. Sridhar
Somebody is
here.
operating from there.
No, Dr. Sridhar sir is there. Maybe you can give the co-host, add co-host, please.
Yeah.
Hi, good afternoon, everybody. This is Sridhar here.
Good afternoon, sir.
Good afternoon.
Welcome, sir. Thank you so much for accepting.
No problem. Pleasure.
Sir, here the another 15 minutes break, sir. 2 o'clock we start, sir. Another.
Okay. 15 minutes. Okay, good.
Yeah, sir.
Good. I'm online, so let me know whenever you're ready.
Yeah, please, sir. Thank you, sir.
Sure.
Hi, so welcome back everyone. Hopefully everyone is excited as I am to get back to our international e-conference on biotechnology, bioinformatics and biomedicine hosted by Ames University. Now, I'm sure that all of you are feeling a bit refreshed after the break and ready to get back to our presenters. Now, before we start, as usual, I would like to remind everyone that we have participants from many different countries and cultures. So please be mindful and kind while interacting or asking questions either in Zoom or also on Facebook or YouTube or any of the other platforms that we are streaming on. Now, we are going to enter our fourth technical session. So I would like to introduce Professor Dr. Lee Soo Yin as the chairperson and Mr. Maheshwaran Soliappan as the co-chairperson for this session. Now, the session is all yours, Dr. Lee and Mr. Mahesh.
Thank you, Mr. Gerard. So a very good afternoon to our distinguished speakers and fellow participants from all over the world. So welcome back to the session 4 of today's program, I'm Lee from Ames University, and with me today as my co-chair is Mr. Maheswaran from Ames University as well. So for session 4, we have one keynote and 2 plenary talks from 3 distinguished scientists from India. Our first session is a keynote speech from Dr. Sridhar Sivasubu, who is a senior principal scientist in the CSIR Institute of Genomics and Integrative Biology, IGIB, from Delhi, India. Dr. Sridhar is also an associate professor from the Academy of Scientific and Innovative Research, India, and an adjunct professor from the Department of Medical Genetics, Kasturba Medical College, Manipal, India. Dr. Sridhar completed his PhD from MS University, India, and postdoctoral research from the CSIR Center for Cellular and Molecular Biology, India, and the University of Minnesota, USA, with specialization in genetics and genomics. He joined the CSIR IGIB in 2006 and established a high-throughput genomics research programs such as the wild zebrafish genome sequencing and human genome sequencing in India. In the year 2009, his lab sequenced the first ever entire genome of an India citizen, catalyzing the application of genomics for diagnosis of genetic diseases in India. His current research interest is focused on sequencing human genomes for adoption and benefit in healthcare and applications in rare genetic disorders. He's also developing zebrafish models for application in precision medicine in humans. His lab also explores non-coding RNA mediated regulation for vascular and cardiovascular development and diseases. And he also co-founded the Genomics for Understanding Rare Diseases India Alliance Network, or Guardian. I welcome Dr. Sridhar to present his speech entitled Personal Genomes to Precision Medicine, Implications for Rare Genetic Diseases. Over to you, Doctor.
Hi. Good afternoon, everybody. And I thank Dr. Lee and Dr. Maheshwaran for the kind introduction. And I also thank Dr. Venkat and Dr. Marimuthu for inviting me into this meeting and the conference. And I can see it's very lively and a lot of participants. Thank you once again. Let me share my screen, 1 minute. Am I visible?
Yes, sir.
Can somebody just, yes? Okay.
Yes, sir. Yes, sir.
Great. Put a full screen mode. Yep, that's better. So what I'm going to do for the next 40 minutes or so is to talk about my personal journey the last 1 decade on how did you go from understanding personal genomes to converting it into a new field called precision medicine, and how did it implicate a field called rare genetic diseases in India. And when I say I'm presenting, it's a collaborative work of a lot of people in my own lab, my own institute, and a whole lot of set of clinicians across India. I'll be acknowledging them later, but nevertheless, it would not have been possible without the clinicians, the patients, and very active support from CSIR India. With that, some mandatory disclosures. We work with a number of companies for translating the knowledge that we get into commercial products. And these are some of the companies that we work with. In terms of research interests, I have 2 major research interests in the lab. One is to look at genetics and developmental biology, where I'm interested in understanding how non-coding RNAs mediates vascular development. I'm not going to be talking about that today. However, I'm going to be talking about the functional genomics aspects of my lab, wherein we are interested in understanding how do you develop this field called personalized and precision medicine in India in the context of rare genetic disease, and how do you use animal models to functionally validate them. That's what I'm going to be really focusing on. So the journey that I'm going to present started somewhere around 2008, 2009, and here is a slide around that time from 2010 from the journal Nature. And this is a very iconic slide, primarily because it starts from the early 2000s in terms of years. And as you can see in the thick red bar that goes across the screen is the rapidly dropping cost of genome sequencing for whole individuals. And you can see On the other hand, in green color is the number of whole genomes, human genomes that have been coming out, from being ones and twos, from the reference human genome to the completion of the Human Genome Project to the first private genomes of Craig Venter, James Watson, and so on and so forth. It kept on growing in a logarithmic proportion. And this was the time when people like me got interested in human genomes, and that was the year 2007, when we were still had the reference genome and the individual private genomes had just come online. We thought, why not use this to actually see if you could drive a whole field of precision medicine. And that led to us to this particular disappointment in many ways, that when we looked at this, how many genomes were done around the world, we really figured out that there were only a handful of genomes, and they represented largely a representative genome out of Africa, 2 from the Caucasian population, and maybe about 3 from, one from Japan, South Korea, and China. Okay. And that was a disappointment, because a bulk of the human race actually resides in Central, Southeast Asia, and in Middle East, where we really did not have a very good representation. So we could not even start unless you could fill this gap. So that was the primary motive that we started out with, and that led to the first human genome coming out of India from my lab, where largely we are not doing it for the purpose of doing human genomes, but trying to understand how do you make sense of this 3.3 billion alphabets for running a field called precision medicine. Okay. And a fantastic graduate student called Ashok from my lab, who actually spearheaded it along with Vinod, who's a fantastic collaborator, laid the foundations of how do you analyze genomes, how do you make sense out of them. We did not stop there. Since we had this small set of information that we had gotten our hands onto, we wanted to pass it on to our friends around the globe. Okay, that led to us actually doing this fantastic collaboration that we had out of UITM in Malaysia, out of Professor Zaki and Professor Tay's lab, where we had the opportunity to sequence and analyze a Malaysian genome, largely again focusing on how do you use this genome, whole genome, to drive a field called pharmacogenomics. Okay, and again, we participated with the government of Sri Lanka at the University of Colombo, and you can see Professor Vajira Dasanaike, along with the Minister of Science, who eagerly collaborated with us to sequence the first Sri Lankan genome. The idea was to go around our part of the world, which is the Southeast Asia, to really see, to figure out what makes us similar and what makes us dissimilar, and that led to a lot of collaborations and fantastic collaboration. I made a lot of friends, and unfortunately in the COVID era, I think most of that is going to be online now. But nevertheless, I had a very good experience working with a set of very good collaborators. And the idea was, it culminated in this large set of information, which you call the SAGE, or the Southeast Asian Genomes and Exomes, which contains over 150 million variants, from again about close to 1,000 plus exomes and genomes that we put together. We have extended ourselves to the Middle East. I'm not going to largely talk about this piece of work coming out of the Middle East. I'm going to stick to what we really did with our collaborators in on the Southeast Asian part of the world. So for what really we boil down to is all about me. It's all about the single nucleotide variants that each of us carry, which really defines who we are and what diseases we carry, what traits we carry. And this led to a new specialty of medicine, if you may call so, okay, which really tried to understand the genomic information as part of his or her comprehensive healthcare supervision. And this was a very evolving definition and evolving field, and we tried to participate into it and see how could we also learn from it and also contribute towards it. Well, one of the major applications of this field of personalized and precision medicine is in rare genetic disorders, and this we realized not that it was just presented to us. It was the patients who read the popular science articles that came out in not in journals but in popular media, newspapers, magazines, about how human genomes can really impact people with genetic disorders, and they approached us. And one such family and patient really drove us to building this part of my science in my lab, where we looked at rare genetic diseases. These are diseases that affect only a handful of individuals. Okay, there are about 7,000 to 8,000 rare diseases that have been documented, and about 1,500 plus genetic disorders are inherited very frequently. And remember, I'll just come to it. In the context of India, rare is no more rare. Okay, we are one of the largest populated country, and close to about 70 million people in India have one or other form of a genetic disease. Now, with such a high proportion of genetic disease, even rare genetic diseases assumes a very large staggering proportion. Added to that, okay, there is not a really easy affordable solution for people who want to get a diagnosis done. There's a lack of understanding of the mutational background because we don't have a comprehensive genome program in the country. So all this led to what happens is a patient with a rare genetic disease goes through 8 physician visits, approximately 3 misdiagnoses, and almost 3 years to diagnosis. So this is what we really set about solving. Okay, how could we convert this to the first year of life? That was the dream that we wanted to achieve, and that's what we pitched to the government. And I'll show you how we have managed to sort of tick off some of these goals, but we are still far from the completion. Okay. So why is India a fantastic place to study genetic disease? One, large population. The genetic pool is very diverse. Many of you may be aware about it, but most importantly, the population is stratified into small endogamous communities and close-knit family structures. So basically, this really gives us a fantastic template to study genetic diseases. So what Vinod Skaria and myself, Vinod is a clinician by training and is a bioinformatician now by practice. So what Vinod and I did was to set up this program called Genomics for Understanding Rare Disease India Alliance Network, okay, which is a pan-India network that looks at how do you get patient samples, use that to do drive genome sequencing and make meaningful sense of it. If you cannot make sense of it, could you do additional biology on top of it and try to contribute back to science and to the family? So this program, which began as one family in 2011, 2012, now has grown to about 5,000 families strong, with about 280 collaborators from across the country, with 70 major clinical or research centers participating into it, making this one of the largest networks in rare disease genomics. And fantastic support by the Council for Scientific and Industrial Research for the last 1 decade, which is very important to run such large-scale programs. Okay. Now, yes, so here is so those who are interested in how we function this, how do you put together such a program, we ask you to please look at this publication that we published earlier this year that not only looks at science, education, research, but also implementation and policy, because it takes a whole lot of effort to implement something at this scale. And I'm going to talk about snippets of our experience that we have had over a decade of working with rare genetic disease, largely in the space of genomics and genome diagnosis. So like I said, so as part of this program, we not only do we not only investigate people with rare genetic disease, we also need to look at the extreme other option. Is it, if you need to look at a person with a rare genetic disease, you need to also look at a person who somehow has managed to escape all these vagaries of nature and able to live. And who would these be? This would rightly be centenarians. Remember, centenarians today would have been born a century ago, when at that point the life expectancy was 29.5 years. And if they have lived till 100, they have 3 times overlived their life. Okay, which forms fantastic controls. So we also do a lot of centenarian genomes, and all this, a lot of exomes done by us and our collaborators and by other research groups from around the Southeast Asia to put up this database called Southeast Asian Genomes and Exome. And this is largely our driving engine at the background to all the genomics that I'm going to talk about. And all this data is published and freely available for those of you who are interested, so feel free to look it up. So again, when you want to do genomics, there are largely 3 basic approaches. One, you could look at candidate gene panels, where they have their own advantages. They're cheap, relatively easy to do, but you cannot identify new genes when you do candidate gene panels. A good example would be if you're looking at a disease called sickle cell anemia or thalassemia, it is really good to do candidate gene panels. On the other hand, if you have a completely unknown disease for which the name also you cannot figure out, best is to do whole genome sequencing, because you're sequencing all the 3.3 billion nucleotides. Okay, but the problem there is lack of tools. Cost is still an issue. Okay, still it is not affordable to most of the people in my country, and I'm pretty sure in most of your countries that would not be an affordable. But there is a middle path called whole exome sequencing, where you only look at a small portion, about 1 to 2% of the human genome, and be able to make meaningful sense out of it. Tools are very good, and 85% of genetic diseases are caused by mutations in the protein coding components of the genome, which is nothing but exomes. Okay, so here is an example of how you deploy this. Here is a family with congenital lamellar ichthyosis. Now, this is a rare genetic disease. Okay, as you can see, it occurs very rare, and it is caused by mutation in 9 genes consisting of 149 exomes. Now, if you were want to drive a Sanger sequencing based panel or the capillary sequencing based panel, it is going to cost really a bomb to get this diagnosis going. However, if you do an exome sequencing, it becomes very easy. And as you can see in this family, the mother and father, they look at the skin of the mother and the father, they're absolutely normal, whereas the skin of the two siblings, the first two siblings, looks very scaly. Okay, and the lack of eyebrows, alopecia, a bunch of other defects that co-present itself. However, by doing exome sequencing, we were able to identify a mutation in a gene called trans-glutaminase 1. And so by identifying this mutation, first thing the family wanted, when they were interested in another pregnancy, we could offer them prenatal diagnosis. And using that approach, as you can see, the parents are heterozygous, whereas both the siblings who have the disease condition are homozygous for the mutation. Whereas because we knew this mutation now in this gene called trans-glutaminase 1, we could offer a solution to the family, and the family could now have a healthy sibling who nevertheless had the mutation in a heterozygous condition, and with proper counseling, they pretty much have a normal life, as we can see. But that's one part of the story. The other part of the story is that now that we knew the mutation and we had a definitive molecular diagnosis available for it, okay, clinician could administer these two siblings with retinoic acid. And as you will see in the next slide, okay, this is just 3 to 4 months after the administration. You can see hair has grown back, the skins have started clearing up, and if they are fully clothed, it will not make much of a difference than a normal human being. So the social stigma is avoided. So you can see how by just deploying a single assay, which is affordable, okay, you are able to transform a full family's life. And remember, I said India is a close-knit family largely. So which means you can go back to the look at the larger family, the community, and suddenly you have a genetic solution in the making. Moving on ahead, okay, here is a family that we actually pulled out of a newspaper, where the parents had written to the President of India for euthanization of their 6 kids because they had suffering from a rare neurological condition which could not be diagnosed. As you can see, the kids in the bed as well as the other 3 kids are all suffering from the disease, which is a progressive disease. Okay, and this caught our attention. We got Shamsuddin, who was a PhD student at that time, along with Vinod, reached out to this family, and we quickly did exome sequencing. As you can see, the eldest kid in the family and the youngest kid in the family did not have any phenotype, whereas rest of all the children had the phenotype. So along with the parents, we did an exome sequencing, and in this case, fantastic help from the All India Institute of Medical Science, Professor Shifali Gulati, who runs the neurology clinic there for pediatric neurology, and we were able to identify the mutation in a gene called MLC1, which causes a very debilitating neurological condition. Anyway, a lot of computational efforts finally identifying this mutation, but that's not really the story. The story is we went back to the family and asked the question, do you know anybody else who suffers from it? They said no. But a quick interview around the family, their extended relatives, people whom they did not know living in the same community, you could reveal that they were an extended large set of consanguineous community that had migrated from in the Middle Ages along with the Mughal army all the way from Middle East. So we could actually draw the map right from there, and they continue to stay a very close community, and we have to identify additional families that were not, they really did not know that they were related to this original set of families that all had the disease. Now, if you did a carrier screening for this 8 members, we just had access to about 82 members. As you can see, about 60% members were normal, about close to 28% of the family members were had a carrier, and about 10% of the family members had affected. So you can see now from going from exome sequencing, for doing a carrier screen, we did not do exome sequencing. We actually only genotype a single mutation. So the cost came down from a few hundreds of dollars, US dollars, okay, to a few dollars. Okay, and that's a significant reduction if you want to run a public health program. And now it can be run practically by any diagnostic lab or pathology lab. And this was fantastic collaboration from Dr. Shifali Gulati and Dr. Mohammad Farooq at IGIB, who helped us run this community service. Moving on ahead, I'm going to talk about a story of whole exome sequencing for rare primary immunodeficiency. This paper got accepted 2 days ago. So we can now look at the raw data. In this case, what Abhinav as a PhD student in my lab and Vinod's lab did is to take up this cohort of 215 cases who had a rare disease called primary immunodeficiency, where the immune system is compromised, leading to a lot of mutations. So this was a fantastic collaboration out of Dr. Geeta Govindaraj's lab in CMC Calicut in Kerala and several other clinicians around the country. Nevertheless, the pipeline works so well that currently we are able to almost diagnose all the cases and be able to identify mutations in them. And this led to this fantastic public health program that's now running at Government Medical College at Calicut, where we have converted it not as from science to community intervention. So we have clinics running now, and with the support of the government of Kerala from the state of Kerala in India, we are really able to run a weekly clinic where people not only get a genetic diagnosis. This program has been upgraded to a stem cell therapy program now, and there are now families that get help from government, because now we have a very clean molecular diagnostics based on which additional clinical programs can be built upon. So fundamentally what we discovered, that if we can get to a molecular diagnosis for this rare genetic disease, rest of the way forward the clinicians can take forward in terms of whatever advanced treatments can be done. So what was missing as a basic biologist we needed to add was the ability to identify the molecular mutation, put a low-cost diagnostic in place, and use that to drive our science, because many of these mutations are novel. So we go back to our own labs and do our cell culture experiments or animal models. I'll show you one example, and then come back to the clinic and tell them, see, now we have a proof, go ahead and implement a clinical solution. And the governments are very forthcoming to put in solutions for public health if you have evidence for it. But that's not the case always. Like I said, the exome sequencing is about only 2% of the genome. It only covers the protein coding component, but the rest of the genome also contributes to a lot of mutations. So we need to sometimes look beyond the 2% of the genomes or the exomes, and to also try to understand what else does the genome carry. Here is a story out of All India Institute of Medical Science in Delhi, fantastic collaboration out of Dr. Madhulika Kabra's lab and a fantastic PhD student called Ankit who worked on it. This is a baby with 9-month-old baby with elevated lactate and pyruvate levels with myoclonus, and clearly this was a telltale sign of a Leak's disease with neurometabolic disorder. Okay, very clean. But if you did a mitochondrial genome sequencing, we had sort of standardized this approach of doing mitochondrial sequencing at high throughput, and we had converted this into a technology that was being offered by a company called Eurofins, which can do clinical diagnosis, prenatal testing, and carrier screening. So we used this, and unfortunately that did not yield us any mutation. That got us to thinking, what else could have gone wrong in this particular baby? That's what we did exome sequencing. Again, unfortunately, we ended up in a dead end, and that's where we started applying our concepts of whole genome sequencing. And we thought we had found something. This was a large deletion in chromosome 5 that we noticed, about 142 kb, and it took away at least 3 genes. And of that 2 genes, one was ERCC8 and NDFAF2, and NDFAF2 has been previously implicated in mitochondrial disorders. ERCC8, we had no clue what it was doing there. We did a biopsy and an RNA expression analysis and found that the RNA expression from both these genes were missing. We went back, did whole genome sequencing for the father, mother, and the proband, and quickly we figured out that the father carried a large heterozygous deletion in the same loci. The mother had a heterozygous deletion in an overlapping loci, and the proband had unfortunately inherited the commonality between the father and the mother and had inherited a homozygous large deletion, okay, about 150 kb. Okay, so we did not stop there. When the parents again wanted to have a healthy baby, unfortunately the next pregnancy also had the same large deletion. Now remember, in this case, there is no medical literature that supports that this large deletion really contributes to the disease. We had to build the functional evidence around it before we could say that what is the consequence of this. And this is the story which happened between two pregnancies that I'm going to say, okay, that basically evolved around Ankit's Sabharwal's PhD thesis, where he basically figured out that this gene called NDFAF2, which is involved in chromosome, which is present in chromosome 5, acts as a molecular chaperone for mitochondrial complex 1 assembly. And this other gene called ERCC8, which is called excision repair cross complementation group 8, also present in that same genomic loci, but had no clue what it was doing. Okay, so he solved this medical mystery. He figured out that both of them originate from the nuclear compartment. The protein is made, the protein is translocated to the mitochondria, and ERCC8 is required for the electron transport chain assembly, whereas and in the absence of a proper electron transport chain assembly, actually it leads to mitochondrial dysfunction, and that leads to a lot of ROS development. And I'm here, please bear with me, I'm cutting almost a 3-year story into one slide. Okay, there's a lot of reactive oxygen species that gets built up, and this reactive oxygen species actually starts nicking the nuclear DNA. The mitochondria gets sick, and in turn, the mitochondria, when it is sick, the cell gets sick, and it leads to a lot of nicks in the genomic DNA. Now remember, ERCC8 is a DNA repair gene, and that's where the ERCC8 is supposed to fix those nicks and damages to the DNA, but ERCC8 itself is absent because there is this large deletion. Therefore, the DNA damage never gets fixed, and it leads to a deteriorating condition, and the babies eventually die around 6 months to 11 months of age. That's about 3 years of molecular biology into one slide. Okay, just take my word for it. But nevertheless, we could go from a simple cell culture assay to a mitochondrial bioenergetics assay, where the red line actually denotes the patient fibroblast, whereas the black line denotes the control fibroblast. As you can see, the patient fibroblast does not show any mitochondrial activity. We could build zebrafish models for it that mimicked most of the phenotypes that we saw in humans, and also finally solve the biology and build a low-cost MLPA diagnostic. Now we are not doing whole genome sequencing, which costs still about, at that point, about $3,000. Now it costs about $1,000, but we need to give them an MLPA diagnostic, which costs them maybe about $20-30 US. So we have boiled it down, and all this happened in about 9 months time between two pregnancies. Okay, and that's the challenge that we had to run up against, because we had set ourselves as saying that we should solve this within 1 year. Okay, and nevertheless, we went back to the family and asked the question, are there other carriers? First answer normally is no. Then you quiz the family, cajole them, talk to them, and over about a 6-month period, Vishu actually figured out that these are not the only cases. Okay, there are additional members of both sides of the family that are in, because this is not a consanguineous marriage. So what I'm trying to tell you is that if in Southeast Asia and many parts of the world where family structures are still intact, if you really go back, genetic diseases and rare genetic diseases are not rare anymore. They will run into families and communities, and that's really important to follow, because now you need to run public health programs. Okay, and that got us, I'm going to skip this, and that got us into a large public health program, okay, called INDIGENE, where the government of India wanted us to see if we can do 1,000 Indian genomes in a very defined time scale. 1,000 genomes can be done today. Take my word for it. It's not a rocket science. There are a few 100,000s of genomes being done around the world, but doing it in a very defined time. And India did not participate in any of the large initiatives for doing population scale genomes. So we had to evolve it from scratch. And within 6 months was the deadline set for it, so that we could build carrier screening for population scales, prevent adverse drug, build evidence that could prevent adverse drug events, and also look at heritable cancers. So Rahul here spearheaded this program in my lab that actually sequenced 1,000 volunteers from around the country. As you can see, the representation is by and large across the country. And in 6 months, we were able to complete it. And Abhinav and Kavita across Vinod's lab and my lab put together this database that is freely available now for looking at about 1,029 Indian genomes with 55 million single nucleotide variants. And the link for this is given at the bottom of the slide. So what do you do when you have such large scale data? You go back and build what we started out with, building carrier screens, pharmacogenomic screens, ancestries, HLA typing, incidental findings, look at rare disease allele frequencies, and that's exactly what we do when you have such a large data set. And each of the volunteers actually have a genome card now, so they are able to report back data, they are able to look at their mutations on their cell phones through an app called the Indigene app, where we provide pharmacogenomics evidence for individual genomes. The incidental findings and carrier status are on the way being loaded. They will come up in the next few months. Of course, there's a lot of fun facts that we put up board. So the idea is to create a secure, privacy protected, scalable and point of care, and most importantly, clinician enabled app. So this is not direct to consumers. That's not something that we want to happen. We actually put in a system of communication where people can receive information that they are a carrier for something, but they have to approach a clinician. Remember, we have 270 clinical outlets that can really offer this. So they have to be referred to one of the public health hospitals where the clinician requests a report, the report goes back to the clinician who counsels them, and only then the report gets handed over. So it is not a direct to patient approach, whereas it involves an ecosystem where clinicians are the primary providers. We are only the enablers. That's what the government of India and others we envision to do, and was happy that we could put this program in place called the Indigene program. But I saw in the title and I could not resist myself, the post-COVID era. What do you do in the post-COVID era? Last 5 months, we have not been doing any of what I talked about. We've all been in COVID mode, and I'm pretty sure most of you have. And the lab did not close down. So we're trying to build large concepts, high throughput detection methods. That's what we wanted to do. So we built something called the mega labs within the CSIR system. So the idea was to do really high throughput. We did a number of approaches, but what really got our attention was Illumina had just come up with this large scale NGS based test for COVID sequencing, and it was approved by FDA in July or June or July. So it has not been field tested as of yet. So it gave us an opportunity, and you can see a number of fantastic, very bright-minded PhD students and PIs participated in this from across IGIB, and we sort of set up during the first clinical trial for this COVID-seq in the world. And that's the COVID-seq at a glance. So we put a program in where people can voluntarily submit their nasal swabs or oral swabs. They go through an anonymization process, and we could do 3,072 samples in 12 hours on a NovaSeq machine. And that's a very complex slide. Bear with me. The process is much simpler than the slide looks. Nevertheless, you can get the patient samples in and be out with the results in about 12 hours. And for a population like India, where we really need to scale up not only detection, but also surveillance, because going ahead, COVID is not going away anytime soon. It's going to be around. So this is a solution that we have put across, and we are happy to do the first field scale trials for this, the clinical validations. And as you can see, each flow cell takes about 1,500 samples, and with a very good sensitivity of 98% and an accuracy of 97% in a time of 12 hours, we were able to turn the data. And of course, as a byproduct, we also got 469 COVID genomes, and this activity keeps on happening on a weekly basis in our labs. And those of you who are interested in the data and the sequences, the bioRxiv paper is up there. People can look it up. All the data is freely available. But what do you do in a nutshell when you have such an ecosystem built up at IGIB? Yes, we not only do population genomics and epidemiology by doing genome sequencing, we are able to use that to run rare genetic disease programs where we help families and communities build molecular diagnostics and public health programs. We also work with private institutions and private companies like Sanofi Genzymes and others to run clinical education programs. And those are very important if you want to translate this new field into clinical practice. And along with my colleague Vinod and Farooq, who both are clinicians, we offer 130 high-end genetic tests free of cost to people who cannot afford. Those who can afford, we do have partner companies through whom on a chargeable basis it is available. So it's a non-exclusively offered to anybody who can take it up, either at hospitals or at companies. And of course, we also build in a lot of biology. That is our core interest to drive our scientific curiosity, because we have a lot of raw material to work with when we do this exercise. I'm going to stop there and acknowledge a lot of people. It runs into a few hundred people, so I don't have space. I'm just going to acknowledge a lot of students, fantastic collaborators from key colleagues at IGIB, Vinod and Farooq, and the guardian clinicians, 280 clinicians, of course, a few thousand patients and family members. But really what I like is, if you need science to happen at scale, you need consistent funding. That was available from the Council for Scientific and Industrial Research, over a decade plus of funding. I'm going to stop there and take questions. Thank you.
Thank you, Dr. Sridhar.
Yeah.
So now we open for Q&A session. So we have one question from the audience.
Yes, tell me,
Mr. Mahis.
Yes. Good afternoon, Dr. Sridhar. It was a very great speech by you, and it's very informative to all of us. And there's one question from the floor. How can we understand a disease mechanism using genomic techniques?
Good. So there's an example that I gave about the mitochondrial disease, which started out as a diagnostic odyssey. Okay, we didn't know what was causing the early mortality in the kids. Okay, and we do genome sequencing. We figured out there was a large deletion, but these deletions don't have any proof to say that, no clinical validation or no scientific evidence to say that they actually cause the disease. So that gives you a template for actually looking at a new set of biology. When you, so like I said, we had figured out 2 new genes involved in it. You go back, build that in a cell culture model, look at what sort of biology they drive. I described a whole set of biology for that one particular disease, and you build zebrafish models, which is our favorite go-to model. You can build mouse models, actually learn new biology that earlier was not available to you. So essentially, by doing genome sequencing or exome sequencing, you open yourself up to new templates for driving biology, and you should be open to new biologies. Like we specialize in mitochondrial diseases, therefore we build a whole set of tools around it. Okay, people who are interested in other areas of science should build up specific areas, the tools, and be able to build up the biology around the field. Okay, thank you.
Yes, thank you, Dr. Sridhar. As from our side, that was the only questions. Okay, there's another question, Doctor. When you mention one of gene being deleted, which responsible for DNA repair, do you find other locations what have error as well? This is from the participants.
Very good question. Whoever asked that, I don't know the name, but very good question.
Muhammad Aizuddin, yeah.
Yeah, Muhammad Aizuddin.
Nudin.
Good question. Yeah. So yes. So in this particular case, okay, one could do a genome sequencing. When you do a genome sequencing per se, for let's say you and me also, you will find a lot of small snippets of missing information. That does not mean we are all fantastically. No, we all carry diseases, but it has to happen in a contextual manner. Only then the disease manifests. Okay, so it will be very difficult for us to now look at this genome and be able to correlate, did this particular one missing DNA repair gene really contribute to all the deletions that we notice? But yes, the short answer is that we did not notice any more than average deletions that you would notice in other genomes, or as you would notice in the parents. That we did not notice. Okay. Now, having said that, one of the driving principles that we use for our analysis is symptom-driven analysis. That means you look at a gene based on the context that it provides. In this particular context, it was a mitochondrial disease that we were looking at because of the lack of the pyruvate and the lactate levels being abnormally, and there are a lot of clinical evidence that we had built up. So in that context, we only looked at that particular loci that we were interested in studying. However, if it would have caused another deletion somewhere else that manifested to this phenotype, we would not have an answer for it, because going and investigating every small deletion is beyond answering the question for the family. It's good, curious biological question, but the short answer to the family is, did it cause the disease or not? The answer is yes. The 142 kb deletion in chromosome 5 was implicated in the mutation and caused the disease. Yes, there could be other deletions that we still will figure it out as we go ahead. Thank you.
Right, Doctor, it was very fascinating talk by yourself. And I mean, what I understood, what was the take-home message was, how do you screen an individual who's diseased and then take it up to the community level, and then further you understand the disease in a model, and then you take it up to the ground and to screen the whole community and then treat them. So thank you very much, sir. Thank you from our side.
Thank you. Thank you for inviting me.
Thank you, Dr. Sridhar, for sharing your vast experience in the field of precision medicine despite your busy schedule. And moving on to the next speaker. So our next speaker is Professor Dr. K. Balakrishnan, who will be delivering a plenary speech entitled HLA Molecular Medicine Perspective. So Professor Dr. K. Balakrishnan is the head of Department of Immunology, Madurai Kamaraj University, India. He completed his PhD from the same department in the year 1994. He was awarded with the Robert Cochrane Overseas Travel Fellowship from Royal Society of Tropical Medicine and Hygiene, London, UK, for training in microsatellite mapping in leprosy families at Wellcome Trust Center for Human Genetics at Oxford University in 1996. His research specialization is HLA immunogenetics, immunogenic polymorphisms, and their susceptible or protective associations in a number of diseases. Presently, he is the Vice President of Indian Immunology Society, New Delhi. He has published 50 research papers in reputed international journals, including Nephrology, Tissue Antigens, Nature Genetics, Genes, and PNAS. He's offering HLA tissue typing and donor matching services to a number of hospitals through Industrial Consultancy Cell, MKU. So, Professor Balakrishnan, are you there?
We can't seem to locate him, Dr. Lee. So
can we move to first Dr. Susweta Das? Is it okay, ma'am? Mr. Mahesh, can you add co-chair?
Yeah, I'm ready. I've already added.
Dr. Susweta Das, madam, can you unmute yourself?
Hello?
Yes, ma'am.
Yeah. Am I audible?
Yes.
Am I audible, ma'am?
Yes.
Yes.
Okay.
So we will go on to the next plenary speaker because Professor Dr. Balakrishnan is not available now. So I would like to introduce the second plenary speaker, Dr. Susweta Das, who is an assistant professor for biotechnology in the School of Basic and Applied Sciences, Dayananda Sagar University, Bangalore, India. She's a gold medalist in MSc from Northeastern Hill University, Meghalaya. She carried out her doctoral and postdoctoral research work in the renowned National Institutes of India, Indian Council of Agriculture Research, Nivedi, and JNCASR, Bangalore. She was awarded the Young Scientist Dr. M.N. Kulkarni Memorial Award by the Indian Association of Veterinary Microbiologists, Immunologists, and Specialists, Infectious Diseases Society, India, in 2012 for her PhD research presentation. She is also the recipient of Dr. Baruch S. Bloomberg Young Scientist Award in 2018 in the World Congress of Infectious Disease and Antibiotics by Biogenesis Health Cluster. Her research publication was chosen as the cover page article in Epigenetics Journal, and she has received 12 Best Research Paper Presentation Awards in both national and international conferences till date. Dr. Susweta's research interest lies in underpinning molecular crosstalk in bacterial infectious disease that can unveil disease-specific immune signatures, diagnostic biomarkers, and key pathways for developing new therapeutic strategies. Another one of her key research agenda is exploring the epigenetics landscape that allows unprecedented insights into understanding the host immune response to bacterial intramammary infection. So with that, let us welcome Dr. Susweta to deliver her speech on epigenetic modulators, key players in host pathogen crosstalk in bacterial infections. Welcome, Doctor.
A very heartfelt thank you to Dr. Lee Soo-Win ma'am for the introduction. At the very onset, I would like to convey my warm greetings from Dayanand Sagar University, India, to all my senior faculty colleagues in the fraternity, to my fellow colleagues and my very dear students. So I'm here today to share my experiences about a very key component which goes unnoticed in the crosstalk between the pathogen and the host, and that's there lies epigenetic mechanisms. And at this junction, I would like to tell which is the key disease, the bacterial infection that I will talk in the further slides, and that is bovine mastitis. This is a mammary gland infection that is caused majorly by bacterial pathogens. You can see here in the picture that there is an inflamed udder of the cow, which results finally in necrosis and fibrosis, and you can see the bleeding of the teat. Now, you know what a teat? Those are the junctions from where the milking man usually milks the cow, okay? So at this point, I would like to tell that what happens when the bacteria gets inside the host. So this is a schematic representation of mastitis development in an infected udder. So this is an infected udder, and this junction has been amplified here, where you can see that through the teat cisternae, the bacteria has made an entry. And once it made an entry, it started disturbing the epithelial cells, finally damaging it in the crosstalk with the host. So the environmental and contagious microorganisms invade the udder through the teat cistern, and then they multiply with the alveolus, where they are attacked by the neutrophils while damaging the milk-producing epithelial cells of the bovine udder. So how does this crosstalk actually occur? The pathogens are known to have some weapons, and those are known as pathogen-associated molecular patterns. And what does the host have? The host has pathogen recognition receptor, the gate point, which will assess and recognize who are the pathogens coming into and disturbing the host. The host includes a cascade of mechanisms because the host army needs to be ready to counterattack this pathogen which is invading. There comes this junction of cascade, where you can see LPS, which is a pathogen-associated pattern which has been recognized by toll-like receptors, which are the master receptors in the TLR group, and a cascade has generated to the activation of the key genes, the army which the host is trying to activate. At this junction, I would like to show you that here you can see there are certain red marks. What are those? Those are certain marks which various bacterial products induce modifications in the chromatin or the histone involving different host signaling cascades. Okay, so this mechanism we wanted to see. Now, why epigenetic regulation in gene expression? In this diagram, on the bottom side, I am showing a enclosed, a tightly condensed chromatin where the histones are tightly packed, right? So what are the histones? Histones are responsible for packing our DNA in a tightly packed manner inside the nucleus. Now, it is obvious that until the histones loosen up and they allow the DNA which is wrapped around it exposed, until it is exposed, the transcription machinery cannot arrive and do the needful transcription. And central dogma is a very important component where your DNA needs to get transcribed to RNA, RNA gets transcribed to protein, isn't it? So at this picture, I'm trying to show you what are the modifications that has taken place in the histones, which has actually resulted a condensed complex to become decondensed, right? So a shift from heterochromatin junction to a euchromatin junction. So you can see this region has become accessible for any other transcription machinery to take up. Why am I bringing this into the notice in this study? Because only when this methylation or histone acetylation take place, that results the opening of the junction. So until promoter junction is not accessible, RNA polymerase cannot come with its machinery to do the transcription, right? So see here, this particular histone modifications have allowed active transcription. But there are regulators which repress transcription. So you can see here the condensed histone, and here you can see how the other chromatin regulators have repressed the transcription by condensing the nucleosome again. Okay, so this keeps on happening. And who does this? These epigenetic events are done by writers, erasers, and readers. From the very layman's way, you get to know writers means they are somebody who are trying to put some marks. And what marks am I talking about in today's presentation? It is about the histone modification. So these are different writers, erasers. Erasers are those who will remove these modifications, and readers are those proteins in the cell who will come and recognize the specific modifications that the histone writers and erasers are doing. And what are the modifications? It can be anything from at any junction. So what is this showing? It is showing the nucleosome with the octamer with all the 4 histones which are responsible for giving the tight packing of the DNA. So you see here that all the histones have an N-terminal tail. And what is the speciality of this N-terminal tail? This tail is susceptible to those modifications which I just now described. So you can see that if the modifications are in the above as mentioned in the picture, it results in activation of transcription. It helps the histone to loosen the DNA. Okay, so when it comes to repression, it is the modifications which is shown beneath of the sequence. So these amino acids are the junctions where the modifications will take place. Why this diagram is important is because in my later discussion part, I will be talking repeatedly H3K4, H3K9. What does it mean? It means that in the histone protein H3, which is the number of amino acid that got modified? Is it the first or the 4th or the 6th or 9th? Then comes the next player, which has revolutionized the understanding of infectious disease, and that is your microRNA. What are they? They are the short single-stranded non-coding RNA sequences. Again, it is our RNA polymerase II in the cell, which is responsible for synthesizing them. So what do? They are always looking for their complementary partner. And who are their complementary partner? The mRNAs which are getting transcribed and ready to go for translation. But these microRNAs are looking for those mRNAs which are complementary to either inhibit their translation or to simply degrade them. And that is a key impact on the cellular process. The right-hand side, I'm showing you a network where each microRNA and their complementary partner mRNA is being shown. And this diagram, again, I will discuss at the end to correlate our findings. So what was our research that was found out? And that's the story unfolds from at this point, where I will be talking about 2 superbugs which I have studied with. So the first story is about the pathogen E. coli, and the second story is about Staphylococcus aureus. So the team investigation started, where the first episode was molecular epidemiology, and these are our research publications which told about the leading pathogens in Indian scenario causing mastitis, and that is E. coli and Staphylococcus aureus. So with this, we wanted to see in vivo model, what are these bugs doing once it is infecting the mammary gland? And there came our episode 2, where we took up the intramammary infection in mice model with clinical isolates of E. coli. When we did H&E staining, we saw the histological changes over the time. So we started noticing at 2-hour, 8-hour, 12-hour, what was happening in the parenchyma. And it was interesting to see that at 24-hour, it showed severe infiltration of polymorphonuclear and mononuclear inflammatory cells. But as time progressed, there was loss of alveolar architecture due to severe necrosis and infiltration. And that was a key point to be noted. What is happening at 24 hours that it is leading to such a drastic change? We wanted to see the histone acetylation marks because we were parallelly carry forwarding episode 3. Episode 3A was to check for histones, and episode 3B was to check for the gene expression. We wanted to see what was the level of gene expression of immune genes leading from IL-2 to SAA3, a series of inflammatory genes we wanted to screen over the time of 2 hours to 48 hours to see what kind of expression is happening. That's how we went ahead with real-time qPCR using Tachman probe-based to maintain the specificity. When we saw that 24-hour infection state was a very critical stage, we took up microarray analysis for that stage to see the global genetic changes in the gene expression. And to see the histone acetylation marks, we took up the most recommended chromatin immunoprecipitation assays, which allows the detection of enrichment of acetylation marks directly in the promoter of the highly expressed upregulated genes. What did we see? We saw that there was selective enrichment of acetylated histone, K14 and K8 at the promoters of overexpressed gene. And that was very, very interesting observation at our junction to unveil for mastitis. Next, we wanted to see the global genetic profile, the global gene expression profile. What was the network? And it was more interesting to reveal that infected versus your uninfected showed an interactome of pathways which were under the control of P300. And you can see at this junction, the left-hand side box talks about the infected status and the right-hand side talks about the uninfected status. And you can see it is red versus green, and that is not just a colored indication. It is a direct take-up of the microarray differential gene expression. So you can see there is a high expression of all the inflammatory genes in the infected tissue versus the PBS. So this data, coupled with earlier observation, clearly showed that E. coli infection activated the NF-kappa B P300 signaling cascade for immune gene expression. So you must be pondering so many changes. So what must be happening? So the road actually that we unveiled was that E. coli infection induces overexpression of inflammatory genes, and these in turn trigger NF-kappa B complex in the cytosol and activate the NF-kappa B to migrate to the nucleus where the DNA is available for all the inflammatory genes. So it binds to the target gene promoters, and the activation of this P300 NF-kappa B complex in turn on going to the nucleus results in acetylation of the histone. So P300 was the key player who was acetylating the histones at selective gene promoters. So those selective gene promoters were resulting in activated transcription. And that resulted in expression of several inflammatory genes such as interleukins, TLR, chemokines, cytokines, etc. What happened when we took the Staphylococcus aureus bug? There was a lot of variations found within the strains. Just now Sridhar sir so nicely explained that how much variations are existing within the pathogen. Same thing we noted even for the bacteria. These two pathogens were different from each other, and when we did molecular characterization, these are all available sequences which we have deposited in the database. We found this SA1 and SA2 to be distinctly predominant in the population. We took up with the histone pathology. We wanted to see the in vivo mastitis mice infection study, and we revealed that SA1 was able to show very pronounced infiltration of macrophages and neutrophils in the alveolar lumen than SA2. I would want to bring your attention at this junction that SA1 throughout I will code with red color, SA2 with green color. So you see here, temporal expression of various genes in the infected tissue against PBS control was analyzed, and we found, see at SA1, the level is so high, but when it came to SA2, it was low. So SA1 infection consistently showed higher inflammatory response compared to SA2 at all points. Same was true when we came to see the histone acetylation marks. Even though we tested for all the important histone acetylation marks responsible for transcription activation, we found that SA1 infected tissue resembled the IHC data, leading to H3K9 and H3K14 residues to be hyperacetylated. So these were the hyperacetylation components involved in the SA1 infection. Next, we wanted to see the small RNA profile, as microRNAs are the key regulators. And if you bring your notice to this diagram on the right side of the heat map, you can see SA1 is showing more of red and less of green, whereas SA2 is showing less of red. Red means higher expression. That means SA1 also resulted in high expression of microRNA. This is a snapshot of the whole array of microRNA which was differentially regulated. At this junction, I would like to bring your attention to the microRNA 301, which was increased at the SA1 infected tissue, but not in case of in your SA2 infected tissue. There was many other correlation established with microRNA, but when the integrom, the interactome network was assessed, that revealed a very interesting connection within the gene expression, because miRNA are aiming to target the mRNA. So if you bring your attention to the left-hand side and the right-hand side, you can see I have given some yellow ovals, and that shows the status of those inflammatory genes in the two states, two conditions. So in the first condition, you can see them in red, but in the right condition, you can see them in green. That means the expression was down. And at this junction, NF-kappa B is high in SA1, NF-kappa B is low in SA2. So with this key observation, we found comprehensively that SA1 was apparently one of the isolate of S. aureus activating the NF-kappa B signaling, leading to drastic inflammatory response and induction of immune surveillance, which could possibly lead to two things: either rapid clearance, or with the stages progressing, it may lead to devastating damage to the tissue. SA2 in the other strain was very, very smart. It repressed most of the inflammatory response, which might help in its sustenance in the host tissue. So one was using the HATs, maybe the other one using the DAC, the histone acetyl marks removal process. So there was a disequilibrium between the HAT and the DAC, which is normally important to maintain the homeostasis. And that was subverted by the pathogen induction. This is a recent lead where we wanted to see the alternative splice variants recently published in Gene in 2020, where we also looked into the transcriptome landscape, and we revealed that alternative splice variants were also key components. But however, today's talk I have just restricted to the epigenetic players, keeping in role your microRNA and the histone hyperacetylation. So as I told you in the introduction about these two key pictures, in our study, we found MIR301 was having an impact on NF-kappa B. So what did we see? SA2 and SA1. SA1, the red color, had high NF-kappa B. Why was it possible? Because it had high microRNA induction. And this microRNA 301 actually degrades NKRF. And what is NKRF? NKRF is actually the repressor which downregulates NF-kappa B activation. So when the repressor has only been downregulated, the repressor is only made inactive, where will it go and do the job? So your NF-kappa B was safe, and that's how NF-kappa B was high in SA1 infection. The same was contrast in your SA2 infection. Coming to your histone acetylation, the HATs, that is your histone acetyl transferase, your P300 was found to be high in the infections, indicating that they were responsible for inducing the histone acetylation marks. At this junction, I would also try to introduce a very good work by Shravan et al., one of our co-researcher, a co-scientist who has found out through omics how serine protease inhibitor can help in a disease. So not to in the purview of further details with that, but through my complete study understanding, what I found was that thanks to the advent of modern biotechnology and more thanks to the more new technologies which has come up over the last few years, and that is next generation sequencing with dual RNA-seq, integrated multi-omics analysis that has started to define the interaction between microRNA and epigenetic factors. At this, to maintain the theme of the conference, I feel the challenges of COVID can be addressed because the advent of modern biotechnology allows the use of correct bioinformatics and correct biotechnology techniques to unveil good contributions to biomedicine. A robust and promising platform to explore emerging diseases. At the end, I would like to complete the talk by sincerely acknowledging the pioneers in the field of mastitis research and epigenetic research in India, Dr. B.R. Shom from ICR Nivedi and Professor Tapas Kundu from Transcription and Disease Laboratory of JNCSR India, who have been the pioneer, and I got the fortunate opportunity to work under both the mentors and in their esteemed lab. Cannot complete without thanking my most respected Dean, Professor Sunil S. More, School of Basic and Applied Sciences, Dayanand Sagar University, who is the guiding force for every step we take forward, my faculty friends of SBS. Big thank you to my team research members, and everything is incomplete without my students. Thank you AIMS University for this the opportunity. Namaste. Over to questions.
Thank you very much, Dr. Suswita. That was a very informative and in-depth talk. So we take some questions from the floor.
Good afternoon, Dr. Suswita.
Good afternoon.
So far, there's no questions. I'd like to open the floor to the audience. Do they have any questions pertaining to your speech?
There is one question from Shrishti to Dr. Suswita. The question is, so which candidate, SA1 or SA2, is better for vaccine production?
Okay, so I feel vaccine production is very, very difficult when it comes to such changing variants of SRS strains. And that was the whole reason why there is a failure of SRS vaccine till date. In fact, to give a better insight about which would be the candidate strain, we need to capture more global isolates, covering more global provinces, so that we have a better picture about the evolution that the strains are taking up. It would be not wise at this junction to tell which will be the candidate, because SRS vaccine studies have already witnessed a lot of failures.
Okay,
Thank
all from, thank you, Doctor.
Okay.
So since there's no more questions, we would like to thank Dr. Suswita. Thank you for spending the time and accepting our invitation to deliver this plenary talk. Thank you very much.
Thank you, ma'am. Thank you for this opportunity.
Thank you.
So I hand over to the MC, Mr. Gerard.
Yes, hello. Okay. Thank you, everyone. Thank you, Dr. Lee. Thank you, Mr. Mahesh. And thank you especially for the keynote and plenary speakers, your insights on the respective topics. And it's much appreciated. And I hope the talks have helped the fellow students or the lecturers, anyone who is tuning in and watching, and who are also working on similar topics as the speakers today. Okay, now to our next agenda, which is the second e-poster presentation for the conference. Now here, I would like to introduce the chairperson and co-chairperson for this round of e-poster presentations, which is first Mr. Kurunathan as our chairperson and Ms. Sumita as the co-chairperson. Over to you, Ms. Kuru and Ms. Sumita. Take it away.
Thank you, Mr. Gerald.
Thank you, sir.
Okay. Good afternoon to all. Okay, I'll be accompanied by Ms. Sumita as a co-chair. All right. Before we start the presentation, let me remind again on the duration. It's 10-minute presentation and 2-minute Q&A. And right after the presentation, we will set the Q&A session. All right. So let us start with the first presentation. The first presentation is by Ms. Persia. Ms. Persia, are you in?
Yes, sir.
All right. Okay, we'll be, thank you for coming. Okay, Ms. Persia will be presenting on evaluation of in vitro antioxidant and anti-advanced glycation and product formations effect of barley extract.
Yes.
All right, we can proceed. Thank you.
Good evening, everybody. My name is Persia, and my topic today is evaluation of in vitro anti-glycation and antioxidant effect of the barley grass. As we all know, barley grass is a well-known cereal crop which has been known everywhere and it has been used everywhere. The objective of my study is to look at the anti-glycation and antioxidant effect of this grass. The methods that I have used is in vitro anti-glycation activity to measure the inhibition of free radicals that are made by DPPH, nitric oxide, hydrogen peroxide, lipid peroxidation. And then for anti-glycation, I have done glucose BSA and then MGO BSA inhibition assays. The hypothesis is that because of the reported antioxidant effect of the barley, so we assume that it might also have anti-AG's effect as well. And this has been reported in many papers before. Based on my results and all of the assays that I have done, we can see that barley showed a very strong antioxidant activity. And because of that, it also showed a very strong anti-glycation activity as well. As you can see, the effect is quite strong and it is comparable to the standard curve. My result is all based on IC50, so the lower the IC50, the higher the potency will be. And as you can see for all the antioxidant assays, the barley effect is better than the standards. And for the anti-glycation also in both of the assays show a very high inhibitory effect. As I said earlier, many of the drugs and many of the compounds, especially those that contain polyphenols, have been reported to have antioxidant activity. And because of that, they're also showing anti-glycation. So we assume that the higher the antioxidant activity will be higher chance to have the anti-glycation or anti-AGEs. And this is also what we have seen in my result as well. The mechanism of action for antioxidant activity of the barley is due to the polyphenols that have the ability to donate hydrogen ion, which will be able to neutralize the free radical damage. And we also found that barley show a very strong anti-glycation activity, and the mechanism of that is because of its ability to scavenge the dicarbonyl intermediates, which are very important in the formation of the AGs. And as you know, free radicals help in the AG formation, and this is maybe because of the polyphenols that it contains. So based on the research that I have done, and this work shows that barley extract contain a very strong anti-AGE and antioxidant activity, and this is mostly because of its metal ion chelating property, because it contains polyphenols, and because of its radical scavenging effect. Thank you very much. That was all from me. God bless you.
Thank you, Pesha. Okay, I open the floor for Q&A. Ms. Pesha, I have a question over here. All right. Have you done any comparison of the barley extract with any other type of extract?
We compared it with the standard, with our standard. For example, in the anti-glycation assays, we compared it with aminoguanidine, which is a very well-known drug, as you know. And for other antioxidant, we compared it mostly with PHD and EDTA.
All right. Thank you, Ms. Pesha. Thank you, Ms. Pesha. All right. Let us go to the next presenter, Mr. M. Rajesh Kannan, with his title, ITS-based genus level identification of Scleratian floral endosymbiotes in Park Bay, southern coast of India. Mr. Rajesh Kannan,
yes, sir.
You can start.
Yes, sir. Good afternoon to all. My title is ITS-based clear level identification of Scleratian coral endosymbionts in the Park Bay, southeast coast of India. Basically, this work is focused on the molecular taxonomical work of coral endosymbionts. Corals are one of the oldest ecosystems in worldwide. Nowadays, corals enter the ecological crisis due to the climatic changes. So, coral mass bleaching must happen worldwide. Coral health system is mainly influence on the endosymbiotic diversity. This endosymbiotic diversity is based on the algae. This is a family of symbiotism. India has a coral-rich environments like Bag Bay, Gulf of Mannar, and that Gulf of Kutch, Andaman Nicobar Islands. especially Tamil Nadu have two coral-rich environments. One is Bag Bay and Gulf of Mannar. The key point of the study is coral endosymbiont act as a bioindicator of coral health system. But we don't have in Indian coastal water, we don't have any baseline data what type of coral endosymbiont associated in that coral reef. This study filled that gap. ITS is one of the fast evolving marker. It can identify lower level of phylogeny. My objective is molecular identification of coral endosymbiont by using ITS marker. Then we go for the phylogenetic relationships. Our study area is Bag Bay. In this study area, we are collecting the samples from two locations. One is Mandapam region, another one is Vedali region. Mandapam region, we are collecting the 8 coral fragments, and Vedali region, we are collecting the 2 coral fragments. So after the collecting the corals, we go for the genus level identification. The coral collection, we have a strategy. We are collecting both fast growing corals or slow growing corals. The Acropora varieties are fast growing corals. Poraites or Favites varieties are the slow growing corals. And then our methodology is we are collecting the coral samples, the scuba diving techniques. Then we go for the extraction of coral endosymbionts by air brushing technique. Here, this work is a critical point is isolation of endosymbiont DNA. Normally, this is a marine biology, marine environment. So the inorganic contaminant is there. Especially this study, we are collecting the coral endosymbionts from corals and the calcium carbonate samples. Calcium carbonate is act as a blocker for PCR amplification. We are isolating very high quality of DNA. Then we go for the PCR amplification for ITS primers, gene specific ITS primer. Then we go for the Sanger sequencing method. Then we go for the phylogenetic analysis. In our result portion, we isolate the genomic DNA. This is a gel picture of genomic DNA. Then our representative gel picture of ITS. ITS size is 750 base pairs. Then we go for the Sanger sequencing analysis, the bidirectional analysis. The sequence analysis indicate that the coral endosymbiont like that Neuroseudinum or Clodocephalus identified the coral endosymbionts in the Bag Bay region. These coral endosymbiont act as a thermal stress indicator. It means Bag Bay regions under the coral thermal stress. So then we submit the nucleotide sequences, the GenMac number, we get the accession numbers. These results are supported with the phylogenetic tree. Phylogenetic tree supported with the bootstrap values and the reference clades of Neuroseudinum. Then conclusion part is the studying genetic diversity of endosymbiont communities is useful to understand the long-term response of coral climatic change. Present study confirmed the dominance of thermotolerant endosymbionts in the coral reef of Bag Bay. Neuroseudinum, majorly dominant, we identify the Neuroseudinum endosymbiont. Neuroseudinum endosymbiont is not only thermotolerant, but also adapted to extreme environmental stresses such as irradiance, salinity, turbidity. It is also the opportunistic endosymbionts. It indicates corals under the thermal stress. This is a pioneering study. Could serve as a valuable baseline data for future research on coral endosymbionts. Now, the global research community focus on thermotolerant endosymbionts. This thermotolerant endosymbionts play a vital role in coral reef conservation. That's all, sir. Thank you.
Okay, thank you, Mr. Rajesh.
Yes, sir.
We open the floor to the Q&A. Mr. Rajesh.
Yes, sir.
Okay, I have a questions. Okay.
Yes, sir.
Is the presence of these embryos class D indicate major changes basically in the bee? I mean, in terms of because it's present for the stress environment, right?
Yes, sir. Yes, sir. Normally temperature stress, sir. If it is corals are under the temperature stress, it's prone to bleach, sir. So this study indicate that corals are under the temperature stress due to the temperature. In this type of coral endosymbiont associations is there, sir.
All right. Thank you, Mr. Rajesh.
Thank you, sir. Thank you.
There's no further questions. Thank you.
Thank you.
We go to the next presenter. Mr. Muhammad Moinuddin, that's going to present on microremediation of reactive red dye by highly potential fungal strain isolated from textile effluents. Mr. Moinuddin.
Hello, doctor, can you hear me?
Yeah, I can hear you.
Okay. Good afternoon, everyone. The title of my poster is The Microremediation of the Reactive Red Dye by Highly Potential Fungal Strain Isolated from the Textile Effluent. Well, dyes used in the textile industries are a growing concern worldwide in terms of water pollution. And due to their non-degradable nature, such dyes inhibit the penetration of sunlight in the water bodies, thereby inhibit the photosynthesis process. In addition, several dyes have been reported to be carcinogenic and they may induce the allergic reaction. Microremediation simply means the application of the fungus in order to decontaminate such organic pollutants. The aims and objective of my research is to isolation of the reactive red dye degrading fungus from the textile effluent. Secondly, determination of their dye degrading capacity, and ITS identification of the internal transcribe region of the fungus for further researches. ITS is the official barcode for the identification of the fungus. Let's jump to the materials and method. Okay. We have collected the sample from the drainage canal, which was the drainage canals of the textile industries, which was untreated textile effluent. Further, we use potato dextrose agar in order to isolate the pure culture of the fungus. In the next step, we perform the liquid state dye degradation by supplementing the potato dextrose both with reactive red dye at a concentration of 50 milligram per liter, and we have incubated the medium at 30 degrees centigrade. Further, we perform the dye degradation SI by a photoelectric calorimeter by analyzing the absorbance at 545 nanometer by the given equation. In the next step, we have isolated the DNA of the highest dye degrading fungi by following a standard protocol. In the following step, PCR of the extracted DNA was performed by ITS 4 and ITS 5 primers. In the final step, PCR products were undergone the gel electrophoresis. Let's jump to the result. Well, as you can see from the top right hand corner, we have isolated the 6 fungal strains that are capable of degrading the reactive red dye and named them as TEF1 to TEF6. TEF means textile effluent fungi, and from the bar chart we can see that TEF3 was the high dye degrading fungus, accounted for 97.41% of reactive dye degradation, and among other fungus, the TF4 accounted for around 93% and TF5 accounted for 82%. From the next figure, we can visually see the dye degrading capability of TF3 by comparing with the control. Dye degrading curve by TF3 shows that at around 60 hours, after 60 hours of incubation, the dye degradation rate was around 70%, but after the 100 hours of incubation, it was 97.41%. The gel electrophoresis of the PCR product shows a single band around for 500 base pair. In conclusion, we can say that TF3 is a highly potential fungi that can degrade a significant amount of reactive red dye. However, we have some future plans with that research. We have to sequence this fungi in order to identify it at a species level, and we have also plans for LC-MS for the analysis of degrading product. But due to the COVID-19 situation, we couldn't go by the timeline. Thank you.
Thank you, Mr. Muhammad Moinuddin. Now we go for the Q&A session.
Okay.
Mr. Muhammad Moinuddin.
Yes, doctor.
Okay, just want to ask you, you have tried with 50 milligram per liter concentration. Yes. Have you tried with higher concentration and its effect on the RR degradation?
Yes, we have tried, but it didn't give that much result. But in our previous researches, we have tried 15 milligram per liter, which was enough. But we have tried higher concentration, but it didn't give the sufficient amount of result.
All right, thank you.
Thank you,
Mr. Moinuddin.
So, is there any further question, doctor?
Yes, that's all. Thank you, Mr. Moinuddin. Okay, we go to the next presenter, Mr. Awolosu. You are here?
Yes, please. I'm here.
All right. Mr. Awolosu will be presenting on unending transmission of malaria parasite in peri-urban community of Ipasa, southwestern Nigeria, prevalence and risk factor enhancing transmission. You can take over.
Okay. Do I have to share my PowerPoint?
Your PowerPoint
No,
already shared.
no, Sean. It's already shared. You just have to present.
Okay, thank you. Good afternoon, everyone. My name is Awosolu Oluwaseun, and I'll be presenting on unending transmission of malaria parasite in peri-urban southwestern Nigeria, prevalence and risk factors enhancing transmission. So malaria is a major public health problem worldwide, and particularly in tropical Africa. In tropical Africa, it causes a lot of high morbidity and mortality rates. And this parasite is caused by parasite of the genus. The malaria disease is caused by parasite of the genus Plasmodium, and it is transmitted by female Anopheles mosquito during feeding on blood. It is estimated that 3.2 billion people are at risk of contracting malaria infection worldwide. And in 2017, not less than 219 million cases were recorded, and also 435,000 deaths were also recorded globally. And out of this, Africa accounted for 92%, while Southeast Asia accounted for 5%. In Nigeria, malaria is transmitted all throughout the year, and over there, 97% of the population are at risk of malaria infection. So for the objective, the specific objective includes to determine, one, the prevalence and intensity of malaria parasite in peri-urban community of Ipinsha, southwestern Nigeria, and also we determine the prevalence with respect to sex and age group in the community. We also determine the prevalence with respect to income in the community as well. The methodology, the area chosen is Ipinsa in Akure South local government area of Ondo State. The ethical clearance was obtained from Ministry of Health in Ondo State, and then consent were obtained from individual participants. For the blood examination, venous blood was collected from patients, and then questionnaires were administered to each one of them. And thereafter, thick and thin smear were actually prepared. After collecting the blood into an EDTA bottle, we collected the blood from there and prepared thick and thin film, and were actually stained with Giemsa 3%. Then we fixed the thin smear and viewed under microscope to determine the various species. Of all, Plasmodium falciparum was the only one that were actually determined in the center. For the results, Table 1 here shows the prevalence and parasite density according to the age and the sex of each of the individual participants. We could see that male had 80.3% prevalence, which is actually far more than that of female. And this actually have been supported by many research that male are more exposed to infection because of the kind of job and activities they are involved into than that of female, which usually stay indoor. And also for the age group, we have age group greater than 21 having the highest malaria infection. 95.7% of them had malaria infection, and this was actually significant. This has also been supported by many studies, previous ones, that those who are age 21 and above are more, they are the age group that are involved in the labor work. Many of them go to farm, And apart from that, they involve in outdoor activities. They are the active age that usually get involved in so many activities that may likely expose them to mosquito bite and therefore malaria infection thereafter. Also looking at the parasite density for the between male and female, it's obvious that female had more infection, have higher malaria parasite density compared to the male. This actually is due to their immune level, immunity. Females tends to, particularly pregnant women, they tend to be immunocompromised and as such are exposed to more infection, even though few of the females are usually being exposed to infection, but those few that have it, most times they do have high density of infection. And also for the age group, it's obvious here that age group 11 to 15 also had the highest parasite density. For the income, malaria parasite, the figure below show malaria parasite prevalence in relation to income. We can see from here. It is obvious that those that have less than 18,000 income have more malaria infection, as we can see from the figure that is right here. As at that time of our research, our study, 18,000 was the minimum wage. So those who have who belong to the low economic status find it very difficult to purchase preventive materials so as to prevent themselves from mosquitoes, bites, such as mosquito net and the rest of it. And aside from that, many of them cannot afford treatment, so they are more exposed. They have infection untreated most of the time, except maybe when they are able to go to the hospital, that is when they are being treated. So in conclusion, it is very obvious that low income and age is major factors contributing to malaria infection in the study area. So education on proper use of mosquito nets is highly recommended and is very important. Thank you for listening.
Thank you. We open for Q&A. Mr. Abolo Solu.
Okay.
All right. I just want to ask, how about between different sex? Is there any significant difference in the malaria parasite infection?
Yes, there are significant difference when it comes to the aspect of age. That is for the age group between the age group 21, they had the highest malaria infection and it was significant. Those who are age group 21 and above. And this is because they are the active people who involve in many activities like farming, like trading, and so on and so forth. So it's very obvious that this group are vulnerable, and attention should also be focused on them as well. Among them are the pregnant. Those ones that are pregnant, many of them, their age group are more than 21, and also, and the rest, many others actually are exposed to the infection that are age 21 years and above.
All right. Thank you. Thank you for your wonderful presentation.
Thank you very much.
Okay. Let us move on to the next presenter, Ms. Reshma. Are you in?
Yes, sir.
All right. Ms. Reshma will be presenting on extraction of eco-friendly natural dye from Indian almond leaf or Terminalia catapa L and evaluation of its antioxidant properties. Ms. Reshma, you can start.
Yeah. Yes, sir. Yeah. So can I start, sir?
Pardon?
Hello.
Okay.
Yeah, you can start. You can
start now.
Hello.
Yes, you can
Okay,
start
sir.
now.
Yes, sir. Good evening to one and all present here. Today I'm here to present. Okay, sir. The major objective of extracting dyes from the sources of plant is to reduce or prevent the environmental pollution. In recent days, having a global concern over the usage of eco-friendly and biodegradable materials, numerous research work is being. In our work, we have studied the method to prepare natural dyes, which is an alternative to the use of synthetic dyes from the extracted substance of Indian almond leaves, that is Terminalia catapa in its period of its brown leaf. Terminalia catapa is a plant that descends under the leadwood tree family Combretaceae. It has been originated from Southeast Asia and grows widely in subtropical and tropical regions. This plant is most recognized in Ayurveda. It is known that consuming the juice of its fresh leaves, which is helpful in treating stomachache and headache, and also as a medicinal lotion for leprosy and scabies. So in our method, we have tested that the plant extracts were extracted from ethanol, methanol, and double distilled water. They were subjected to qualitative phytochemical analysis using standard methods. It was recognized that there are few phytochemical constituents available in the natural products, such as seeds, barks, and leaves of the plant, Terminalia catapa. And secondly, we tested for antimicrobial activity. The study was performed to determine the effects of Terminalia catapa brown leaf extracts at its various concentration, which can inhibit the growth of bacterial strains. We took actually 5 bacterial strains, such as E. coli, Klebsiella, Pseudomonas, Staphylococcus, and Streptococcus. And their antibacterial activity was determined using the agar-disk diffusion method. So in the agar-disk diffusion method, a concentration of 500 mg plant was extracted using the solvents such as ethanol, methanol, and double distilled water. The microorganisms were inoculated in nutrient broth and were taken, and 100 mule of the culture was taken and spread over the plate of mulla into agar using cotton swabs. The sterile disc, having a distance of 6 mm in diameter, were impregnated with 30 mule of a leaf extract. And the streptomycin antibiotic discs were placed in the middle of the plate as a positive control. And coming to the antioxidant activity of the plant extract, it was determined using the DPPH radical scavenging assay. So DPPH was dissolved in ethanol methanol, around 20 mg of DPPH. We dissolved it in 250 ml of ethanol methanol and double distilled water to obtain a concentration of 18 mg per ml. The stock solution was prepared to achieve 1 mg per ml concentration, and 1 ml of plant extract were mixed with 1 ml of DPPH. Dilutions were made to obtain different concentrations. So after 30 minutes of incubation at room temperature in the dark, the absorbents were recorded. So coming to the results of this, the phytochemical screening of the crude leaf extracts of Terminalia catapa showed the presence of alkaloids, sterols, and carbohydrates and tannins. Antibacterial activity of Terminalia catapa revealed that both ethanolic and double distilled water extract showed remarkable activity against Pseudomonas aeruginosa and Staphylococcus aureus. However, the various test microorganisms differ in their level of susceptibility towards various extracts of plants such as ethanol, methanol, and double distilled water. So the gram-negative bacteria E. coli was significantly inhibited in methanol extract, and all the 3 crude leaf extracts of Terminalia catapa revealed an antioxidant activity. The natural dye extracted from the leaves of Terminalia catapa gave a brownish color, which indicates that tannin content. Yes, tannin is a polyphenol compound that can form complexes with other proteins and macromolecules under certain environmental conditions. So the dyeing of the white cotton materials utilizing Terminalia catapa leaf arrangement with various fixer arrangements have brought about by various fixer solutions utilized, like alum fixer gave a light yellow color, and therefore the results of the study revealed that the crude leaf extracts of Terminalia catapa contains bioactive components such as alkaloids, carbohydrates, tannins, and sterols. The leaves have been maturant and abalent used in treatment of wound and ulceration. So due to its rich tannin content, it can be used as a natural dye. That's all, sir. Thank you.
Thank you, Ms. Reshma.
Yes,
now we open for the Q&A.
Yes, sir.
Right, a simple question. How about its antioxidant properties? Can you explain a bit on the antioxidant properties?
So I couldn't hear you, sir.
Okay, can you explain more on the antioxidant properties?
Oh, yes, sir. Yes, sir. Sure, sir. So the antioxidant activity of Terminalia catapa using the 3 extracts such as ethanol, methanol, and double distilled water were assessed by DPPH scavenging assay. So this DPPH, in DPPH assay, we have used ascorbic acid as the standard, and DPPH was taken around 20 mg and were dissolved in ethanol, methanol, and double distilled water, around 250 ml of ethanol, methanol, and double distilled water, to obtain a standard concentration of 18 UG per ml. And this stock solution was prepared to achieve around like 1 mg per ml concentration, and 1 ml of plant extracts were mixed with 1 ml of DPPH. So these dilutions were made, we obtained different concentrations such as like 1000 mg per ml, 750 mg per ml, and 500 and 250 mg per ml at 5 different concentrations. And after the incubation of around 30 minutes, and we have recorded the observance.
Okay, thank you,
Hello?
Ms. Reshma.
Thank you, sir.
Thank you for your wonderful presentation.
Thank you, sir.
Next, we move on to the next presenter. Next presenter is Ms. Adina Frank. Ms. Adina Frank, are you there? All right. The title of presentation is Computational Analysis and Annotation of Elaphase oleifera, Naringenin Cyclone Synthase, and Palmitoyl protein thioesterase. We can proceed.
Thank you, sir. Good evening, everyone. I, Adina Frank Peter from Faculty of Applied Science, Ames University, will be presenting on my research findings of my final year project entitled Computational Analysis and Annotation of Elaphase oleifera Naringenin Cyclone Synthase and Palmitoyl protein thioesterase. I'm very sure everyone knows oil palm. It is an important crop for Malaysia for its economic growth. So the American oil palm, or also known as Elaphase oleifera, is a palm species that has low yield of palm oil, but has a better oil quality composition compared to African oil palm, also known as Elaphase guinensis. The Naringenin Cyclone Synthase and Palmitoyl protein thioesterase are the proteins which are the functional contributors to the flavonoid biosynthesis and fatty acid biosynthesis respectively in American oil palm. So the main aim of the study is to annotate and deduce the Elaphase oleifera Naringenin Cyclone Synthase and Elaphase oleifera Palmitoyl protein thioesterase encoding cDNA sequences, and also to predict the secondary and tertiary structures of the both proteins. Can you go down, please? So I have used my laptop for this whole study, and what I've done is I've retrieved the cDNA sequence from the NCBI and annotated it, and then deduced the protein sequence to verify its attributes, and then perform the multiple sequence alignment for phylogenetic analysis using cluster X, and then construct the phylogenetic tree using TreeViewX. As for the prediction of the secondary and tertiary structures, I have used the tool FIRE2. Moving on to the results and discussion. For the annotation of cDNA of Elaphase oleifera Naringenin Cyclone Synthase, it contains an open reading frame which encodes for a peptide that contains 389 amino acids, whereas for the annotation of cDNA of Elaphase oleifera Palmitoyl protein thioesterase, it contains an open reading frame which encodes for a peptide that contains 332 amino acids. As you all can see, figure 2 and figure 3 shows the phylogenetic relationship of both the proteins with the counterparts from the other species. In the figure, you can see the yellow highlighted box, which shows the American oil palm, and it is closely related to the African oil palm, also known as Elaphase guinensis. To your right, please.
All right.
No, I'm not done yet, sir.
Okay.
To this section. Up. It's up there. The figure is up there, sir.
Sumita, can you go?
Okay, the figure 4 and figure 5 shows the primary analysis of secondary structures. Yeah, thank you. Yeah, figure 4 and figure 5 shows the primary analysis of secondary structures of the proteins. It consists of alpha helix and beta strands, which are held in shape by hydrogen bonds. And figure 6 and figure 7 shows the predicted 3D structures of the proteins, where it is based on the coverage score of the template. It contains coils, helix, and strands, where the sequence is written from the N terminal to C terminal. So to summarize, I've successfully annotated and predicted the secondary and tertiary structures of both the proteins. The predicted 3D structures will serve as a foundation for further studies. I would like to thank Department of Biotechnology, Faculty of Applied Science, IMS University, Malaysia for facilitating my project. That's all from me. Thank you.
Thank you, Ms. Adina. All right, now we open for the Q&A session. Ms. Adina?
Yes, sir.
Okay, you have annotated the reduced protein sequence, right?
Yes.
Okay, how you perform this? What method you use?
I've used the Just Bio online tool to from the I retrieved the cDNA sequence from the NCBI, and then I used the justbio.com to clean the cDNA sequence and then translate the cDNA sequence into protein sequence, and then I've performed the multiple sequence alignment, phylogenetic tree, and so on.
All right, thank you, Ms. Adina, for your presentation. Let's move on to the next presenter. Ms. Avina Frank. Ms. Avina? Irene?
Hello, sir.
Yeah, okay.
Hello, sir. Can you hear me?
Okay, yes, I can hear you. Ms. Avina going to present on in silico analysis annotation for LFS olifera, NOI, and methionine-like proteins. You can proceed, Ms. Avina.
Thank you, sir. A very good evening to everyone. I, Avina Frank Peter from Faculty of Biotech, will be presenting on my research finding on my final year project entitled In Silico Analysis and Annotation of Allies olifera nitrate-induced and metallothionein-like proteins. So as we all know, the American oil palm Allies olifera is an oil-producing plant. It's an important crop in Malaysia, best known for its oil quality. So in this study, I've used these two proteins, nitrate-induced like protein and metallothionein-like protein. Let me address them. Let me address the nitrate-induced like protein as NOI and metallothionein-like protein as MT. They play a central role in plant defense mechanism and also in regulation of metallic ion concentration, respectively. So the main purpose of this study is to annotate Allies olifera NOI and MT proteins and deduce the protein sequences, and also to predict the secondary and tertiary structure of this protein. So what I've done is, so I've retrieved the complementary DNA sequences from NCBI and annotated and deduced the protein sequence to verify its attribute, and then performed multiple sequence alignment using Crustal X to construct phylogenetic tree, and then predicted the secondary and tertiary structures of the proteins using Phy2. So moving on to results. The annotated Allies olifera NOI has ORF that encodes a peptide that contains 228 amino acid, while the Allies olifera MT has ORF that encodes for peptide that contains 62 amino acid. The predicted secondary structure for Allies olifera NOI contains 4 alpha helix and 1 beta strand, while for Allies olifera MT contains 4 beta strands and no alpha helix. The predicted tertiary structure for Allies olifera NOI was based on Pseudomonas template, which shows more than 90% confidence level. And as for Ales olifera MT, was based on sea urchin template, which shows 74% confidence level. So figure 2 and 3 shows the phylogenetic analysis of the proteins. Both the protein shows the close relation with Ales goenensis species, which is African oil palm. So moving on to figure 4 and 5. Okay, the figure 4 and 5 shows the predicted secondary structure of the proteins. As you all can see, the green color structure there indicates the alpha helix, and the blue color structure indicates the beta strand. And figure 6 and 7 shows the predicted 3D structure of the proteins. So the peptide sequence are written from N terminal to C terminal. As you all can see, that for the NOI protein, there's coil, strand, and also helix. But for empty protein, there's only coil, strand, but no helix. As for conclusion, now that I've predicted the secondary and tertiary structure of the proteins, it will serve as a basis for further molecular modeling work that needs to be done. And lastly, I would like to thank Faculty of Biotech for facilitating my project. Thank you.
Okay, thank you, Ms. Avina. We proceed for the Q&A section.
Okay.
Ms. Avina, you have mentioned that for MT, the coverage was low. Any reason for this?
The coverage is low because the confidence level is also low, and the coverage the coverage is low because of the confidence level. It is also low, only 74%.
We do have another question from the participant. All right. Do you find significant differences in structure by comparing these two gene sequences between species? And is there different, and the difference might reflecting the trait shown by these species?
Okay. Hello?
Okay. Do you find differences in structure by comparing these two gene sequences between species?
Yes. Shall I continue?
Yes, yes, continue.
Okay. Because if you can see, the Ales olifera NOI has all the helix, strand, and also coil, and Ales olifera MT only has strand and coil, but no helix. There's no alpha helix shown. So in the 3D structure, you can see there's no helix shown, because in the secondary structure of Ales olifera MT, there's no alpha helix present, but 4 beta strands are present.
Okay, thank you, Miss Irina.
Okay, thank you.
Okay, we move on to the next presenter, Miss Meroshini.
Very good morning to everyone. I'm Miroshi Nageshwara Rao here, going to present on my research topic entitled STYR 143 non-protein coding RNA gene knockout in Salmonella typhi to understand its role in biofilm formation. And this is my abstract, and move on to the introduction part. S. typhi is gram-negative bacterium, gram-negative broad-shaped bacterium, which actually causes acute systematic infection called typhoid fever, and it is quite challenging. This is quite a challenge for the underdeveloped country where a poor water treatment system for available, and this bacteria are transmitted to oral fecal route. And in recent study, we found out there is several non-coding protein RNA which actually associate with the virulence and pathogenesis of these bacterial strains. And and the virulence of the bacterial strain are associated with the biofilm formation. And in this study, I'm focusing on one non-protein coding RNA is known as STYR143, which is actually associated with the biofilm formation in the bacterial strain, will actually give the pathogenicity of these bacterial strains. And I've performed a gene knockout to study the functional role of this bacterial cell. And move on to the methodology part. First, I've done the I've done the transformation of the PKD46 plasmid into the bacterial strains, and then I've done the amplification of kanamycin acid with homologous blocks with some FRT sequences, and I purified the cassettes, and I've inserted into the bacterial genome where the ST non-coding RNA, where I perform a gene knockout of the non-coding RNA gene, which is known as STRY143. And move on to the research, result and discussion part. This is the results of my transformation of PKD46 plasmid into the bacterial strain, which is S-typhi, and I have done the transformed the PKD46 plasmid into the bacterial strains, and as a confirmation, I have done the colony PCR with the set of PKD46 confirmation primers with the amplicon size around 10,000 base pairs, 1,000 base pairs, and why I've used PKD46 is PKD46 is a unique plasmid. Is actually consists of gene of break homologous recombinase, which actually expressed by induced through L-albinose. Presence of L-albinose, it will be expressed and facilitate the homologous recombination. And then it is ampicillin resistant marker as a selective marker for the screening of transformed cell. And move on to the amplification of kanamycin acid. We have designed specific primers which can amplify the kanamycin acid from the PKD13 plasmid with some homologous blocks and FRT region, some FRT region, along with the kanamycin resistant gene. And the overall the cassette size is about 1.5 kilobase pairs. And this plasmids is actually going to used as the cassette. We're going to insert it into the non-protein coding RNA gene in the S-typhi genome. In the S-typhi genome, where gene knockout will be done, and then this is my results of gene knockout colony after the transformation of the kanamycin cassette, and the L-albinose are used to induce the expression of the homologous recombinase of the plasmid, PKD46 plasmid, and colony PCR will be done with the FRT set of FRT primers to confirm the insertion of the kanamycin cassette into the bacterial genome, and the successful gene knockout of the STYR143 non-protein coding RNA. And for the conclusion part, before the gene knockout, there will be STYR143 will be present in the bacterial genome, will give the virulent property which involve in the biofilm forming pathway. And by knocking out the STYR143, we can study the functional role of this particular non-protein coding RNA by doing the transcriptome analysis. And moreover, it is proven that using PKD46 is one of the potential plasmid that can facilitate the homologous recombination through electroporation method. And that's all from me. Thank you. This is my references, and thank you very much.
Thank you, Ms. Maroshini. We open the floor for Q&A session. Ms. Maroshini.
Yes, sir.
Why is biofilm very important for Salmonella typhi?
Actually, biofilm forming ability is actually associated with the survival of the bacterial strain in the gallbladder of the infected individual. We actually transmitted from the transmitted as a individual transmitted from the infected individual to the uninfected individual as the bacterial cell form a biofilm around the gallbladder of the infected individual, and the infected individual became a asymptomatic carrier of the bacterial strain and transmitted the diseases through one person to another person.
All right. Thank you, Ms. Meroshini.
Thank you very much, sir.
We move to the next presenter, Ms. Lavanya Rajendran, that would present on STYR143 NPC-RNA gene knockout in Salmonella typhi, which unregulated completely in biofilm forming pathway. Sorry, Ms. Lavanya Rajendran, the next presenter. The title is Overexpression and Purification of Acinetobacter baumannii HFQ protein in BL21. Ms. Lavanya. Ms. Lavanya, you are in.
Lavanya, unmute yourself. Lavanya.
Mr. Kuru, maybe she can do presentation later. You can go to next one.
Okay. All right. Because Ms. Lavanya is having some technical difficulties, we move on to the next presenter, Ms. Ang Xin Yi, entitled Evaluation of Cold Chain Free Live Attenuated Oral Cholera Vaccine Against Enterotoxigenic E. coli as a dual-use vaccine. Ms. Ang, you can proceed.
Hello, can you hear me? Can you hear me?
Yes, yes.
Okay, good afternoon, everyone. My name is Ang Xin Yi from ACE University. My topic today is evaluation of cold chain free live attenuated oral cholera vaccine against enterotoxigenic Escherichia coli, which known as ETEC, as a dual-use vaccine. I will describe about the introduction. Two pathogens, which are Vibrio cholera and ETEC, will cause direct disease, and pathogenesis of both pathogens is quite similar. There is no vaccine exclusively licensed for ETEC, and existing cholera vaccine Cholera with RCTB can only afford short-term cross-protection against ETEC infection. For my study is to evaluate the prototype cholera vaccine for its cross-protectivity against ETEC strains. For the methodology, phenotypic assessment will be done, such as gram staining in selective media, include EMB agar and McConkey agar, and biochemical tests, then followed by genotypic assessment performed by PCR by using 3 sets of primer, which are MDH, LT, ST. MDH is malate dehydrogenase gene, LT is heat-labeled toxin, ST is heat-stable toxin, followed by the ETEC strain undergo LPS isolation by using biovision kits, and followed by ELISA to determine anti-LPB and anti-LPS. For the result part, in gram staining, others move out a bit. For gram staining, all the strains are gram-negative and in rod shape. For McConkey agar, E. coli and H10407 are growth as pink colony, while ETEC growth in colors colony. For EMB agar, all the strains are lactose-fermenting bacteria and colonies are green metallic sheen. For biochemical tests that are shown in table, for PCR result, show that 3 primer used for LT, which are detected by H10407 at 400 base pair. For MDH, primer will be detected by all the strains, which are under the categories of E. coli at 300 base pair, and SD toxin detected by H10407 at 160 base pair. Now I proceed to ELISA. For anti-LTB, show there were significant 30-fold increase of anti-LTB IgG antibodies and a 2 to 8-fold increase of anti-LTB IgA was found in all the vaccinated rabbit serum samples. For anti-LPS, there's no antibodies against LPS of ETEC and H10407 strains. In conclusion, the vaccine formulation mimics a natural infection and has the potential to provide protection against ETEC infection by neutralizing the heat-labeled toxin. The cross-protection study should be further investigated in animal models. Last but not least, I would like to thank my supervisor, Dr. Prabha, and Ms. Teo for all the guidance and support throughout the project. Thank you very much.
Okay, thank you, Miss Ang. Miss Ang. Okay,
Yeah.
now is a Q&A section. All right. Under what basis you use the attenuated cholera vaccine against E. coli?
For now, it's still using the cholera vaccine called Cholera, but there is still short protection for against the effect.
Okay, why the cholera vaccine now can protect against the E. coli? What's the relation between them?
Can you repeat again? Hello?
Sorry, you are using cholera vaccine, right? But you are studying on E. coli. What's the criteria for this kind of studies?
I'm not sure about this. So.
Okay, never mind. Thank you, Ms. Ang. We move to the next presenter. Ms. Uma, are you ready? Thanks, Ms. Ang.
Okay, thank you, sir.
Next presenter, Ms. Uma, are you ready?
So,
Ms. Uma Mageshwari.
Good evening, everyone. Hello.
You can start. Thank you.
Okay, thank you. Very good evening to everyone. I'm Uma Mageshwari, and today I'm going to present a poster about evaluation of co-aggregation between live cholera vaccine candidate VC-USM14P with enterotoxigenic Escherichia coli, which is also known as ETEC, towards the biofilm formation. As we all know, cholera is a life-threatening secretory diarrhea that caused by infections of Vibrio cholerae. In this direction, the present study is focused to evaluate the co-aggregation of VC-USM14P strain with ETEC strain and the formation of biofilm. The methodology starts with bacterial purity assessment. The bacteria is tested on eosin methylene blue agar and McConkey agar, gram staining, and also biochemical test. The confirmation PCR gets done for epic identification. After the purity assessment, it is proceed with the co-aggregation assay. The standardized bacteria incubated for an hour, then the OD were taken. As for the biofilm formation, the crystal violet assay was performed. The standardized culture allocated into a 96 bar plate and incubated for 24 hours. The OD was taken after the addition of crystal violet and DM also subsequently. Result and discussion. For gram staining, all the 3 stains were stained in pink color due to the thin peptidoglycan layer. Thus, it is deduced as a gram-negative bacteria, and the morphology of the bacteria is in rod shape. As for the selective media, in EMB agar, colonies formed with a dark center, while in McConkey agar, E. coli and H10407 strain formed the pink color colonies, but ETEC strains from the colorless colonies in McConkey agar. The biochemical test table deduced that the bacteria tested is indeed ETEC, so a motile bacteria. The PCR performed using 3 sets of primers. MDH is for screening the malate dehydrogenase gene. LT primer is for screening the heat label gene, and ST primer for screening the heat stable gene. For the MDH primer, all the 3 strains, E. coli, E-tech, and also H10407 were detected, the gene. But for the heat stable and heat label, only H10407 detected. And for the co-aggregation rate for the Vibrio cholerae strain and E-tech strain were calculated using the formula. The co-aggregation rate for VC USM14P and H10407 shows the highest reading about 97.3%, and the lowest is E. coli, has the rate about 90.8%. Overall, the H10407 strain has the highest aggregation among other E-tech strain. The biofilm layer by crystal violet, quantified using the UV spectrophotometer, and the readings are tabulated. E-tech strain has the highest absorption reading, about 1.0876 of biofilm formation with VCUSM14P, while the 596B strain has the lowest biofilm formation with the E-tech strain. In a nutshell, the aggregation and biofilm formation playing an important role in the gut colonization and the diarrheal disease. Thus, this study will be helpful to develop a better vaccine that could protect E-tech infection. Nonetheless, the colonization study will be further investigated in animal models. Thank you very much.
Thank you, Ms. Uma. Okay, I open the floor for Q&A. Ms. Uma?
Yes, sir.
All right. Is there any relation or association between the co-aggregation and biofilm formation?
Yes, sir. Because the bacteria will aggregate together in order to attach to the post-intestinal layer lining. So that after they aggregate, they will be formed the biofilm slime layer. So we call it a biofilm layer. So in order to form the biofilm layer, they have to aggregate together. So they will aggregate together to form the biofilm layer. That's the relationship between that.
All right, thank you.
Welcome, sir. Thank you, sir.
Okay, we move on to next presenter. I would like to call Ms. Lavanya. Lavanya? Ms. Lavanya, are you in?
Yes, sir.
All right, you can start your presentation.
Sorry for the technical error. I myself, Lavanya Rajendran, will proceed with my poster presentation regarding overexpression and purification of Acinetobacter baumannii HFQ protein in BL21 cells. HFQ is actually a RNA chaperone protein present in many bacterial species, including this Acinetobacter baumannii. It plays a vital role in virulence of various bacteria. HFQ exhibits its function by binding with the non-protein coding RNA, which will be needed for the trans-acting non-protein coding RNA, mRNA interactions. To gain a further insights on the target non-protein coding RNA that interacts with the RNA chaperone, HFQ with a high purity is needed. Therefore, first of all, I amplified the HFQ Acinetobacter baumannii gene and further digested and purified using the EcoR1 and restriction enzymes. And then I ligated into PEG28B positive cloning vector and at the respective cloning sites. And then I transformation of it into the E. coli top 10 competent cells and subjected for the screening by kanamycin selection methods. And then the kanamycin positive colonies were then plasmid extracted for PCR conformation and transformation into E. coli BL21 cells for IPTG induction and protein overexpression studies. Finally, I purified the HFQ Acinetobacter protein by nickel NTA affinity chromatography. These are my results and discussion. Successful amplification of ABHFQ gene, and then proceed with the successful ligation of it with the into the PEG28 cloning vector. and then transform into top 10 competence cells, and then proceed with BL to 1 cells. So the IPTG induction that I use for the protein purification is actually works like usually the expression of the bacteria is controlled by a lag operon, which consists of linked genes under a coordinate control. So when I induce this IPTG, it will trick into the making the protein clone with the lag operon rather than permease and beta-galactosidase expressions. So before the IPTG expression, the culture have been pellet down until the culture reached the 0.6 OD, and then with the suitable antibody culture kanamycin, and then proceeded with the IPTG induction. Finally, I've done with the protein purification using the affinity chromatography method. The column contains this nickel NTA, which has higher affinity towards this his-tag protein specifically. So the protein mixture is percolated. When the protein percolated through the column consists of the specific DNA sequence with the his-tag sequence attached to a matrix. So the protein, HFK protein with a high affinity for the sequence bind and then retain with the his-tag. So the target protein, finally, the HFK target protein is eluted with a higher concentration salt concentration. So in this case, I've have done this imidazole washing method. This imidazole will interfere with the weak binding proteins and finally elute the his-tag proteins in a high concentration. So that's all from me. Finally, I've this is the conclusion. We had successfully purified the HFK protein from the Acinetobacter baumannii. The identification and the characterization studies will be carried out by the RNA binding study for the selection of the non-protein coding RNA and binding to the protein. So I'm thankful to the faculty for giving me a hands-on experience on it. Thank you to my supervisor, Dr. Suresh, my co-supervisors, and Ministry of Higher Education for the research project. Thank you. That's all from me.
Okay, thank you, Ms. Lavanya. Next, we open for the Q&A session. There are 2 questions from the floor. First, did you measure the OD level before IPTG expression?
Yes, of course, sir. I've done the bacterial culture, the shake culture analysis until the selective positive colony is inoculated with the kanamycin culture, and then it will be grown until the 0.6 OD reach.
All right. Another question. At what concentration did you elute with imidazole?
I'm not sure with the concentration of the imidazole, actually.
All right. Thank you, Ms. Lavanya. Thank you.
Thank you.
To the next presenter. The next presenter, Dr. Abhinaya Subramaniam, presenting on fecal religiosa in mitigating hyperandrogenism in PCOS-induced rat, a phytotherapeutic approach. Dr. Abhinaya Subramaniam.
Yes, sir. Yes, sir. Can you hear me? Am I audible?
Yes, yes, you're audible.
Yes, okay. Thank you so much. Okay, thank you. A very pleasant evening to everyone present over here, connected here for my presentation. My topic of presentation is ficus religiosa and mitigating hyperandrogenism in PCOS-induced rats, which is a phytotherapeutic approach. To start with the background information, we all know that what is PCOS? It is nothing but a common endocrine disorder in women of reproductive age, where they'll be presenting symptoms like irregular menstrual cycle, then presence of cyst in the ovaries, and also they exhibit hyperandrogenism, which is nothing but the presence of higher level of male hormones like testosterone, which is also exhibited by the masculine features like facial hairs, balding, etc. So these are the basic symptoms of PCOS women, and right now there is no prescribed treatment strategy or specific drug to treat or address PCOS. And in order to search an alternative approach or some pragmatic approach to address this issue, we sought out for a phytotherapeutic approach, which will be promising alternative for the existing things, because the existing treatment strategies mostly those include like hormonal therapy or some surgical invasions or something. Most of them were invasive kind of things, but phytotherapy may be a safe and better alternative to manage the PCOS condition. So we thought of choosing Ficus religiosa, which holds a very prominent position in the religious and traditional literature of ethnomedicinal importance. And so our study animal was Rattus norvegicus, female Rattus norvegicus, and we induced PCOS using letrozole administration. The experimental animals were divided into 7 different groups, different experimental groups, and upon PCOS induction, the whole animals were studied for estrous throughout the experimental period. Before induction, also we studied whether the animals were regularly cycling, and only the regularly cycling animals were taken for consideration. And then upon PCOS induction, it took nearly 21 to 28 days for PCOS induction. And after induction, we administered them with Ficus religiosa fruit extract. We used both fresh and dried fruit extracts. And after the treatment period, we observed the estrocyclicity, then the serum hormonal levels, and we came to a conclusion. And can you please move on to the results? In the results, we can see that the number of estrocycles, means the number of normal estrocycles, was drastically reduced in the PCOS induced group, which was completely revived to normalcy in the groups which were treated with fruit extracts of Ficus religiosa. Similarly, the hormonal level testosterone was elevated in the PCOS induced group, which was revived back to normalcy in the fruit treated groups. And the estradiol level was also revived to normalcy when the animals were exposed to Ficus religiosa fruits. And alongside, we also tried to unravel with the mechanism through which this hyperandrogenism can be balanced. And we were targeting on the aromatase enzyme. Aromatase enzyme is the enzyme which is responsible for the conversion of testosterone to estrogen, which is absent in the PCOS women. Okay, so we found that this Ficus religiosa, can you come to the conclusion picture? We found that the aromatase gene was upregulated by the expression of exposure to Ficus religiosa fruits. So the hyperandrogenic state was balanced by converting the testosterone to estradiol through aromatase activation, and hence the hormonal level was balanced in the PCOS induced rats. So this was a very fruitful result which we obtained, and this is all about our this is a part of my work. And thank you. I thank the organizing team for giving me such an opportunity, and also my team colleagues from Kaveri College and my research supervisor, everyone. Thank you, one and all.
All right. Thank you, Dr. Abhinaya. All right. We open the floor for Q&A.
Yes, sir.
Let me ask a question to you. All right.
Yeah.
Will these Ficus will have any adverse effect if it's used as a treatment?
No adverse effects. As for, we have seen even the toxicity data also, and we have also studied the antioxidant status. Now we don't have any toxic effects. Actually, the fruit is also edible, so no toxicity was observed.
All right. There's another question.
Yeah.
This is from the floor. How to control the overexpression of the gene producing the face hair in women?
Exactly, I'm not sure about that, as we have not seen. Actually, the expression of testosterone, when it is controlled, it can reflect in the hair growth also, because testosterone is responsible for the facial hair pattern also indirectly. So once the male hormone levels are normalized, these features will also get normalized from my point of view, if I'm right.
All right. Thank you. Thank you, Dr. Abhinaya. All right.
Thank you.
Okay, thank you for all the presenters for the good presentation. All right, and then I would like to pass mic to Mr. Gerald.
Yes, thank you, Mr. Kuru. So it's thank you, Mr. Kuru and Ms. Sumita. So it's now time for final session of the day, which is the oral presentations. For this session, our panel will include Evaluators will include Senior Associate Professor Dr. Subhash Bohr as the chairperson, and also Associate Professor Dr. Siva Kumar as the co-chairperson. Over to you, Dr. Subhash and Dr. Siva.
Thank you, Mr. Gerard.
Thank you, sir.
Everybody, hello everybody. Because in this session, we have 12 presentations. We have students and presenters, those are presenting from 4 countries, Malaysia, India, Nigeria, and Pakistan. So I'm going to serve as a chair for this session, and co-chair is Associate Professor Dr. Siva Kumar. So let's start with the first presentation. I just want to remind the presenters altogether you have 10 minutes. So I would like to request you to just stick to the time, and including Q&A, altogether 12 minutes. So 10 minutes for presentation and 2 minutes for questions and answers. So our first presenter is Shweta. She's going to talk. Shweta, are you there in the room? Our first presenter is Shweta. Her title is Screening and Identification of Cellulose Digesting Bacteria from Termites towards Developing a Termite and Cellulose-Based Sustainable Nutritious Solution in a resource-limited setting. So I invite the first speaker to deliver the presentation. Shweta, are you there? Shweta, are you there? Dr. Suresh, those are co-hosts. Please, can you help me to co-volunteers? Volunteers, is that? Can you check? Shweta, are you there?
It's not there, yes. I think. Yeah. Is there? Okay.
Yes.
Yeah,
yes, Shweta,
there.
now I can see. Yeah. Shweta, I'm putting you okay, as co-host. So you can share your presentation slides and you can start. Okay, Shweta, you can start your presentation.
Yes. Just 1 minute. Okay.
You can share your PowerPoints, Shweta.
I'm not able to share. Okay, I think I got it. Okay. I hope it's visible.
Yeah, we can see. Yeah, you can go ahead. You can start.
Okay. So I'll start now. Good evening to the chairman, co-chairman, and everyone present today. My name is Ravichandran Sweta, and I'm from Sunway College. And today I shall be presenting about my research on the screening and identification of cellulose degrading bacteria from termites. And its aim is to
is towards developing a termite and cellulose-based sustainable nutritious solution in resource-limited settings. Today, I shall be talking about the rationale of my research, objective, experimental overview, and several experiments conducted and results. The image on the right shows a termite which has just consumed wood particles, and the microbes present in the termite digest the cellulose present in the wood particles as well as the cellulase enzymes present in the termite itself to produce reducing sugars and other byproducts such as hydrogen and carbon dioxide. The rationale for my experiment first started with cellulose. Cellulose is an organic compound which consists of a linear chain of several hundreds to many thousands of beta-linked D-glucose units. Humans are incapable of digesting cellulose, as we all know, due to the lack of enzymes which are able to break down cellulose. However, cellulase from external sources can be used to digest the cellulose. So this project is aimed to use the cellulase enzymes produced from the termites to digest the cellulose and to use it as a nutritious solution to alleviate hunger in resource-limited settings. The objective of my research is to firstly cut and identify the various species of termites, to optimize a DNS assay using a cellulase cocktail. CTEC2 was used in this case, and to determine the presence of reducing sugars in the termite homogenate with CMC or carboxymethyl cellulose as a substrate. The last objective is to culture the most active cellulolytic bacteria from the CMC agar and identify it using 16S rRNA PCR and DNA sequencing. Firstly, let me tell you an experimental overview. We first optimized the DNS assay to construct a standard Malthus curve to use as a reference. Secondly, the collection of termites and how its separation, its separation to different hierarchies. Separation to different hierarchies was done, so I can form a termite homogenate. Before I did the termite homogenate experiment, I used a cellulase blend enzyme using CMC Whatman paper and tissue paper as substrate to produce reducing sugar. The darker Eppendorf tube on the right side shows the reducing sugar present. Secondly, the same experiment was repeated for termite homogenate as used as a so-called enzyme, and successfully reducing sugar was produced. To further find which bacteria had the most cellulolytic activity, it was spotted in CMC agar to view the hollow zones. And the bacteria that produced the largest hollow zones were isolated and cultured and identified using 16S rRNA PCR and DNA sequencing. The bacteria identified were submitted to the gene bank. Firstly, the first step was to do the DNS assay method. The DNS assay method is to test for reducing sugars, and it was used, as I mentioned earlier, to plot a standard curve for maltose. This standard curve was used as a reference to quantitate the reducing sugar produced by cellulase, Miller et al. As a reference. These are the volume of solutions that I used and the standard curve constructed. The optical density measured was at 540 nanometers. The three types of the next step is to make a termite homogenate. And firstly, I collected the termite from Ames University, and 3 species were curatively identified. Firstly, the Formosan subterranean termite. Secondly, Pacific damfoot termite, and lastly, black. It was separated into different hierarchies to identify which hierarchy produces the most cellulase to digest the cellulose. These are some magnified pictures of the termite. The features in which the physical features were used to identify what type of termite it was. And definite identification of these termites are currently being conducted using the COX-1 PCR and DNA sequencing. So these are the images of the worker termites. The brown specks are the wood that it had consumed and is currently being digested. This is the Pacific damp-wood termite soldier, and in a soldier, the head is larger than the abdomen. And this is for Pacific damp-wood termite. And the third type of Homosan subterranean termite is the winged reproductive termite. This is the image of a black termite worker and a major soldier. Okay, and there were 3 cellulase assay conducted. Firstly, using CTEC2 enzyme for both of these cellulase assay, but the only difference is 1% carboxymethylcellulose was used for the first one, and Whatman paper and tissue paper were used for the following ones. The third cellulase assay conducted was using termite lysate as a so-called cellulase, just to see whether reducing sugar was produced. So the process in which the cellulase assay was done was firstly combining the substrate with the enzyme and incubating it. After incubation, conduct the DNS assay and measure its optical density. The same was repeated for the cellulase assay with Whatman paper and tissue paper. The only difference is the substrate. These are the results for the first 2 cellulase assay, and as you can see, the optical density for Whatman paper is lower, which indicates that the Whatman paper is harder to digest compared to tissue paper and carboxymethylcellulose. So the third cellulase assay conducted was using the termite lysate. So after isolating and identifying the type of termite and its hierarchy, they are homogenized with citrate buffer to form a homogenate, and they were added to the substrate, in this case carboxymethylcellulose, and the DNS assay was conducted, and the optical density at 540 nanometers were conducted. So this is the negative control, and this is the first one is the positive control. 2, 3, and 4 are for Formosan subterranean termite, and 5 and 6 Pacific damp-wood termite, and 7th one is for black termite. As you in reference to the previous picture, the absorbance optical density for all of these termites match to the positive control, which shows that reducing sugars are produced from the termite homogenate, which again proves that cellulose degrading bacteria which produce cellulase are present in the termite. However, in the head of Pacific damp-wood termite and the black termite, that lower reducing sugar was produced. So yeah, and this indicates that lower concentration of enzymes is in the head of the termite. Secondly, the termite homogenate was spotted on CMC agar to view the hollow zones, and the hollow zones were further accentuated using congruent solution. Having confirmed that the hollow zones are produced, which means that there are cellulose degrading bacteria produced, PCR was conducted on the 4 samples which had the highest cellulose degrading activity in the CMC agar. In this case, coincidentally, all these samples are from Formosan subterranean termite. The primers used in this are 16S rRNA PCR, 16 SPF, and 16 SPR. In the DNA sequence, this is the DNA sequencing process that we did after PCR and DNA sequencing and blast analysis. The black bacteria from the termites were identified. The 4 bacteria from the termites identified were Cosaconea oryzantophytica, Bacillus parenthesis, Bacillus megatherium, Bacillus pamilus. So all these bacteria have references which show that they have cellulolytic activity and are able to produce reducing sugars from cellulose. So in a partial partial gene sequence for Cosaconia orzenica identified was submitted to GeneBank with accession number MN991174. So the conclusion of our research is the termite homogenate had bacteria and enzymes with cellulolytic activity. The 4 strains of bacteria identified to have cellulase activity. The outcome of this study will form a base for research on developing a termite and cellulose-based sustainable nutritious solution in a resource-limited setting. So that is all that I have to say. And just to confirm, we identified the morphology of the termite curatively. We didn't use any PCR method, but it is being conducted currently to identify what species of termite it is. So these are the references that I used, and thank you for your time.
Thank you, Sweta, for your presentation. So now any questions from floor? I think there is one question. Somebody from floor is asking how you identified the termites. Sweta, can you hear me?
Yes, I can hear you.
Sweta, one question. Yeah, one question from the floor. How you identified the termites?
Okay, so I used the morphology. Oh, you can't see any reference on morphology features. So in this case, I identified the termites using curatively just observing it with other images with similar species, and the morphological features such as like there's like in soldier termites, there's emphasis on its mandibles. So that's how I identified what type of termite it was and its habitat that it came from. Like the black termite is from a palm oil plantation nearby the university. So that's how I concluded what type of termite it was. And yeah, so I hope I answered your question.
Yeah. So I have one question because when you did the assay, did you use cellulose, authentic cellulose, to compare the activity of cellulose produced by, your isolates, to compare the, how is the, comparison with the standard enzyme? Did you use the cellulose enzyme for comparison?
Yes, I did use C-Tech2 Novozyme as a comparison, and the same substrate was used, which is carboxymethyl cellulose, which is authentic cellulose, like how you said.
Yeah, so another question I have is when you identified the isolates by using 16S ribosomal RNA sequence, so how much was the identity with the hits? To confirm the identity of the isolate, Sweta?
100%.
100%?
Yes.
Okay. So I don't see any other questions in the group. Thank you very much, Shweta, for doing presentation and finishing on time. So let's move on to the next presenter. Yeah, thank you, Shweta. Next presenter is Paul. She's going to talk on her research findings on health risk assessment of macro, trace elements and heavy metals in various Indian anti-diabetic polyherbal formulations. She's working at Department of Biotechnology, Jain University, Bangalore, in India. So,
hello.
Yeah, can you?
Am I
please,
audible,
please,
sir?
you can go ahead.
Yeah,
Yeah,
you're
yes,
audible.
yes, please, yeah.
Thank you, sir.
Yeah, we can start. Thank you.
So good morning, everyone present here. I am Saptadeepa Paul. I'm working under the guidance of Dr. Mala Majumdar from Jain, deemed to be university. Today, I am presenting a part of my PhD work, and my topic is health risk assessment of macro elements, trace elements, and heavy metals in various Indian anti-diabetic polyherbal formulation. So Ayurveda was originated in India 2000 years ago, which has a wide range of use in various human healthcare system. World Health Organization has also recommended use of herbs in national healthcare system as an emerging therapy for treating various chronic disorder. Among those chronic disorder, diabetes mellitus is also considered as one. Diabetes mellitus is an endocrine metabolic disorder, and it is considered as global pandemic. As the figure here that I have given, in the first figure, we can see the countries with most diabetic patient, and India has the highest with 69.2 millions. Further, the second image shows the number of deaths due to diabetes in million, which is also quite high. And this gives an overview of the global scenario of how many diabetic patients are there, or how diabetes is occurring predominantly. There are various synthetic treatments that are available to treat diabetes mellitus, but a lot of them are associated with disadvantages, such as a lot of adverse side effects are reported due to use of prolonged use of this synthetic formulations, and they are also expensive. Therefore, it will cause economic burden, and it's a huge challenge in developing countries because of the inadequate resources. The present scenario, therefore, can be resolved by using anti-diabetic polyherbal formulation as an alternative, because an anti-diabetic polyherbal formulation will consist of n number of herbs, and they will work in synergistic mechanism to give an overall treatment. Suppose caring for diabetes, and also the other complications that are arising for diabetes, such as obesity, and it will also be cost effective. With the expanding demand, there is a rising concern for safety and efficacy of these herbal formulation, and previous reports has shown lack of safety assessment studies which are associated with contamination and toxicities of the herbal formulations. One of these safety issues are related to metal ingestion, as metal are ingested in human through plants, where plant is also a source, as this plant accumulates essential and non-essential element by absorbing them from the surroundings, such as water, soil, and air, and these are accumulated in their tissue. Further, these plants are taken for herbal formulation preparation, and environmental pollution is also one of the challenging criteria here, because due to these too much of environmental pollution, there are elevated level of these elements. Heavy metal is one of the major issue, because even low intake of these heavy metals are associated with detrimental health issues. Whereas essential and micro elements ingestion above a permissible limit also is connected to several health hazards. Therefore, a systemic study to monitor safety is also required. Our study will also provide the overview of health benefits that are found to be, if the elements, macro elements or trace elements are found to be within permissible limit. The methodology that I have followed here is, I have taken 6 anti-diabetic polyherbal formulation from Indian market and given them a sample code as BG, DB, DT, MA, MH, and SN. And further, metal analysis of 35 elements were done by handheld X-ray spectrophotometer. Health risk assessment was done by the 2 formula that was given by United States Environmental Protection Act in 1989 of estimated daily intake, which takes into account the body weight of an individual and also the amount or daily dosage of these formulation. And further, these estimated daily intake values were taken in account to calculate the hazard risk associated with the following elements. Our results are shown here. The first figure shows the spectral beam for all the 6 anti-diabetic polyherbal formulation. This is shown here for all the 6 of them. And our result has shown the concentration of metal was found in various significantly. And one of the reason would be the different kinds of herbs that were used in all the different 6 antidiabetic polyherbal formulation, and the geochemical characteristics of the soil from where the plant would have been collected. Among the 35 elements tested, these following elements were not found in all the 6 antidiabetic polyherbal formulation. Hence, it can be concluded that these polyherbal formulation are devoid of all the following elements that are mentioned here. The highest concentration of elements that were observed in all the 6 antidiabetic polyherbal formulation was calcium, potassium, and phosphorus. Further, this table shows a decreasing order of elements that were detected in 6 antidiabetic polyherbal formulation. Here we can also find out what are the elements that are present in respective polyherbal formulation. As we can see in the polyherbal formulation DT, a large number of elements were detected, which again reduced in DB. And macro elements that were detected in all 6 polyherbal formulation was found to be in permissible limit, which was in accordance to dietary reference intake. In this table, I'm showing all the concentration of the elements that were detected in all the 6 antidiabetic polyherbal formulation for 35 elements in ppm. I have found out that potassium was present in all the 6 polyherbal formulation, and potassium, one of the role for potassium is it controls glucose level in humans. Also, manganese was detected in all the polyherbal formulation. One of the role of manganese is it is associated with carbohydrate and fat metabolism. Further, magnesium is also found in the polyherbal formulation DT, which is again associated to control glucose level. Zinc was also found in all the polyherbal formulation, and zinc, it has been reported previously that deficiency of zinc is related to diabetes mellitus. Zinc is essential since human body do not have the specialized storage system for zinc. Therefore, the daily intake of zinc is essential for maintaining the zinc level in the body. Further, from the the concentration of elements that were found in the all the polyherbal formulation, I have calculated the estimated daily intake of all these 6 polyherbal formulation and 35 elements. Further, from this estimated daily intake, I have calculated the target hazard quotient. Here in chromium was also found to be with exceeding the level, permissible level. The permissible level of chromium was found to be was given by 20 ppm, but in my polyherbal formulation MH, it was found to be 30 ppm, which can be considered as health hazard. Whereas the target hazard quotient calculation, it was found out that chromium was below the risk level. One of the reason for this may be the dosage and also the time interval of how long the chromium is being exposed was less for it being a health hazard. Whereas the polyherbal formulation MA had rubidium exceeding the risk level, and rubidium, but the previous reports of rubidium has shown that it is rarely associated with toxicities, and it is rapidly excreted in sweat and urine. Rubidium is used in treatment of also used in treatment of antidepressant and goiter. Total hazard quotient for these elements, silicon, zirconium, thorium, and bismuth, was not calculated because there was lack of study on dietary intake or reference intake, and hence total hazard quotient was not calculated for these elements. This is a pie chart that I have made, which shows the percentage contribution of each element on the target hazard quotient for all the 6 antidiabetic polyherbal formulation. As we can see in antidiabetic polyherbal formulation DT, DB, and MA, rubidium was found in major contributor to the total hazard quotient. When I go to polyherbal formulation BG, lead was found to be a major contributor. Again, uranium in polyherbal formulation MH, and lead again in polyherbal formulation SN. from the summary of my study is, in India, a large number of people relies on herbal source of medicine. Also, diabetes, as we have seen in the first slide, that 69.2 million people are affected with diabetes. Therefore, the chances of people using anti-diabetic polyherbal formulation increases. Hence, I have from the 6 formulation that I have collected for my study, 5 of the formulation, namely MH, DB, DT, SN, and BG, were devoid of any health risk associated with total hazard quotient. Total hazard quotient of rubidium in MA were found to be more than unity, and it can also pose threat for longer exposure time. The macro elements were found within permissible limit and hence may enhance proper biological function. Elements such as potassium, calcium, manganese, zinc, chromium, and also magnesium are associated with stimulation of insulin action. Hence, it might help in management of diabetes mellitus. The presence of these elements in the polyherbal formulation might help in management of diabetes mellitus. Therefore, to avoid toxicities and safety issues, stringent guidelines are vital for preparation of herbal formulation. Also, health risk assessment regarding elements can be considered as essential criteria for valuation of these formulations. I am very thankful to my guide, Dr. Mala Majumdar, and Dr. Daisy Joseph for doing meta-analysis for me. These are my references. Thank you.
Thank you very much, Paul, for nice presentation. It's quite interesting study because if you know, India is deemed as a capital of the diabetes, you know. So very soon, total number of patients, I mean, those are having diabetes, India will be #1.
Yes.
So in that context, since you are working on diabetes, I have two general questions, because from the floor, I don't see any questions. So I will ask a couple of questions. The formulations which you have collected, all you collected from the Indian market. So I just want to cross check, all these were manufactured in India or somewhere imported from other countries?
No, sir. All the formulation was manufactured in India only, like Indian companies.
So that means all local products, right?
Yes, sir.
Okay. And one general question I want to ask you, because since you are working on diabetes, because prevention is better than cure, isn't it?
Yes, sir.
So we can have formulations, we can have insulin, we can have different things. This one is a cure, means after having diabetes. So what we should do, I mean, to prevent to have a diabetes means, I mean, they're very broad, but what do you think?
So I think what we can do from our part, suppose if it is genetics and all, we cannot help it out with, but maybe we can delay it with the food habits, since it is a lifestyle oriented disease. So exercising and food, healthy food and exercising can help, sir, I feel.
Okay.
The sedentary lifestyle is one of the major reason of diabetes, sir. So
yeah, true. Yes. Any other question from the floor? So there is no any question from the floor. I just want to signal. So thank you very much for wonderful presentation, Sweta. Keep it. I mean, sorry, Paul. Please keep your work, good work. Take it further, and hopefully you will achieve your goal. So thank you very much
thank you, sir,
for your presentation. So let's move on to the next presenter, presenter number 3 in this session. So our next presenter is Selvaraj Mohana Rupan. The presentation title of this speaker is Biosynthesis of Copper Oxide Nanoparticles and its Utility in Organic Synthesis. So this speaker, Selvaraj, is working at Department of Chemistry as an assistant professor, senior grade at School of Advanced Science, Vellore Institute of Technology, Vellore in Tamil Nadu. So with this brief introduction, I want to invite Selvaraj to start the presentation. Selvaraj, floor is yours.
Sir. Yeah, I'm audible, sir?
Yes, sir.
Yes, sir. Very good evening to one and all present here. Myself, Dr. Selvaraj Mohanurban, Department of Chemistry, VIT Vellore. Today, my topic of the presentation is biosynthesis of copper oxide nanoparticles and its utility in organic synthesis. So my content is I'm going to discuss about the introduction, what is green chemistry, okay, what is the uses of green chemistry, then literature background, what are the reports are available for the preparation of copper oxide nanoparticles by using the green methodology. So how we are overcoming with the reported method, and then next month conclusion followed by references. So coming to the introduction, biosynthesis. What is meant by biosynthesis? We can call it as the other terms, we can call it as green synthesis or biological synthesis. It means most of the research people are focusing towards the this approach, like green approach. The reason is it is a sustainable one, eco-friendly, also it is a reliable method. By using this methodology, by adopting this methodology, we can prepare various nanomaterials, even metal oxides, even hybrid materials, also bioinspired materials we can prepare. So our focus is also based on this bio approach only. So coming to the nanomaterials, there are generally naturally nanomaterials are available. Also the synthetic materials, people are focusing much towards the synthetic nanomaterial. The reason is, so based on our application, we can design the material. The synthetic nanomaterials are coming under the two categories. One is incidental nanomaterials, another one is engineered nanomaterial. What is incidental nanomaterial? It is more variable. It means it will have a different size and different shapes. You can see this image. There are spherical, like rod shape, even star, like that. Various shapes will be there. Even the compositions will be varied. So people are much focusing on the well-defined material that is called engineered nanoparticles. It will be like particular core material, organic coating, and cell material, like that. And there will be well-defined manner. It will be there. So people are focusing. So engineered nanomaterial. So coming to the synthesis of nanoparticle, this nanoparticle generally we can prepare in two approaches. One is top-down approach. Top-down approach means from the bulk molecule we are breaking into your smaller molecules. There are many approaches already available. That is mechanical milling and sputtering method, even laser ablation, lithography, thermal method. These are the various methods already available. The other approach is called bottom-up approach. From the small molecules, we are going to combine and we are bringing into the big molecules. So the examples are spinning method, pyrolysis, supercritical fluid, then CVD, chemical vapor deposition, and salt gel method. So our approach, it's based on bottom-up approach by using green methodology, that is biosynthesis. What is in my biosynthesis? So biosynthesis, we are going to use the secondary metabolites which is present in the natural source, like plants, algae, bacteria, fungi, whatever it may be. The secondary metabolites we are extracting, that secondary metabolite is going to act as a capping agent, stabilizing agent, or reducing agent. By using these chemicals, we are bringing into the nano level. Okay, right. So this is the overall pictorial representation of biosynthesis of nanoparticle. The first criteria is we have to choose the natural source, either plant, bacteria, or fungi. But when you are going for the bacteria and other things, fungi and other things, the process is a little difficult. Also, there is a possibility of getting contamination. Cost is high. So better always the plant medium is, it is easy. So easy, cheapest method. So we have to choose the biosource. What type of source we are selecting, either plants, fruits, vegetable, whatever it may be. The selection is very important. So then we have to wash with the distilled water to remove the impurities which is present in the surface. Then we have to go for the air dry. Then we have to make it into the powder by using mechanical grinder. Then the suitable amount of the powders has to be immersed in the solvents. Okay, so then after a certain period, we can filter the sample. The filtrate will contain the secondary metabolites. That metabolites will act as a capping or stabilizing agent to form the nanoparticle. By using this, even this extraction, there are many methods are there. Immersion methods, okay, even microwave assisted method. Nowadays, when we are focusing on the green chemistry, there are many methods. Even in short duration, we can extract our secondary metabolite by using microwave also. Okay, right. Similarly, the solvents, we can go for either aqueous, water, or you can use for the ethanol also, green solvent, or ionic liquid also nowadays they are using. Right. So coming to the literature report, since we are focusing on the copper oxide nanoparticle, we have gone for the literature survey. We found that some of the recent literature were very interesting that we are discussing here. So you know this is called Tamarindus indica. This is turmeric. So what they are doing is they are taking this leaf as well as this fruit. Okay, both they are taking, and from this they are extracting the secondary metabolite. By using the secondary metabolites, they are preparing the copper oxide nanoparticle. It means the secondary metabolite is acting as a stabilizing, capping, and reducing agent. Once the nanoparticle is formed, they have measured the size. So by using fruit extract, they got 5 to 10 nanometer. Whereas leaf extract, they got 50 to 100 nanometer. Then they compared the nanoparticles like copper oxide with their catalytic behavior against the rhodamine B. It's one of the molecules. They want to go for the degradation, photocatalytic degradation. So they got around 65% in fruit mediated, leaf mediated preparation, whereas in 77% it is fruit mediated copper oxide nanoparticle. That is, less size is giving more percentage of reduction. Similarly, in another report, they have used the flower, that is Mimoso hameta. That flower, they have flower extract, they have used for the preparation of copper oxide nanoparticle. They got they have used calcination around 500 degree in this report. Also, this nanoparticle, they have screened for the antibacterial activity. They got good results. Then apart from that, some other report we have looked. So the garlic, in the garlic, the scientific name is Alim santivum. This has been used for the preparation of copper oxide nanoparticle. They obtained the size between 20 to 40 nanometer, and they have done the antibacterial activity of copper oxide nanoparticle. Similarly, in other report, they have used Herbaceum thapsus. This is one of the plant. So that they have used for the nanoparticle preparation, and they have done antibacterial activity. This made us to concentrate for the preparation of copper oxide nanoparticle. So since most of the people are concentrated on biological activity, so we want to concentrate on the organic application. It means pharmaceutical applications, because in pharmaceutical there are many organic transformations going on to prepare the drug molecule. So we want to utilize our catalyst, our molecule copper oxide nanoparticle as a catalyst for the organic preparations, right? So our plant selection is Indicofera tintoria. It is one of the plant easily available. It is having a various medicinal property. It will cure even fever, asthma, stomach pain, liver disease, skin condition, various things. Apart from that, it is having the tendency of making blue color. Okay, so it is having some dye substance. For our hair purpose, we can make, if you want to convert your hair as a blue in color, so we can use this leaf extract. So the main phytoconstituent responsible for this phytoconstituent present in this plant is like glycoside, flavonoids, terpenoids, even alkaloids is present in that. The color blue color is formation is due to the indicotin molecules. Okay, so that's why we have chosen this molecule. It is easily available. So what we have done is we have collected this plant powder directly from the Ayurveda shop. So we have soaked with the distilled water and heated by 60 degrees Celsius for 3 hours. So after that, we have checked the TLs each and every 1 hour. Once the secondary metabolite is appear, then we have filtered it, and we have taken the extract. Then that extract has been mixed with the copper acetate solution, 1 millimolar solution. Then it has been heated. So we have optimized with various conditions. Then we have gone for the centrifugation, and we gone for the calcination purpose. Okay, 500 degrees Celsius, we got done calcination. So you can see here, this is the XRD pattern for copper oxide nanoparticle. It was exactly matched with the JCPDS number. It is nothing but joint committee powder diffraction data. It was exactly matched. So the formed particles are copper oxide nanoparticle. Still, we want to prove that our plant extract is acting as a capping and stabilizing agent. We have done our experiment even without plant extract also. That is controlled experiment. During that time, we have not observed the copper oxide peak. We have taken the XRD pattern, so we have not observed the copper oxide band. Okay. So further, we have gone for the SEM analysis. It was so spherical in shape. Then for identifying the elemental things, so we have gone for the EDAX analysis. It was clearly so copper and oxygens are present in our sample. So further, we have confirmed by the TEM analysis our samples. So it was around 30 to 40 nanometers. It was distributed from 10 to 60. The most of the nanoparticles are 40 to 50 nanometer. Then we thought of applying in as a catalyst in organic transformation. So as you know that medicine is one of the important things. So for producing the pharmaceutical, like producing the pharmaceutical important molecule, some functional transformation is required. So in this, carbon sulfur bond formation is one of the important thing. So we have chosen CS bond forming reaction. So we have taken this molecule thiol. To that we have reacted with aryl iodide. So we observed that this is our product in the presence of KOH, dioxide, and copper oxide. What we have done, we want to study the effect of reactant. For that, we have changed various reactant. One is aromatized, then aromatic molecules, again another one aromatic molecule. Even simple and difficult aromatic molecules we have changed. We found that in all other conditions we got good yield. Okay, so this is the optimized parameters. We have varied the base, we have varied the time, as well as we have checked the catalyst also with various catalysts like copper acetate, copper sulfate, even copper oxide nanoparticle. What we have prepared 5 mole, 10 mole, like that we have varied. We observed that in the presence of 10 mole percentage of copper oxide at 80 degrees Celsius with the 8 hour, we got 87%. It is a higher yield we observed. Then again, we want to try for a little bit, we increase. So we tried for 20 mole percentage. There is a slight changes, only 2% was observed. So around 89%. So even if you are having 10 mole percentage, you are getting higher quantity of the yield. So further, the molecule has been confirmed by proton NMR, C13 and mass analysis. As a representative sample, I here with presenting only one analysis, proton NMR. You can see here the CH3 is nothing but CH3 group which is present in the docile group, docile here. Then all other aromatic proton, 1, 2, 3, all these aromatic protons are up between 7 to 8.5. So we have confirmed this molecule by proton NMR. Further, we confirmed by C13 analysis. Also, we have confirmed by the mass analysis. So coming to the conclusion. So successfully we have prepared our molecule in green route. Successfully we have prepared a copper oxide nanoparticle by utilizing our plant extras. Also, we have used our prepared catalyst for carbon sulfur bond forming reaction. That is one of the important reaction in pharmaceutical industry. These are some of the references. Thank you. I would like to thank organizing committee for giving me this opportunity. Also, I would like to thank the management of Vellore Institute of Technology for their constant support and encouragement. Thank you.
Thank you very much, Dr. Selvaraj, for wonderful presentation. I think it's a good piece of work, and I can see even though you are in chemistry, so you can contribute for developing something useful for the healthcare industry.
Yes, sir.
So from the floor, there are no any questions. So I will ask you a couple of questions.
Yes, sir.
Because while talking, you have mentioned tamarind. So since you are working in that area, so do you think, is there any role of tartaric acid? I mean, secondary metabolites. I mean, you was talking about secondary metabolites. So
yes,
tartaric
sir,
acid, is there any role of tartaric acid?
Yes, sir. Yes, sir. Of course. Actually, this is part of our work. We are mainly concentrating on the agricultural waste. For example, Coccus nigrifera, coir, like that. The main role is, if you are having the secondary metabolite, what will happen? It will act as a reducing agent. Okay. For example, if you are having the polyphenol, it will convert it into a keto. Okay. It will get oxidized. When it is getting oxidized, this salt, metal salt, if you are having suppose copper 2, it will become a copper oxide. It will get reduced. Instead of using some chemical reducing agent like sodium borohydride or lithium aluminum hydrate, we are going for the green chemical. The reason is, if you are using these chemicals, lithium borohydride, sodium borohydride or lithium aluminum hydrate, when you are going for the industry level, bulk level, there is a possibility of getting accident. Whereas if you are going for this natural source, biosynthesis, we can prevent the accident. Also, only less quantity we can get our product.
Okay, interesting. So another general question I have, yes, because I mean, your work is quite, I mean, interesting, and that one, I mean, this brought a thought in my mind. Yes. So because you have done this work, so how your research findings will be useful for healthcare industry in the near future?
Yes, actually, our aim is mainly focusing on the pharmaceutical company. The people's what they are using is, whenever you are doing the process, when you want to prepare some drug, you have to go for multiple steps. Okay, during each and every step, they are using different solvent, mainly organic solvent. For example, you can say that methanol chloroform, chlorofluoro compound. It is obviously toxic to the environment. So when you are using our catalyst, you can reduce the processing time. Also, when we are proposing the method, we are trying to use the green solvent like water. Okay, water and ethanol. So when you are using this type of solvent, it is obviously environmental safe. Another thing, the process is less. you can get higher amount of yield. So they are producing the pharmaceutically important drug, so which will be useful for the society.
Okay. Thank you very much, Dr. Selvaraj, for sharing your research findings.
Thank you,
it was
sir.
a wonderful, very good piece of work. Thank you very much.
Thank you, sir. Thank you so much.
Okay, so let's move on to the next speaker. So, I would like to invite my co-chair, Associate Professor Dr. Sivakumar, to introduce the next speaker. Dr. Sivakumar, over to you.
You can unmute your mic.
Yeah, Dr. Sivakumar, yeah.
Am I audible, sir?
Yes, yes. Yeah, please go ahead. Yeah.
Okay, so now we will move on to the next presentation. So the next presenter is Ms. Shobha Gunasekaran. Actually, she is working in the Department of Biotechnology, Mother Teresa Women's University from Tamil Nadu, India. So her topic of presentation is molecular interaction analysis of lanosterol derivatives from Letiporus versisporus as potential inhibitor of BCL2. So now I would like to invite Ms. Shobha Gunasekaran to carry on with her presentation.
So can you hear me, sir?
Yeah. You can start sharing your screen.
Yes, yeah, yes, sir, I'm sharing. A very good evening to one and all present here. I'm Shobha. I'm a research scholar from Mother Teresa Women's University. I'm working as a lecturer in Vijayavashmi College, Chennai. And my topic of study is molecular interaction analysis of lanosterol derivatives from Letiporus versisporus as a potential inhibitor of BCL2. So this Letiporus versisporus is an edible mushrooms. Usually, medicinal mushrooms have a long history of traditional oriental therapies, and fungal metabolites are increasingly being used to treat wide range of diseases. And edible mushrooms have been used to enhance the immune system activity as well as it has a tumor activity. This Letiporus versisporus has a long history of consumption, especially in North America and Japan. So now I have chosen a species called Letiporus versisporus, which is a wild edible mushroom which was widely used, especially in the rural areas of Kodaikanal. This has different morphological features, which has a mature fruiting bodies and are semi-globulous and has abundant chlamydospores in the context. So this this particular species I have been working for my PhD work. I have done an animal study for the liver cancer and it has shown a positive results. So I want to find what were all the derivatives which was involved in the mechanism. So I have chosen a lanosterol derivatives which have been published from this particular Letiporus versisporus, namely letiposide B, E, F, and G, eburocoic acid, trimethanolic acid, and sulfuronic acid, which was published by the author Yoshikawa et al. In the year 2000. These lanosterol, it is a tetracyclic triterpenoid that is lansto 8,2,4-diene substituted by a beta hydroxy group at 3 beta position. So this triterpenes usually from plants, animals, or fungi, which has anti-cancer property, anti-inflammatory property, and antioxidative properties. Currently I was working on liver cancer. So liver cancer, it is a growth of abnormal and unhealthy cells in the liver. It is a most common type of cancer that is hepatocellular carcinoma, begins in the main type of liver cell called hepatocytes. So it is one of the 6th most common cancer in the worldwide, which accounts for about 7,82,000 deaths in 2018. And in long-term association, it has long-term association with hepatitis B and C infection linked to liver cancer because it often leads to cirrhosis. The majority of hepatocellular carcinoma develop on the basis of chronic inflammatory liver damage leads to the dysregulation of multiple cellular signaling pathways. So in the current study, I have chosen a gene called BCL2. The mechanism of BCL2, this BCL2 gene generally expressed in certain tumor cells. It is an important factor in regulating the apoptosis. BCL2 family proteins regulate both pro-apoptotic protein and anti-apoptotic signals and thereby mediating the mitochondrial membrane potential. The overexpression of this anti-apoptotic BCL protein, that is BCL2 and BCLXL, is found in most common cancers like hepatic, colorectal, breast cancer, lung cancer, and ovarian cancer. So the targeting the apoptotic protein, specifically these BCL2 family proteins, have an attractive strategy for the treatments of cancer. So this is the mechanism of BCL2 in the apoptosis. Normally, mitochondria contains pro-apoptotic and anti-apoptotic protein belong to this B cell protein. This anti-apoptotic protein BCL2 and BCLXL will inhibit the cytochrome C. This due to the mitochondrial dysfunction, this back and BID. So the BAX is BCL2 associated X protein, BAC is BCL2 homologous antagonist killer, and BID is BH3 interacting domain, which is a pro-apoptotic protein. Due to the mitochondrial dysfunction, it was released in cytosol, and it promotes the release of cytochrome C and deoxyadenosine triphosphate. This in turn bind to APAF, that is apoptotic protease activating factor, which to form a multimerit complex subunit recruits the activation of procaspase 9, which in turn activate the caspase 3, which forms the results in the cellular apoptosis. So the main objective of this current study is to retrieve the lanosterol derivatives and a cyclophosphamide structure from the PubChem databases, which is a biological databases where we can retrieve the compound. And next is the structure of BCL2 were retrieved from PDB database, and the drug likeliness property of the compounds were being predicted using mole inspiration tool. And to perform molecular docking analysis of these lanosterol derivatives and the control drug against BCL2, I have chosen a control drug cyclophosphamide for the current study. And the last step is to view the docking interaction using BioVia Discovery Studio. So the methodology, so from the PubMed, I have chosen the disease and its mechanism, and I have chosen a protein BCL2, and I have studied the mechanism of the protein, that is a pathway related to BCL mechanism, and I have retrieved the structure from PDB database, and compounds data that I have retrieved from PubChem databases. I have drawn the chemical structures using ACD Chem Sketch and optimized the 3D structure and converted into mole format to PDB format using Open Babel converter, and finally predicted the properties using mole inspiration tool. And the structures, that is the protein structure, PDB structure, and protein PDB structure and the compound PDB structure is subjected to auto docking, that is a molecular docking software, Autodock 4.2, which is widely used software for protein ligand docking. This software will prepare protein ligand, as well as we use in a protein and ligand in a particular grid, and we'll run a grid. Later, the docking would be run by the using the genetic algorithm. We'll get the 10 confirmation based upon based upon the binding energy. We'll select the best confirmation and we'll build the confirmation and visualize the docking interaction using a BioVia Discovery Studio visualizer. So next is ligand preparation. So ligand preparation, as I told you, the structures I have taken from PubChem database. I have with the cyclophosphamide drugs, and I have drawn the structures and converted it, saved in MDL mode format, where it is optimized and converted to PDB format using Open Babel. These were all the structures, that is 2D and 3D structures of the compounds which I have chosen for the study. And this cyclophosphamide drug, which is already currently using for hepatocellular carcinoma, it is nephrotoxic. When there was a long-term usage, this drug was nephrotoxic. So, so there could be an alternative strategy. So I have chosen this drug as a control drug to check my activity, to check the compound which I have taken for my activity. So these were the bioactive properties that I have predicted from the mole inspiration tool. This is a molecular weight, hydrogen bond donor, acceptor, and log P and rotatable bonds, and topological polar surface area, which was linked to the drug absorption, intestinal absorption, and the volume of the drug and number of atoms present in the drug. So the main property we'll check is donor, acceptor, and log P, and number of rotatable fault present in the compound. Which there was 1 or 2 violations present in the drug, but we have chosen these drugs because it has potential properties. So next important property that I have checked is the drug likeness property of these drugs. So it has shown the positive value for enzyme inhibitor activity, which is equivalent to the which is equivalent to around 0.4 greater than, and I'm sorry, sir. So this value is greater than 0.4. So it has the enzyme inhibitor activity. The compounds which I have chosen have the enzyme inhibitor activity. And this is a structure that I have retrieved from PDB database. The protein, for protein preparation, the structure which we have downloaded is the extra crystallographic structure of BCl2, where the water molecules were removed and co-crystallized ligand were removed from the protein molecule. So then this is the docking results of the derivatives. So based upon the binding energy, I have tabled the results. The amino acid atom of BCl2, the interaction with the compound atom with the hydrogen bond. So I have tabled the results of hydrogen bond atom, hydrophobic atoms, and van der Waals atoms. So this from the results I have suggested this trimethanolic acid has the least binding energy of minus 9.38 kilocalories per mole, and compared to the cyclophosphamide, which utilize minus 6.68. So the least the binding energy, the best the conformation would be, and it has shown 3 hydrogen bond interaction for trimethanolic acid, and 4 hydrophobic interaction and 5 van der Waals interaction were produced. Whereas in the case of cyclophosphamide, it also shows 3 hydrogen bond and no hydrophobic interaction, and it shows van der Waals and electrostatic interaction. So this is a confirmation of docked position of trimethanolic acid with BCl2 showing minus 9.38 kilocalories per mole energy. This is a docked position of BCl2 utilizing the energy minus 6.68 kilocalories per mole. So this is the hydrogen bond interaction that is shown in green color dotted lines, and pink color is the hydrophobic interaction, and the green color surrounding atoms is your van der Waals interaction. Whereas in the case of cyclophosphamide, it shows 2 hydrogen bond interaction and one hydrophobic one charged interaction which was shown in orange color, and this is a van der Waals interaction which was shown in light green color. So conclusion, among the 7 lanosterol derivatives, trimethanolic acid shows a promising inhibitory activity against BCl2 with the binding energy of minus 9.38 when compared to the cyclophosphamide, which utilized minus 6.68. The further analysis of this trimethanolic acid on experimental study on animal models to study its mechanism on hepatocellular carcinoma. Thanks for the opportunity given. These were the reference I have chosen for my study, and thanks for the opportunity given to me. Thank you.
Okay, thank you, Ms. Shobha, for the very nice presentation, very informative presentation.
Thank you, sir.
And we have a few questions from the audience. The first one is, how are the molecular properties checked? Which docking software did you use?
I have used Autodock for my studies, sir. Actually, I have used Autodock 4.2, which utilizes genetic algorithm for building the confirmation. That is, it runs several times. It will run the simulation several times. I have used Autodock 4.2 software, sir.
Okay, fine. There's one more question, madam.
Yeah.
What are the specific objectives of the study? Because you mentioned general objectives that can be further published.
Yeah, actually, like I have already, it was a part of my PhD work. I have done on wet lab animal model study with the ethanolic extract of this particular mushroom. So I want to understand what is a particular compound that involves in the mechanism. So I have collected from the article, already published article, that NMR compounds published in the NMR studies, they have published this compound. So I have chosen this compound. I have and studied about this concept, this particular derivatives which has the activity of anti-cancer activity. So I have want to check the interaction. So I have done bioinformatic analysis for this.
Okay, fine. And is there any specific reason why you have selected this species? Because
actually, this species was edible species in Colorectal mushroom. People eat. So this is a rare species. No work was gone on this particular species, Veticulus versus Chorus. Only this particular compound alone was published in the year 2000. No other novel works have gone in this particular mushroom. So I have chosen this mushroom and I have chosen this extract for my study.
Right. Okay,
yes.
ma'am.
Thank
Very
you,
nice
sir.
presentation. Thank you once again, Ms. Shobha.
Thank you, sir. Thanks a lot, sir.
Move on to the next presentation. And next presenter is Ms. Tej Emma. She is from Department of Botany, University of Science Technology from Meghalaya, India. And her topic of presentation is isolation and characterization of biosurfactant producing bacteria from hot spring zone of West Kameng district, Arunachal Pradesh, India. So I welcome Ms. Tejyama. So you can start your presentation now. Is she there? Can you hear me, Ms. Tej? Hello?
Tej, are you there?
Yes, sir.
Can you hear me, ma'am?
Yeah.
Yes, sir.
Okay.
Yeah, you can share your PowerPoint presentation.
Yeah, you can start your presentation. You can share your screen.
You can share your PowerPoint presentation.
Yes,
you can
sir.
Start.
Sir. So is it visible now?
Yeah.
Yes. Yeah.
Okay. Good afternoon, everyone. Myself, Tageyama. I'm a PhD scholar from Northeastern Regional Institute of Science and Technology, India. So today I'll be sharing on the topic isolation and characterization of biosurfactant producing bacteria from hot spring zone of West Kameng district, Arunachal Pradesh, India. So first, brief introduction about the biosurfactant. Biosurfactants are the biosurfactants are an amphiphilic compound which can reduce the stress of the particular area by reducing the surface tension between the molecules. It might be liquid to liquid or solid to liquid. So these biosurfactant compounds are synthesized by microorganisms like bacteria and fungi. So they can also help in, they can also increase the biodegradability of insoluble pollutants. So biosurfactants possess the properties like low toxicity, biodegradability, and they can tolerate the high pH and temperature also. So because of such properties, they are used in fields like food industries, pharmaceutical industries, and cosmetics, and many others. So based on their structure, they might be of linear and spherical. These are the objectives of my work. First was to isolate the bacteria from the samples, and since only some of the bacteria, only some bacteria, because only the some bacteria will only some bacteria produce the biosurfactant compounds, not all the bacteria. So screening was done for the biosurfactant production, and their kinetics of biosurfactant production was studied, and later the characterization of those efficient bacterial isolates were done, and lastly the optimization of different carbon sources were done by using different oils. So in methodology part, here it is given a brief detail about the study site.
This hot spring is situated in Dirang, that is in Arunachal Pradesh, India. So this slide showing the photographs of the sites, and this is the exact location. This map is showing the exact location of the site. For pH, the standard protocol was maintained, and it was determined by using the electronic digital pH meter. Then for the isolation of bacteria, the media used was enrichment media, which contained both the mineral salt media and nutrient broth media in 1:1 ratio, and 2% of kerosene oil was used in this media, and their incubation temperature was 30 degrees Celsius. And after incubating for 5 days, a small amount of inoculum was taken out from that culture, like 5 ml, and it was transferred into the next fresh media, and again they were incubated for next 5 days, and this process was repeated for next 4 cycles. And from the fourth set, the bacteria was isolated on a nutrient agar plate by strict plate method. And for screening test, 24 hours old culture were used, and the method used was emulsification index assay. And for the kinetic studies, again, the same method was used, that is emulsification assay. And here the cultures were incubated for 12 days. And the emulsification assay was done every after 24 hours, continuous for 12 days. So for characterization of bacteria, these 3 tests were done, that is gram staining, biochemical test, and cultural characteristics. For the optimization of carbon source, three different types of oils were used, that is crude oil, coconut oil, and kerosene oil, all in 2%. They were used as the sole source of carbon. So for optimization, the cultures were incubated for 7 days. So after the incubation time, again this emulsification assay was done, and the result was recorded. So so in the result portion, first pH was found to be different for both the site. In the first site, it was alkaline, and second site is what it was acidic. And total 22 bacterial isolates were isolated, 6 from site 1 and 16 from site 2. After isolating these 12 bacteria, the screening of biosurfactant was done, and only 12 isolates were found to be positive, and the rest were showing negative for biosurfactant production. So this slide is showing the pictures of this isolation and screening. So in kinetic studies, the different isolates show the different production on different days. It was done, since it was done for continuous 12 days, it was incubated. So we can see from this table that some isolates did not produce, some isolates were not so efficient, and others were more efficiently producing this biosurfactant. So those isolates which were producing more than 50% were selected for the further studies. This slide is showing the result of gram staining. The first isolate was found to be gram negative and rod-shaped, and second isolate was gram positive spherical, and third isolate was gram negative and rod-shaped. So this is the result of biochemical tests. These are all the results of biochemical tests, and here in this table, it is the compilation of both morphological and biochemical test results. So this table was compared with the data given on the book of Microbiology Laboratory Manual by Cappuccino and Sherman, and a research paper of Rozanski et al. 1950. These 3 isolates were identified identified as the Pseudomonas micrococcus and Stenotrophomonas species. And for the result of optimization of different carbon sources, it was found that the isolate 1 and isolate 4 was showing the maximum production when the kerosene oil was used, and in case of isolate 3, crude oil showed the maximum production. So this is the summary and discussion of my work, which I have already discussed earlier. So moving to the last slide, in conclusion, I can conclude that from this study, I can confirm that some high number of bacteria are present in that Dirang hot spring, which can produce a significant amount of biosurfactants, and since that hot spring is surrounded by a vast forest, so this bacteria might be helping them to adapt to that environment by reducing the stress of that area. So also this bacteria can use the oil as their carbon source, so they can also increase the biodegradation of insoluble pollutants, especially in oil polluted environment. So because of their non-toxic nature, they can be used in industries like cosmetics and pharmaceutical and food industries also. So this study can be considered as a fundamental work for other researchers, those who are willing to research on a topic related to biosurfactant producing bacteria. Thank you, sir.
Okay, thank you, Ms. Tejiyama, for nice presentation, for sharing your research findings with us. Thank you. And we have a few queries actually from the audience. The first one is, why did you take different number of samples from the sites instead of same amount or ratio? So are you not considering bias towards the selection for the isolation?
I'm sorry, sir. Can you please repeat the question?
Why did you take different number of samples? You would have taken same amount of the samples at the same ratio, right? So don't you think that will cause some bias towards the selection?
No, sir, because the site I have chosen is not far from each other. It is located almost 10 meters apart from each other. So I have collected same amount of samples from both the sites.
Okay. And actually, there is one more question. Is the incubation done while centrifuging? Because you mentioned it is 30 degrees.
Yes, sir. Yes, sir. Yes, sir. During incubation, it was kept at 150 RPM.
Okay. Fine. So, I mean, just one general question.
Yes, sir.
Is it like which one will be cost effective? Is it biosurfactants or synthetic surfactants? Of course, biosurfactants have a lot of advantages over synthetic ones, but
yes, sir.
regarding if you take cost into consideration, which one will be the cost effective?
Sir, if we talk about the cost efficiency, then now this synthetic surfactant will be more cost efficient, because for this biosurfactant it is for culturing them also we need this this is very expensive. I mean, the medias and all we are using, it is not so cheap. So
Okay, yeah, that's why I'm asking. Okay, fine. Anyway, nice presentation. Nice presentation, Tejam. Thank you.
Thank you, sir.
So before we go to the next presentation, I think we have, I'll invite Dr. Subhash, chairperson for this session, to invite the next speaker. We have a plenary session, if I'm not wrong.
Okay.
Thank you very much, Dr. Sivakumar. Audience, those are online either on YouTube or Zoom or Facebook. I just want to update you because there was one plenary talk in session number 4. That one was not completed, so that plenary talk number 6, the speaker was not able to join because of some technical reasons. So the speaker is now able to join. So sorry for the interruption because we have to include his talk in between because of this technical reason. So I'm going to invite Professor Balakrishnan to give his plenary talk. But before he starts, I just want to introduce him. I have his very, very brief CV in front of me. So I'll just introduce him by taking the points from his CV. Professor Balakrishnan, he is serving as a head professor and head of the Department of Immunology, and he has completed his PhD from the same department. It's quite interesting. Then, Madurai Kamaraj University in year 1994. He cleared UGC's, UGC means it's a University Grant Commission of the Indian government. He clear NET JRF, which is conducted by Government of India. In 1986, he cleared this exam. Then he was awarded with Robert Cochrane Overseas Travel Fellowship from Royal Society of Tropical Medicine and Hygiene, London, UK, for training in microsatellite mapping in leprosy families at Wellcome Trust Center for Human Genetics. His research specialization is in HLA immunogenetics, and he's working on immunogenetics polymorphism and their susceptible and protective associations in a number of diseases. Presently, he's vice president of Indian Immunology Society, New Delhi. He has published more than 50 research papers in national, international peer-reviewed journals, including nephrology, tissue antigens, and nature genetics, as well as PNAS and Journal of Infectious Diseases and some other well-known reputed journals. He has completed a number of government-funded research projects, he managed to get a huge amount of money as a research grant of 2 crores and has organized several workshops, conferences, and last but not least, he is offering HLA tissue typing and donor matching services to a number of hospitals through industrial consultancy cell MKU. So with this brief introduction about Professor Balakrishnan, I want to welcome him so he can deliver his plenary talk. Professor Balakrishnan, can you hear me? Professor Balakrishnan, can you hear me?
Yes, sir.
So, sir, floor is yours. So you can share the slides and you can start the presentation.
Yes. Sir, good afternoon. It's a very nice introduction about me. Thank you very much, sir. I thank the organizers for having this nice opportunity for me to talk to present my talk on HLA molecular medicine perspectives. The experiences that we gained at Madurai Kamaraj University over last 30 years. As he pointed out, I joined research scholar in this department, and I later joined as a faculty continuing on the work on HLA. I'm sorry that I missed my bus in the morning, but anyway, I'm happy that I'm be with you all. Thank you at the outset. So my HLA system is a highly migration in Africa. Then human populations moved to Middle East region. Today's Middle East. It goes last 20 years as a lot of molecular genetic evidence has supported this theory of human migration. So in the course of this migration and the adaptation of agriculture has introduced a huge number of pathogens into fungal viral infections, which resulted in increased mortality, and also the humans are transferred from hunter-gatherers way of life to disease, settled way of agricultural groups in which the disease burden is very high. So the point is that over this period of migration and movement of human populations across continents, in human body also, due to mutations, HLA system, human leukocyte antigen system that we are going to see today, also evolved. A lot of molecular evolution takes place in hand in hand with the pathological evolutions. So this is a human leukocyte antigens. To brief, it is present in the chromosome number 6, very big, highly diverse complex in human genome, and which is basically broadly divided into class 1 genes, class 1 alleles, class 2 alleles, and class 3 alleles. The class 1 and class 2 alleles are important for immune response. The class 1 alleles include HLA A, B, C proteins, and HLA class 2 comprise of HLA DR, DQ, and DP proteins. These molecules are very important in presenting the pathogen-derived system. Is a Jean Dossier from Paris. In 1980, he got Nobel Prize for the discovery of HLA system. As I said, it is a highly complex system in which occurs 1,000 different alleles. So in a given individual, say for instance, if an individual is getting 2 alleles from father and 2 alleles from mother, means out of these 6,000 alleles, 2 alleles are transferred to he or she. Likewise, fetus from father and mother. So such a complex system of immune response genes are involved. So this which comprise of A, B, C, D, R, DQ, and DP alleles. As I said earlier, the huge number of alleles are, and any permutation combination, say for instance, 2 alleles from A and 2 alleles from B, the possibility is one set out of 71,30,475. Such a very huge diversity is there in selecting the HLA alleles into their newly developed, new sibling. So if you take one more locus, HLA-C locus, the possibility is very huge. That is 2 from A and 2 from B, 2 from C. So such a diverse system is the HLA system. There is one more system called T-cell receptor, which is also very important in antigen presentation and recognition. So if the MHC properly presents the peptide to the T cells by its T cell receptor recognize the peptide, and hence if it is a foreign and immunogenic, they produce cytokines, attract more cells to the site of infection, and immune response proceeds further. Likewise, the other diverse molecules in the human system is the antibodies and killer cell immunoglobulin-like receptors, which I'm not going to touch upon. We are going to restrict only with the major histocompatibility complex HLA genes. So the over the by which the HLA genes are identified, immunohistocompatibility works up over So starting from 1987 onwards, different histocompatibility recorded, documented HLA alleles. This is the number of class 1 alleles total, and these dark ones are the alleles. So the number greatly increased over the years. Means that the more the new populations are studied, the more the populations from different countries are included, more HLA alleles are being identified. So that shows the diversity of the HLA system. So the logic is the more the pathogens or the pathogen evolves, the new HLA alleles also evolve. There are beautiful illustrations with reference to the evolution. One good example is HLA and HIV we are going to see. And this will show you small gives you an idea that how big the diversity of HLA alleles. See, if you take one example, HLA A1 in European cases, frequency is 27%, whereas in Japanese it is only 1%. Whereas if you take the other allele, HLA A3, in European Caucasians it is 21%, in Japanese it is 1%. And if you see the different populations, the frequency differs in different populations. It means that allele frequency in different populations varies. It is because of the geographical and also the pathogen pressures that prevails in the environment. So put in a nutshell, for the introduction, the HLA polymorphism is hugely enormous, which is meant for combating pathogen infections. And disease diagnosis is useful for disease diagnosis and treatment options. Vaccine designing, the immunoinformatics data is being used for vaccine designing and drug designing and drug validation. Very good examples are there to validate the drug selection and administration, and also for combinatory therapy. Also, the HLA data in a population is being used. And this is how the HLA antigens present the pathogen-derived peptides. Say, for instance, if a virus gets attached to the receptor, internalize and the genome integrated with the host genome, and the viral mRNAs reaching cytoplasm produced the viral peptides reach the endoplasmic reticulum, where the HLA molecules are waiting, and these molecules, HLA molecules, picks up the antigen and present to the outside through the cell membrane. And this is how the HLA molecule, the pink one is present pathogen. This is called antigen presentation mechanism by the class 1 HLA molecules. So these are HLA class 1 molecules, and this is a class 2 molecules. Class 1 molecules is a single chain, alpha chain, having 3 domains, alpha 1, alpha 2, alpha 3 domain. This is a peptide binding cleft in which the pathogen-derived peptide normally binds. And in class 2 molecule, which is made of 2 chains, alpha chain and beta chain, there is a cleft. This is a peptide binding cleft in which the pathogen-derived peptides normally binds. And this is X-ray crystallographic structure of T cell receptor one side, the HLA molecule the other side. In between, you can able to see the yellow colored the pathogen-derived peptides. Basically called as a T cell receptor MHC peptide ternary complex. This is a diagrammatic illustration of the antigen presenting cell with MHC class 1 molecule, and T cell CD8 T cells with the T cell receptors recognizing the peptide. So in turn, these T cells produce cytokines and attracts more cells to the site of infection. Of course, this diagram illustrates the critical amino acids peptide binding group in which the HLA molecule, the pathogen-derived peptide binds and elicits immune response. These critical amino acids are highly essential or critical. The mutation in the gene of HLA and change of any amino acid in the critical position really matters, and that drastically alters or change the immune response. This is how it looks like when a peptide binds in the peptide binding cleft of the class 1 MHC molecule, and the peptides are being processed and presented. So this is normally the class 1 peptides are amino acid 1 to 10 size, and for class 2 binding, the amino acid, peptides are pathogen-derived peptides are a little longer. Peptides, this is a pathogen-derived peptide from 1 to P9, and there are P9 highly conserved amino acids are present in different HLA molecules. We take HLA A2 68, B7, whatever. But whereas in P1 or to P8, except that P2 and B9, the amino acids vary accordingly. Whereas the P2 and P9 are highly conserved. So this is very critical in recognizing the foreign peptide by using these amino acid sequences. And these are all the methods of HLA typing: serological typing, PCR SSP, PCR SSOP, SBT typing, and microarray and flow sorting methods are being used clinically to type the HLA molecules for various purposes. So this is how, after collecting the blood loaded onto a lymphoflot, a density gradient medium, after centrifugation, you can able to see the lymphocyte layer. This layer is scooped out and washed and typed on HLA typed Tersaki tray, and the dead cells are being counted under the phase contrast microscope. Even we can use fluorescent stainings to read the dead cells are in red color and live cells in yellow color. And this is a classical Tersaki plate for HLA serological typing being practiced in tissue matching in transplantation clinics. And this is PCR results for HLA DR typing. This is specific bands showing HLA DR10 and 15 genes amplified for HLA matching purposes. There is a mechanism called a linkage disequilibrium wherein the two genes are linked together for generation after generation, or in large populations, the HLA molecules are these HLA genes are joined together and occur again and again in huge populations for many number of generations. This mechanism is called as linkage disequilibrium. Say, for instance, HLA A locus allele A3, B7, C7, DR7 from DR, and DQ2 from DQ locus joined together occur in several individuals, thousands of individuals, more than expected number of times, and this principle is called linkage disequilibrium. This is very important in genetics, and more particularly very important in immunogenetics. And this is an example for linkage disequilibrium. If you take the frequency of A1, it is in a given population, it's a case study, 0.138. For another allele in B locus, B8, 0.09. If you multiply you will get the expected frequency of two alleles. It is 0.0124. Whereas normally in population studies, we observe the frequency is much more than the expected frequency. So these two alleles are said to be in linkage disequilibrium. This is a beautiful mechanism, and this is because the selected alleles are joined together and occur again and again more than expected times because of the natural selection process, because of the pathogen pressure. So this HLA study is very important in the context of disease association, and a beautiful study is illustrated earlier times. A huge number of studies were there in the world, and in Polish population, HLDR16 allele frequencies increased in TB patients, whereas in Indian population, it is HLA A locus A10, B8, and DR2. More particularly, the DR15 subtype is increased in the risk of tuberculosis in population. We do in Madurai population also the HLA-TB and several other studies. And this is Professor Adrian, with whom we collaborated some 20 years ago for our microsatellite mapping studies. He actually identified HLA-B53 in African population, which affords a protective association with towards the cerebral malaria in African population. And based on this, HLA-B53 derived peptides, he developed CTL vaccines against malaria. So these are all our studies based on which we identified 2 locus, 3 locus, 4 locus haplotypes. Beautiful haplotypes we identified in several diseases, including pediatric nephrotic syndrome, cervical cancer, type 2 diabetes, psoriasis, ischemic stroke. And these haplotypes, 7-2 is very interesting. We took this haplotype further. This is a very common haplotype in Caucasian population and also to some extent in Indian populations. The 7 and 2 were very important haplotype, 2 locus haplotypes. We will discuss shortly in another few slides. This is another application of HLA study. You can hand in hand with the molecular evolution that's happening in HIV, being published by Kaipela et al. In 2004 in Nature, in which they demonstrated the antigenic variation that is happening mutation in HLA B restricted alleles. So 25 different viral polymorphisms were encountered with B-restricted manner when they analyzed the data. Means that the HLA viral mutations are being developed with reference to there is one-to-one interaction between the viral mutations as well as the HLA frequency in a given population in a geographical setting. Of course, I'm skipping the haplotype is beautifully illustrated in viral infections. And in this study, the Chong et al. Is beautifully illustrated, explained about the HLA-A3 supertype and HLA-B7. And the A3 supertypic allele group includes A3, A11, A33, A31. Important haplotypes with reference to viral infection, and the identification of A3 and B7 restricted CTL response to hepatitis C virus patients with acute chronic infections. And also we identified this A3 B7 in pediatric nephrotic syndrome cases in South India. There is a case study we will see quickly. And this actually A3 alleles superantigen group consists of several antigens, very big group. So very importantly that this big supertypic group is useful, highly useful in viral infections and containment. Likewise, the B7 group of supertypic allele group. And this B7, DR7, DQ2 haplotype I told you that we identified in South Indian haplotype is identified to be susceptible in several diseases, myasthenia gravis, celiac disease, and several other world populations. Likewise, the same haplotype is protective in several diseases such as breast cancer, rheumatoid arthritis, T1DM, in several other communities. So means that the haplotype is same, but in different diseases, in different environmental setting, the haplotype plays differently in different populations. So this is what we identified, observed in our population. The 7-2 we observed in pediatric nephrotic cases, particularly the focal segmental glomerular sclerosis cases. 70 individuals, we got these 2 locus haplotype. When we typed further for HLA-A and B, we got 35 with the 4 locus haplotype. And we typed when we typed for HLA-C locus, 18 individuals were with the extended haplotype that we have seen earlier. The huge extended haplotype transferring from generation to generation and conserved in a population is highly dramatic. It has a lot of evolutionary significance. But of course, I'm skipping a couple of slides for the time constraint. I want to explain very important last 10-15 slides. So this is what, when we mark this extended haplotype that we identified in South India, A3, B7, C7, DR7, DQ2, and the data collected from world populations, the haplotype is identified in Australian population and Turkish minorities, Jordanians, and Northwest England population, and also in Hispanics living in US and Brazilians. And if you take the HLA data, not only the population genetic studies and all the disease association studies, for identification of or estimation of hypersensitivity, drug hypersensitivity, particularly in 2002, this particular allele HLA-B57 was shown to be associated with the abacavir hypersensitivity. And in 2004, carbamazepine hypersensitivity was recorded with individuals having B15 allele. Likewise, you can able to see so many different HLA alleles are associated with hypersensitivity with several drugs. Later, based on this hypersensitivity, the drugs are withdrawn. Best example is abacavir, which is being used for HIV drug and which is not in use now because of the extensive hypersensitivity which is developing in individuals. Drugs for which the individuals or the populations are showing hypersensitivity. So that really matters. And also another important point with reference to drug protein interaction is the abacavir, with reference to abacavir and individuals with the B57. initiates another series of immune response, ultimately resulting in a lot of consequences. And when they compare some sequences of HIV, the self-peptides, which is binding with the abacavir and leading to the hypersensitivity, the sequence homology is self-peptides which are having some HIV pathogen peptide which is having some similarities and bound with the abacavir drug metabolite elicit immune response, hypersensitivity leading to reaction of HLA molecule, and the HLA molecules present in the peptide plus the drug the unwanted consequences. This is also once again important in dealing with the drug components. So this is also the same picture. I mean, drug abacavir with the specific HLA molecules in populations. This is also some good example of development of races and other allergic reactions. So this is another new technology in which by using the avidin compound, 4 HLA molecules are attached by chemically, and these molecules are being used for cell sorting in advanced immunological assays. Other aspect of the HLA study of HLA molecules, the HLA molecules in tumors, there are different phenotypes you can classify based on the expression of HLA molecules. Phenotype 1 is a total loss of HLA, and it's a very important concept in study of HLA in molecular medicine. In tumors and some viral infections, the HLA is lost on the cell membrane. So if HLA present, they present the peptide to the T cells. When it is not presenting, when it is not present on the membrane, the antigen presentation failed. So total loss may be there, or sometimes the HLA out of 2A, only 1A alleles are expressed. So phenotype loss may be there, or responsiveness to interferon gamma. So in that way, the viral in patients. So there is a last concept of with reference to HLA technology is a selective high affinity ligand antibody mimics used for cancer diagnosis and therapy. Of course, this is by Rod Balgan's group from California. Actually, we collaborated, Madurai Kamaraj University collaborated with this group lately. And I'll just explain another 2 minutes. That is selective, what is selective high affinity ligands and what way this HLA technology is being connected with cells. The HLA molecules are abundant on the surface and inside the malignant B lymphomas. The monoclonal antibodies recognize malignant cells selectively, but the size limits the blood clearance and tissue penetration. So this is a problem with the monoclonal antibody. There is limitation. Whereas the salts, high affinity ligands really penetrate much smaller than the antibody molecules, penetrate into deeper into the cells and into the inside the cell. So these small novel organic ligands have been selected to bind to sites within the LIM1 monoclonal antibody epitopic region of HLA-DR10. So this is what we collaborated with the Californian group. This is basically a company, Salt Technologies. And as I said earlier, the size of the cells is 50 times smaller than the antibodies. This is really advantage for undertaking these salt-based treatment therapy options for cancers. So this is how it looks like, right? There is a CT ligand, there is a DV ligand, there is a CB ligand in the construct of salts. The expansion for CT ligand is 3,2,3 chloro trifluoro. It's a very long chain of chemical components. And likewise, the CB ligand is 4,4,4 chlorophenzyl piperazino. It goes very big one. Of course, it is there in the references that I am citing here, right? Any of these references, you can able to get these complexes. And DV ligand. So this is how it looks like. All right, CT ligand, CB ligand, and DV ligand. And biotin is a base structure to connect all these three. So when they use the salt molecules, selective high affinity ligands, and when the animals induced with the Raji xenograft, by inducing the xenografts by using the Raji cells and administering the salts later, the size really diminishes and greatly reduces the tumor size, right? So this is how they started their work. And of course, this is our recent work with the group from Madre Kamaraj University and the University of California and Salt Technologies, Livermore. Small molecule antibody, novel molecule, they identified, and we tested with the HLA lymphocytes, SH7139. The results are tested for different HLA alleles, DR10, 15, 11, 1, DR3, 4, 14. So all these, the 10 bearing HLA alleles really bind the drug very nicely. Of course, other alleles also present. So in that concept, such drugs are very important for drug delivery or the cancer treatment, as these monoclonal antibodies are conjugated with the salts, target the cancer cells and kill the cancer cells. So actually, by which this diagram, one can understand the binding of SH7129, another ligand, which gives a brownish color, and the other cells which is not expressing DR10 are not showing brown color, is not binding the salt molecule. Can also coupled with therapeutic, particularly taxorubicin, directly into human lymphoma cells expressing HLA-DR10. So you can use these novel molecules. And the salt nanoparticle constructs enhance the cell killing efficiency of radiotherapy and eradicated more than 80% of the previous lymphocyte staining assay, or we can use SH7139 for treatment regimen, treatment options as a therapeutic for cancer patients. So actually, coming to finally, genes is very important in donors for organ, particularly the kidney, as well as any organ for tumors or in viral infections. The HLA estimation will tell you the level of infection. Disease association, there are beautiful examples are there. Very good example studies are there for rheumatoid arthritis and ankylosing spondylitis. And population admixture and migration patterns are studied by the and in vaccine design and development, also the HLA data of a population is very important. Whether the peptide T cytotoxic response is very important, and that once again depends on the type of HLA or super antigen type. HLA antigens are important in vaccine development. And drug validation, we have seen the abacavir example as one example for drug validation and testing. And you can, to some extent, demonstrate or elucidate the evolutionary principles, as we have seen in the case of HLA and HIV coevolution or molecular evolution. That's beautifully illustrated by a few papers. And finally, we discussed drug shell nano conjugates can be utilized by studying the HLA of populations. With this, I thank the organizers, I thank the participants for your patient listening and opportunity. Thank you very much.
Professor Balakrishna, your talk was really wonderful and gave many insights. But unfortunately, there were some glitches because of internet connectivity. I think you have done lots of work, and there is lots of lots of things which could be useful for participants to know more about all these things, because this human leukocyte antigen, because they play an important role in fine-tuning the adaptive immune system. So I just want to cross-check, is there any question from floor? So far, nobody posted the question. Maybe I will ask you a couple of questions, Professor Balakrishna, because you highlighted, I mean, a number of applications and usefulness in many diseases and diagnoses. So you are also offering HLA tissue typing and donor matching services. So would you please elaborate a little bit about cost involved in HLA tissue typing and getting the services done?
You want to know the cost of HLA typing?
Yes, I mean, how much it cost for HLA tissue typing?
Yeah, got it. Actually, as I said in my presentation, the technology of HLA typing was transferred in the year 1985 from Paris, directly from the Nobel laureate, Professor Jean Dosset, by my professor and guide, Professor Pichapan. And since 1985, we are offering. So actually, the least we are charging very least in this whole country. Say for instance, for HLA A, B, and DR typing for 2 individuals, it roughly comes around 14,000 Indian rupees. 14,000. You accordingly convert to your this thing. So it is roughly 14,000 for matching, single matching, including the cross match test. Yeah.
Okay.
Yeah.
Okay. Thank you very much.
But the same test, if you,
go ahead.
But in private companies and private laboratories, they are charging 4 times this rate. Yeah.
Okay. Another question I have, because the lifestyle is changing, and now in many countries, I think health expenses of the countries are increasing. So based on your experience and based on your knowledge, what do you think? What's going on? What's wrong in the system? How we can mitigate the risk of all these diseases and make human population more healthy?
Very good question. Actually, there are beautiful genes are there in our body. The human system, human evolution has time and again selected good genes, right? The nature has eliminated all those bad genes. But one of the problem with the genes that we acquired and maintained over these years against the pathogens and the nutritional system that we evolved, we are having a good number of genes which will accumulate more energy even if we starve, right? Even at starvation, there are genes to protect us. But because of the selection of such good genes, and we overeat and not doing any physical work, and which accumulate a lot of unwanted material in our body, and also we are intervening with the natural systems, natural biodiversity systems, which unnecessarily attracts the pathogens physiological problems we developed due to food habits, right? right? It is attacking, it is highly deleterious only for those who are having more than 1 or 2 comorbidities. It means that our system is wrong. We need to have a very good change in lifestyle in the whole of country. Definitely we will change the life.
Professor, can you hear me?
Yes, sir. Yes, sir. In between,
Yeah,
there is a problem in the net. Yeah, yeah.
Yeah, because I think the internet connectivity you are facing, most probably because whole India is online now by sitting at home. Maybe that is the reason.
Exactly. Yeah.
Yeah. So it was a great honor to host you as a speaker. So thank you very much for delivering your plenary. It was a wonderful talk, and we got many insights from your presentation. So once again, thank you very much on behalf of organizing committee. So thank you very much, and we will proceed to the next speaker. So maybe you can unshare your slide, Professor Balakrishna.
Yeah. Thank you, sir. Thank you very much.
Yeah. Thank you very much. Take care. Stay safe. Okay, so ladies and gentlemen, we are going to the next presenter's presentation, oral presentation number 28. This presentation is by Kirtani and Malay. She is from Department of Food Science with Nutrition, Faculty of Applied Sciences, UCSI University, Kuala Lumpur, Malaysia. She is going to talk on evaluation of in vitro antioxidant and anti-diabetic effect of water extracts from oil palm fiber. Talk is very interesting. So let's listen to this presenter. So Kirtani Anna Malay, floor is yours. You can start your presentation, and maybe before that one, you can share your slides. Then you can start.
Thank you. Good evening, everyone. My name is Kirtani Anna Malay, and my research topic is on evaluation of in vitro antioxidant and anti-diabetic effect of water extract from the oil palm fiber. So what is diabetes? So basically diabetes is a non-communicable disease, so which the blood glucose level is above the normal. So in the pancreas, pancreas makes a hormone called insulin to help glucose get into our bodies. So when we have the diabetes, your body either does not make enough insulin or can't use its own insulin as well it should. So this causes the buildup in your body. So diabetes mellitus leads to the prolonged hyperglycemia and impaired metabolism. So these are the type of diabetes. So type 1 diabetes, actually a pancreas not able to produce the insulin, whereas the type 2 diabetes is insulin produced, but the beta cell is damaged or insensitive, or no production of insulin. And the gestational diabetes basically occurs in the pregnant woman. So according to the WHO, on the Southeast Asia, we can see that the unhealthy diet causes rise in the diabetes. According to the International Diabetes Federation, in Malaysia, we can see that about 3.6 million total cases of diabetes in adults. In recent time, our health minister says close to 1 in 3 adults diabetic by 2025. In addition, an alarming 7 million adults, both diagnosed and undiagnosed, are estimated to be affected by diabetes in Malaysia by 2025. Not just that, Malaysia has the highest in Asia and one of the highest in the world. So this is the causes of diabetes mellitus: sedentary lifestyle, insulin insensitivity, lack of insulin secretion, genetics, and also reactive oxygen species. So What worries me is the complication of the diabetes mellitus. So in complication, they are divided into two parts. It's acute complication and chronic complications. So here are the type of antidiabetics are currently available in the markets. Mostly they're using metformin, acarbose, and stigmaglutide. So from this chart, we can see that metformin has the highest usage in the diabetic patient. So have you wondered why there's still rise in diabetes? So now I would like to introduce my compound from a nature source as act as a complementary. So the compound is oil palm fiber. In Malaysia, widely available, and oil palm fiber trees, lignocellulose fiber can be extracted from the empty fruit bunch. The empty fruit bunch has a potential yield up to 70% fibers and hence a preferable in terms of cost and availability. According to a research, that's shown that oil palm has a range of biological activities such as anti-hyperglycemic, anti-inflammatory, and antioxidant properties. So the problem statement is diabetes mellitus cases are rising in Malaysia, and due to this, and the anti-diabetic drug are lack the ability of proper management in the disease. So there's a need to find a new lead with the bioactive component which can be developed as a complementary for the treatment for the diabetes. So my objective is to study the in vitro anti-diabetic and antioxidant of the oil palm fiber in water extract. The hypothesis is the anti-diabetic effect of oil palm fiber that maybe have the radical scavenging properties. So looking at the methodology I have done on the diabetic link enzyme inhibitory assay and also the antioxidant activity. Before that, we'll ask to see the pre-screening test. So the oil palm fiber extractor was provided by an alpha active company, and I've used the concentration from 10 to 100 microgram. So this is my experimental design. So I have done on the positive control, negative control, and also a triplicate for all. Carbohydrolysis inhibitory assay. So it's the same thing as the anti-diabetic enzyme assay. So I have done on the alpha glucosidase and also alpha amylase. This is the methodology for alpha amylase, alpha glucosidase. Moving on to the antioxidant, I have done 5 tests, which is on the nitric oxide, hydrogen peroxide, DPPH, metal ion chelating, and lipid peroxidation. This is the this is the method for DPPH, lipid peroxidation, nitric oxide, and hydrogen peroxide. So now the results part. So I have conducted my result and represent in the IC50 form. So as you all know, IC50 is a half maximal inhibitory concentration of 50%. So from this graph, we can see that nitric oxide and hydrogen peroxide have the high IC50, which means they have a low free radical scavenging and anti-glycation activity. And for the lipid peroxidation, chelating capacity, and DPPH can have the low IC50 for my oil palm fiber, so which means it has a high free radical scavenging and anti-glycation activity. For the anti-diabetic activity, we can see that both for my compound OPF have on the alpha amylase and alpha glucosidase have the low IC50 value, which means it have a high free radical scavenging and also anti-glycation activity. So in the discussion part, the OPF possesses the ability to scavenge the free radical generated by the hydrogen peroxide, DPPH, nitric oxide, lipid peroxidation, and as well as the metal chelating activity. So we in scavenging free radical generated. So in OPF, they've shown that it's a better free scavenging effect generated by the DPPH, lipid peroxidation, and metal chelating ion activity compared to the standard drug. So the mechanism is thought to be ability of polyphenol present in OPF to donate the hydrogen to the free radical to remove the odd electron during the oxidations. OPF inhibited activities of carbohydrate hydrolysis known as the antidiabetic, so in alpha amylase and alpha glucosidase also. So the molecular mechanism of alpha amylase and alpha glucosidase by OPF could be through the presence of hydroxyl group attached in the structure of bioactive component in OPF, which have the high affinity to bind with the active site of the enzyme and inhibit the alpha amylase and alpha glucosidase. OPF has a more potent compared to the drug acrobos. So in my research, I use the acrobos as a positive control. And OPF has the ability to delay the absorption of carbohydrate from the small intestine and thus has a lower effect of postprandial blood glucose and also in the insulin levels. So OPF shows the highest activity at the lowest concentration. So in conclusion, that I would like to conclude that oil palm fiber possess free radical scavenging and inhibition of carbohydrate hydrolysis property. The mechanism is thought to be due to a hydrogen donation ability of polyphenol present in the OPF, and also the presence of hydroxyl group attached in the structure of bioactive component in OPF, which have the high affinity to bind the active site of the enzyme and inhibit the alpha amylase and alpha glucosidase. So this is my references. And also I'd like to acknowledge the Alpha Active Company industry for providing the funding for the research and the extract, UCSI University for the facility, and also my supervisor, Associate Professor Dr. Patrick, for guiding through the research development and analysis. Thank you.
Thank you, Kirtani, for your wonderful presentation. So because for, Malaysia, oil palm industry is very, very important because it contributes significantly for the GDP of Malaysia, and Malaysia is the second largest producer of the oil palm. So from that context, what you are doing is very, very important because the oil palm mills, those are in Malaysia. There is a lots of waste and best can be made out of waste. So in that context, I think your work is very, very good. So let me just take any questions from audience. There is one question for you. You are saying diabetic can be genetically. Question is not written very properly. So the parents and diabetic offsprings most inherit it, and is there any precaution offsprings can take to low or stop from inheriting it genetically? So while talking, you mentioned about genetic inheritance. In that context, this this question is there. So would you like to answer this question? Because it's not very clear, but it's about the inheritance of the diabetes. And are you clear about the question?
Not really.
Yeah, so question is not typed properly. Maybe it's
highlight still, Dr. Subhash.
It's on my another computer. I'm just watching. Maybe I'll just see if I can see it.
They are asking, the audience are asking, is there any precaution offsprings can take to low or stop from inheriting it genetically? What are the precautions offsprings can take to stop that genetically inheriting it? That's what they want to know.
Sorry, I have not done in genetic kind of thing. I've not gone to the genetic research kind of thing, because mainly what I understand about it, because our body naturally produces free radicals. So when you consume food that have the high antioxidant, it enables to stop or reduce the from the disease like obesity and so on, like that. Yeah.
Okay, thank you very much, Kirtani. Another question is there from the audience. Audience is asking, is any animal you used in your study for, this antidiabetic study? And if yes, which animal model was used? This one was not highlighted in your study, so now they're asking about it. So maybe you can highlight.
Basically, I've never used, because this is an in vitro, so we don't use on the animal work, but I would like to try on for my future research, I might try on the animal work. But for now, we just use the plant extract totally on the plant extract only. We have not done on the animal one.
Okay, so I have one general question because since you are talking about oil palm, I'm quite excited, Kirtani.
Okay.
So in oil palm, because waste comes in different forms, it comes from the fruit bunches, it comes from the leaves, it comes from the trunk, and other sources, because it's lots of waste in oil palm plantation. So your focus is just on the fibers from fruits, or
Actually, I'm taking this compound is given by the company, so it's basically is an empty food bunch. It's a wastage from the oil palm tree itself. So we're taking the fiber from that and we're using in a water extract.
Okay, so I think there is no any more question, and we're also running out of time. So thank you very much for your wonderful presentation. Hope in the future we will hear some breakthroughs from your laboratory. So thank you very much and keep it up. Thank you.
Thank you.
Okay, so let's go to the next presenter. Our next presenter is Mugilan, Mr. Mugilan. Let me check. He's there, Mugilan. Mugilan, are you there in the room? Yeah, okay. Mr. Mugilan is here. So our next presenter is Mr. Mugilan. He is going to talk about a very interesting topic. He's working at Department of Biotechnology, Faculty of Applied Sciences, AIMS University, Malaysia. And this project is in collaboration with the Ferry Food Industries Sendirian Berat, Malaysia, Sungai Petani branch. So he's going to talk about the conversion of bakery waste material into compost and evaluation of its physiochemical properties and phytotoxicity. So with this brief introduction, I would like to invite Mr. Mugilan. Please, you can start the presentation. Floor is yours. Thank you. You can unmute the mic, Mr. Mugilan. Unmute your mic.
Can you hear me, sir?
Yes. Yeah.
Yes, sir.
Yeah, you can share your slides. Put it on presentation mode. Yeah.
Yes.
Yeah. Floor is yours. Please go ahead. Yeah. Thank you.
Okay, sure, sir. Okay, so good evening to everyone. So my research title is conversion of bakery waste materials into compost and evolution of its physicochemical properties and phytotoxicity. So this research is done under the supervision of Professor Dr. Kadirisan and Professor Dr. K. Marimtu. And for the introduction, studies shows that as much as 1/3 of food waste intentionally grown for human consumption is never consumed and is therefore is just wasted, meaning that we are just throwing the food into the rubbish. So currently worldwidely handling the food waste is very challenging, and most common solution people taking is just landfilling, just throw into rubbish. So I'm from Ferry Food Industry. Actually, this research is funded fully by Ferry Food Industry. Ferry Food Industry is a well-known bakery in northern region of Malaysia for its products such as bakery, products like cakes, bread, pastry, cookies, and so on. So every day we are producing a lot of waste such as like cake waste, bread waste, expired products, eggshells, carton boxes, and so on. So what we realize is that there's a huge amount of waste created every day. So we do a simple studies, data collection, and we found that the cake waste, bread waste, eggshell. Around 1 week, we are disposing about 1 ton of bakery waste simply like that. This is a huge amount. So that's why we decided to cooperate with Ames University to produce composting, to produce compost. So actually composting is the bio-oxidative process involving the mineralization and a partial humidification of the organic matter, leading to a stabilized final product free of phytotoxicity and pathogen with certain humid properties. And come to the aim of the research. So the aim of the research is, first, we want to convert the bakery waste into green and brown materials. Bakery waste into compost using green and brown material. The second is to identify the physical and chemical properties of the bakery waste based compost. The third thing is to study the phytotoxicity study of the compost produced. And the fourth thing is we want to study whether the commercial effective microorganism have any impact on the composting bakery waste. This is the research design flowchart. So first, we will collect the feedstocks. Second, we will prepare the feedstocks like shredding, screening, and so on. And next, we will analyze the feedstock for a few properties like pH, moisture, AC, NPK, TOC, and so on. Then we'll be culturing the effective microorganism, and this feedstock and culture of effective microorganism will be mixed to produce composting. This composting will take around 4 months, under 20 days, approximately under 20 days. And after composting is over, we will do the physical-chemical analysis on the compost, and we will conduct the phytotoxicity study, and at last, we will do the statistical analysis on the experimental data. So this is the bread waste and cake waste produced. And this is the dry leaf collected from the surrounding, the factory surrounding, and also collected from the Ames. This is the eggshells collected from the production. We clean the eggshell, we dry it, and we can use it for the composting. And next is the carton boxes. This is the carton box. We are every day using various kind of carton boxes, and these carton boxes we have to cut into smaller pieces so that the composting can be done very fast. This is the cow dung collected from the local farm, and this cow dung need to be dried for 1 week to let it cool down before we are using it for composting. And next is the EM culture. So this is the EM culture. So result of feedstock analysis. So from the results, we can see that the total organic carbon, this is the important thing, the total organic carbon of the non-creamy bakery waste, creamy bakery waste, and dry leaf paper boxes are high. And the CN ratio, we can see that CN ratio for bakery waste is below 30, and same goes to eggshell and cow dung, and this can be considered as a nitrogen source. And dry leaf and paper boxes can be considered as a carbon source for our composting. So using all the feedstocks, we have created 6 kind of trials where we are using different ratio of the feedstocks. And for 6 trials, every trial we do triplicate, and we got 18 pails. And for this composting, we have used a bin composting technique where all the compost bins are placed in an open place under canopy to avoid rainwater, and covered with netting to avoid pest issues. And temperature is a very important component for the composting, where we can see that the temperature range at the initial first week, second week, and third week is more than 45 degrees Celsius. And after third week, the temperature start to drop into below 40, and also after 10th week, the temperature become below 30 degrees Celsius. So what we can say is that this temperature, our composting, there is 4 important stage in every composting. First, we call it as first mesophilic stage, which is between 25 degrees Celsius and 40 degrees Celsius. Second is a thermophilic stage. Third is cooling stage. Fourth is maturation phase. So our compost have reached all the 4 important stage for composting. And for this level, for this small bin composting, 48 degrees Celsius to 50 degrees Celsius maximum temperature is sufficient enough according to Kadir et al. 2016. So next is the moisture content. What we can observe is the standard moisture content for compost, the finished product of compost is 45 degrees Celsius, 45% and 45 to 65%. And we can notice that it is within the acceptable range, which is within 45 to 65. The pH, what we can observe from the pH at the beginning stage is acidic, and this is due to a lot of production of acidic organic waste due to the decomposition of a lot of food waste and other wastes, and gradually the pH increased to alkaline. At the end of the composting process, the pH is within 6 to 8, and the standard range acceptable for compost is 5.5 to 8.0, and some studies suggested 6.7 and 9.0, and our compost starting from 28th day giving the standard range, which is within 6.0 to 8.0. And next is electrical conductivity of compost trials. So electrical conductivity study is done to study the soluble salts found in the compost extract. So our study showed that our compost extract got electrical conductivity within 1.0 to 2.5 millisiemens per cm. So the acceptable range for a compost is below 4.0 millisiemens per cm. Above this level, it will create phytotoxicity, where the plant might facing difficult to adapt to the high content of salinity. Order analysis. Throughout the process, we have conduct order analysis, and notable order was noticed during the first 3 weeks, where first and second weeks the compost giving out unpleasant smell, and third week it started to give earthy smell. Earthy smell is a sign of maturity of compost, and we can see that this is the appearance and color change throughout the composting process. On the 7th day, we can see the color and appearance of the feedstocks, where we can clearly identify which is paper, which is eggshell, which is bakery waste, and so on. But when time move on, we can see that the structure and the color changes gradually, and at the end of the composting, it is already changed into black in color. So after 13 weeks, the compost can be considered start to reach maturation when there is no increase in temperature when we are turning. There is the temperature reach maximum not exceeding 30 degrees Celsius, pH within 6.8, and we cannot recognize any feedstocks. Earthy smell was given up, and the end product in dark brown in color. So maturation phase take about 4 weeks, where we do not perform any turning, any disturbance not given for the compost. So we leave it just like that until 4 weeks is completed. After 4 weeks completed, we will conduct water holding capacity study, phytotoxicity study, and peak analysis, TOC analysis. So for water holding capacity studies, we will soak the compost for 2 days, and after that we will filter out the not absorbed water, and this is the result that we have obtained. And what we can notice is that compost C1, C2, C3, C4 having high water holding capacity compared to C5 and C6, and the lower water holding capacity for C5 and C6 might be due to the presence of eggshells in C5 and C6, where eggshell contains 97% of mostly is calcium carbonate. So there might be some reaction between calcium carbonate and water, so which resulted in the less amount of water holding capacity for the C5 and C6. Next, we move on to the phytotoxicity study. So we use a common bean, which is a green bean, to study the phytotoxicity level, and this is the result. So our result is just nice, because according to past studies, phytotoxicity 80% and above considered is completely mature phytotoxin free, and below 50 is considered is immature compost. So our compost, all the compost have reached more than 100%. So this is the condition for the 0 day, first day, second day, and third day. And according to Zacconi et al. 1981, so a germination index below 50 is considered immature, greater than 80 is a phytotoxin free, and completely mature. All our compost considers completely mature and phytotoxin free. So next we move on to the TOC and NPK studies. So what we can observe from this study is that C1 and C2 contain higher amount of total organic carbon content, and NPK compost trials showing that C1, C2, C3, C4 having higher amount of NPK level. And this results is used to determine the CN ratio of compost trials. And we found that C1, C2, C5, and C6 having compost having standard standard value which comply to the standard. Okay. According to studies by Neves 2019, high lipid content food produce compost with CN ratio of 10 to 18. So we have C1, C2, C5, and C6 with this range, and C3 and C4 having compost CN ratio with very low amount. So all the data were analyzed using a statistical software, SPSS. And conclusion, so what we can conclude from this study is that composting can be a feasible alternative method for handling bakery waste, and we can break the concept that high lipid food cannot be composted, where bakery waste which is high in oil amount can be composted, and we can get a satisfactory value for temperature, pH, moisture, EC, and TOC, which comply to standards. And addition of creamy bakery waste and also commercial effective microorganism did not impact the compost development. So at last we can consider C1 as C2 as the best recipe for composting bakery waste. So this compost actually can be used as an organic source for soil amendment. And since there is a lack of scientific data on bakery waste compost studies, we believe that our work can provide useful new data to strengthen our local industry for more sustainable waste management. These are the references. Thank you. Yeah.
Thank you very much, Mr. Mugilan. It's I think it's a very, very interesting work because now everybody is talking about the environmental health. Global community is talking about the environmental health. So if the environment is not healthy, human population cannot be healthy. So I think in that context, your work is very, very good. So there is one question from the floor. Do you feel that the bakery compost would be eco-friendly and safe for the plants? This one is one question from the floor. So you want to add anything, Mr. Mugilan?
Actually, this is part of our studies, and we are going to use this compost. Actually, the process is already started, and it can be used for the plant and agricultural purposes, because there is no any toxic materials that we used in this composting, because all the feedstock is from the food waste, another these papers and eggshells. So nothing to be considered as toxic materials used in these studies. So I think it is safe to use for agricultural purposes.
Okay. Any other question? Just let me check. There is one more question. Did researcher face any problem on pest or animal due to food waste compost?
Sorry, sir.
So this second question is about, did you face any problem on pest or animals due to food waste compost? I mean, is there any negative impact of your compost made from the bakery waste on animals or, I mean, flora, fauna, I mean, in general?
I couldn't understand the question very well. Are you asking that using of the cow manure affect the compost? Is it?
Yeah, this researcher is asking about, I mean, did you face any problem? I mean, this compost, which was prepared from this food waste, do have negative impact on the animals or, I mean, flora, you know, flora, fauna? Animals, pests,
I don't think. I don't think so, sir, because the feedstocks is from the environment, and it is the level of toxic is very low. So I don't think so it will affect any of the living organisms.
Mugilan, I have one general question. I think there are no more questions from the audience. I have one general question because what you have done at laboratory scale, this one is a wonderful work. So to take it further and to deal with the massive waste which is coming from the industry, I mean this type of industry, so how we can take it further to solve the waste management problems?
Okay, so actually, this is very. If you want to go for big scale, it's actually is not to say it's difficult, but if we have sufficient machineries to handle those big waste, it will be very simple only to deal with this compost in a large scale, because we already do it perform in a small scale and laboratory scale. When we want to scale up to the big scale, scale up this composting, what we need is just more feedstocks. So more energy will be needed to prepare the feedstocks. So if we have enough machineries or equipments to prepare the feedstocks, and I think it won't be that difficult to deal with waste when we go for large scale.
Okay. Thank you very much, Mr. Mugilan. Your presentation was very, very impressive, and it's useful for the society because you are trying to solve the problem of the society, how to get rid of the waste. So please take it to the next level. Thank you very much.
Oh, sure. Thank you. Thank you. Thank you.
Okay, so ladies and gentlemen, let's move on to the next presenter. Our next presenter is G. Madhumita. She's going to talk on very interesting topic. She's going to talk on bioactive compound loaded nano emulsion from edible fruit against insects. So topic is very interesting. She's working at Department of Chemistry as an assistant professor, senior, and she is affiliated to Vellore Institute of Technology, India. So with this brief introduction, I invite G. Madhumita to start the presentation. Madhumita, floor is yours.
Yes, sir. Yes, sir. Am I audible?
Yeah, yeah.
And my slide is fine, sir?
Yes, it's visible. You may start now. Thank you.
Yes, sir. Good evening, one and all present over here. I would like to introduce myself. I'm Dr. G. Madhumita, working as an assistant professor senior, Department of Chemistry, School of Advanced Sciences, Vellore Institute of Technology, Vellore, Tamil Nadu, India. The topic of my presentation is bioactive compound loaded nano emulsions from the edible fruit against insects. So day-to-day life, we face a lot of problems regarding the insecticides and pesticides. So let me be very brief about what is an insecticide and pesticide. So insecticides and pesticides are the chemicals which is used to kill the kill or harm or repel or mitigate the insects or pest which is having troublesome towards the agriculture or the plant species. So after identification of the insecticides, it is claimed that there is an increase in the total agriculture productivity. So this insecticides works in the different mode of actions. So though insecticide is helpful for the agriculture, it has its own demerits. So what can be done for the demerits? So there is a chance like botanical insecticide, which has been our ancestors was using. So like back to Bavarian, we can bring back those botanical insecticides with the documentation or with the blend of the scientific evidence, we can make it as a successful one. So what might be the advantages of having a botanical insecticide? It can increase the yield of a crop. It can prevent the spoilage of stored foods, prevent the certain diseases. Naturally, since it is a naturally occurring chemicals, which is obtained from the plants or animals, it is quite safe. It is an alternative for synthetic insecticide. It has a very strong selectivity and very low toxicity towards human. So it has the fast action on the targeted insects. So these presence of insecticide agents will be having a different special mode of action. So due to the botanical insecticide, the insects have difficulty in developing the resistance. So this is also a non-phytotoxic, that is just not toxic to the plants. So these are the advantages of having the botanical insecticide. So taking this into consideration, we thought of developing a botanical insecticides. So commercially, when we gone through the commercial shops and searched what are the different insecticides present, we have come across one insecticide called as Alastan silvoxin, which has been used as an antimicrobial property in the rice fields. And I have shown an image here, which is used as a antimicrobial property, and it creates an incapability for reproducing the microbes. So this commercial agents stops the reproduction of microbes. It changes the DNA of the microbes and unable to replicate, and it gives a higher yield, and it is a disinfectant and non-toxic. Though it has enormous of advantages, in a long stand, it may be toxic to the humankind, and it may be toxic to the non-target organisms. So what we thought is like this silver compounds can be structured, or it can be blend or coupled with the naturally existing plant compounds, and we can make it as a combination of bio components with an inorganic compound. So what we have done is like, we have taken a plant species. So we are working in this plant species that is called as Carica edulis. This is our research work, and our laboratory is concentrating on this plant. So what we have done is we have taken the fresh fruits of Carica edulis from the local market of Vellore, and we have got authenticated from the Plant Anatomy Research Center, which is situated in Chennai, and I have given the number. So the fruits were freshly collected, washed with the distilled water to remove the dust or any kind of impurities or microbial attack. So the washed fruits are then kept in the air dry. We have not kept under the direct sunlight, so some bioactive components might get volatile. So we have done it under the air shade, and we have sprinkled toluene to prevent the fungus formation. The dried powder are then taken into the mechanical grinder, and the powder is stored into the room temperature for further use. So here I have given the pictorial representation of the whole tree, flowers, fruits, and the leaves in detail. And the fruit has been used for our research, and the certificate shows that we have got authenticated from the Anatomy Research Center. So how are we? So this is regarding to the plant that we have collected. So now we were going to collect the insect from the paddy field. So how we have collected and what we have done is like, the adult mouth of the insects, we have taken 3 species. So one is Mendelus, second one is Maritia, and third one is Interhias, were collected from the Brahmapuram paddy field, which is near Vellore, and we have given the latitude and longitude. So during the daytime, in a plastic covers, they have kept into the glass beakers and covered with the plastic covers with a small holes for the breathing and to feed the leaves. So here we have given the rice leaves as a feed. And what we have done is we have made it to lay the eggs on the beakers and on the leaves. After the insect has laid it, we have untouched. We have left it as such for 3 to 4 days, so that the eggs are converted into a larva, and this larva is fed with the rice leaves. So once the larva is grown, they have then placed in the plastic box, and the small holes are kept on the top for the air supply. The boxes are under the mild sunlight to avoid the death of the larva. There should not be a direct sunlight. So in the summer season, what we have done is like, we have covered the boxes with the white cloth, so that there will not be temperature increase in the boxes, and the larva is taken in for the larvicidal activity, and the rest is made it to grow into the other stage for the insecticidal activity. So this is what we have done with the collection and the maintenance of the insects. So here we have shown the larvae, different larvae that I have taken, and the corresponding adult stage. So here we have done the activity, and this activity is done by using the beakers. And what we do is like, we will supply the extracts with the bioactive components into this larva, so that what happens, this larva tries to eat the food, and if the emulsion is contagious, or if the emulsion can kill it, it will kill the larvas. And if it is not, if the larvas will be as alive or the insects will be as alive. So based on this, we find out the mortality. So the mortality is the death rate of the larvas or the death rate of the insects. Here, percentage mortality of the tested insects minus percentage mortality in the control divided by 100 into 100. So that we will calculate the percentage. So this is what the activity we do it. Before going into in-depth, so how we take the extract and what we do, it's like the fresh fruits of Carica edilis are taken, made into a small pieces and dried at the room temperature. The dried powder is taken and it is extracted with the microwave assisted extraction, and we get the petroleum ether extract. From the after we remove the petroleum ether extract, the remaining dried powder is again incorporated to the microwave extraction. We get with the help of ethanol. So we get the ethanolic extract. Why we are removing two different extracts is the petroleum ether extracts will be having the hydrocarbons or long chain of fatty acids or long chain of hydrocarbons, which is not essential for our bioactive components. So we remove the petroleum ether extract, and the rest, which is having the bioactive components, we take it and we extract it with an ethanol. So after doing this, what we do is like we have extracted, after extraction, we have gone for the isolation part. Here we have taken the crude ethanol extract and we have packed it with the column with the petroleum ether and ethyl acetate as a solvent ratio, and we have eluted the column. After we eluted the column, we have got the different fractions of compounds. After we get the different fractions of compound, we have structurally isolated and identified one single carbon compound called as caryophyllene. So this caryophyllene is mixed with the nanomaterials, and we have converted into an emulsion, and the same emulsion is tested for the insecticides as well as larvicides. So this is the representation of the isolation of the bioactive component from the crude ethanol extract. So based on this paper, these two papers gave us an idea of incorporating the bioactive components with the nanoparticles. So what we have done is like before going into the nano emulsion, we have individually manufactured or synthesized the nanoparticles. Here we have taken the silver oxide nanoparticles. That is about 10 milligram of the extract is dissolved in 20 ml of water mixed with 80 ml of 1 millimolar of silver nitrate to synthesize Ag2O nanoparticles. The formed nanoparticles are checked with the UV visible spectroscopy for the complete formation of nanoparticles. After the nanoparticles has been formed, the mixture was centrifuged to remove centrifuged, and we have obtained the nanoparticles as such after the centrifugation, and it is calcinated at 300 degrees Celsius for 3 hours. So this is the pictorial representation of how we have made the nanoparticles and how we have calcinated the nanoparticles. Coming to the nano emulsion, so first we have isolated the compound from the bioactive extract. Second, we have manufactured the nanoparticles, or we have synthesized the nanoparticles. Now what we have to do is like we have to incorporate the nanoparticles with the bioactive components and form the nano emulsions. So what we have done is like we have taken the different concentration of nanoparticles of silver oxide, and we have mixed it with water, and we have got the resultant solution. This resultant solution is to convert into a nano emulsion. We have to add the surfactants plus co-surfactant. Here we have taken the 2020 or 2080. So here, based on the optimization, we have taken the different surfactants. Once the surfactant is added, we have get the resultant solution. To the resultant solution, we have added the caryophyllene, which we have isolated and identified. We have got, after we are adding the caryophyllene, we have sonicated so that the nano emulsion is formed. This is a zeta potential of nanoparticles concentration on the surface. So this zeta potential is to find out the stability of nanoparticles. We found out the nanoparticles in this are stable. So these are the different concentrations of the stabilized nanoparticles. So we have the different concentration which varies from 10 to 60 milligram. All these nanoparticles are stable after adding with the bioactive component. So this is the silver 2 nanoparticles with caryophyllene. From this graph, we can easily find out it is highly stable. And this graph shows the FTIR. and SEM analysis. This SEM analysis shows the surface morphology, and you can find out the small dots, that is the caryophyllin is.
Dr. Madhumita?
Yes, sir.
You have 2 minutes to wrap up.
Yes, sir. Yes, sir. Yes, sir. So this is the same FTIR and same image. This is the same image. You can easily find out at the background. We have the caryophyllin, which is acting as a back, and EDAX. The EDAX is a composition identification. Coming to the insecticidal activity, so we have the silver oxide nano emulsions, which is done with a different kind of insecticides. From the LC50, we have the different concentration, which varies from 50 to 500 oil concentration. From the LC50, we can find out all the LC50s are nominal, and you can see that 214.99 that is the least among all the 3 species. So this is the larvicidal activity. Here also we have got the LC50 of 200 milligram UL. So coming to the conclusion, we have found out the microwave assisted synthesis of silver oxide nanoparticles by and it was used for a nano emulsion, which was found out by FTIR, zeta potential, SEM and TEM analysis. The insecticidal activity was carried out, and we found that most of the 3 species are sensitive to silver to oxide nano emulsion, and it has shown a very high mortality. Okay, coming to the acknowledgement, I would like to thank the VIT management for the constant support and encouragement. I also extend to our thanks to DST for their research fund. I would like to express my deepest thanks to my research scholars for their continuous support. And last but not the least, I would like to thank the organizers for providing me this platform for giving my to share my research in this esteemed conference. Thank you.
Thank you very much, Dr. Madhumita, for nice presentation. It was very informative, and I think even though it's at lab scale, but it will be useful for agriculture industry. So any question? There is one question for you.
Yes, sir.
That audience question is why nano emulsion for the delivery, and which particular component is active as you studied with extract?
So we have isolated five bioactive components. Out of this 5 bioactive components, we have taken caryophyllin as one of the bioactive components. So we have 4 other components, and we have studied, and that result has not been incorporated. So we have 5 compounds totally, which was isolated, identified. Among that, I have presented here only one component which has been converted into a nano emulsions. Only a part of work I have presented here.
Okay, Dr. Madhumita, there are many compliments from the audience. Nice presentation, good presentation. Just want to pass the message to you. I have one question because while talking, when you was talking about the silver, and you mentioned that it can change the DNA of the microbes, isn't it?
Yes, sir.
So my question is, because when we are using these nanoparticles, I mean, with silver or this type of the products in the agriculture or open field in environment, so do you think this silver can can cause the mutations in microorganisms, and those microorganisms are not harmful? They can become more virulent, and maybe we'll have new pathogens, you know?
Yes,
what
so
do you think?
2 things I want. Yes, sir. 2 things I want to say. In the commercial product, they take the bulk silver nitrate as the component for the pesticides or insecticides, whereas what taken is in the nano form. Second one is we have incorporated the nano with the bioactive species or the bioactive compound. So the toxicity might not be there.
Okay. So one more question I have, because now many people are working on nanoparticles, nano formulations, and this field is growing very, very rapidly, and there are a number of applications in agriculture, in healthcare sector, as well as in industry. So one general question I have is, since you are working on nanoparticles, what's about the environmental pollution by these nanoparticles? I mean, any thought about environmental pollution? Because I mean, in the future, I mean, we don't know at the moment. So that's
always, as we say, too much of anything is not good for health. So we have to find some way so that reduction of this nano exploration also. That should be like, since most of the researchers now focuses on the nano. So that should be something which makes the nano less toxic.
Okay, okay. Thank you very much, Dr. Madhumita. Your
Yes,
presentation was very, informative and wonderful presentation. So all the best for your research work. So maybe you can unshare the screen.
Yes, sir.
Ladies and gentlemen, we are going to listen to our next presenter. Our next presenter is Mr. Irshad. Mr. Irshad is working at the Department of Biotechnology, School of Basic and Applied Sciences, Dayananda Sagar University, Bangalore, which is located in Karnataka state of India. Mr. Irshad is going to talk on co-occurrence of mutations in quinolone resistance determining region observed in global E. coli genome alarm for fluoroquinolone therapy in urinary tract infection. So this one is very interesting work. So Mr. Irshad, you can share your PowerPoint presentation and you can start the presentation. So floor is yours, Mr. Irshad.
Sir, I'm not able to share the presentation, sir.
Oh, you have any technical difficulty?
No, sir. The option, it's not showing the option here. Share option is share screen, but then it's not.
Just let me check it.
Jansi, can you?
Your PIR Irshad is your ID,
yes,
right?
Sir. Yes, sir.
Can you try now?
Sir, I'm trying continuously, sir.
Now it should work.
Sir, share screen option is there. When I am clicking on that, three options are coming. One is screen, iPhone, iPad, and Zoom. Which one I shall select, sir?
You are using which device you are using?
Sir, a laptop, Dell.
You have to select screen.
Yeah, at the bottom there is a share screen, green icon you can see.
Yes,
yeah,
sir. When I'm clicking on that, three icons are coming. One is screen, another one is iPhone, iPad, another one is Zoom.
Yeah, your file. You can go to your file. If it's not open, you can go to that folder and open the file.
Just select the screen.
Portion screen, content from Zoom camera.
You can open the file first.
Shall I minimize this?
Yeah, so from here you can select your file. In which folder it is located?
Yes, sir.
Yeah.
Is it coming now, sir?
We can see, but can you go in presentation mode? Some text is missing, I think. Maybe you can see only half slide we can see. Mr. Harshad, we can see the slide, but only the half part of the slide we can see. So I think some setting of your device, I guess.
So is it visible now, sir?
Only the bottom part of the slide is visible. We can see the top, the bottom part is missing. Now, great. So Mr. Irshad, the now slide is visible. Floor is yours. You can start. Thank you.
Thank you, sir. Respected judges, other respected teachers, and my dear friends, Myself, P. Rashad from Dayanasagar University, Bangalore, present to discuss my findings on a very important emerging problem into this global scenario, and that is multi-drug resistance. The study revolves around co-occurrence of mutations in quinone-resistant determining region observed in global E. coli genome, which is an alarm for fluoroquinolone therapy in urinary tract infection. So my presentation will be revolving around urinary tract infection, E. coli isolates, and mutations observed towards quinone resistance. Presentation will involve introduction, followed by the gaps, objectives, findings, discussion, and significance. Antibiotic resistance is known that it is the biggest threat to mankind because several fields of modern medicine depend on the availability of effective antibiotic drugs, starting from chemotherapy for cancer treatment, organ transplantation, intensive care for preterm newborn, and several other episodes where effective antibiotics are required. Infections caused by multi-drug resistance bacteria are among the main factors influencing morbidity and mortality in patients undergoing these procedures. In this scenario, bacterial antibiotic resistance becomes a global challenge threatening human and animal health, food, and environment safety. So the study is devoted to a specific cluster of organisms, that is E. coli, and a specific disease, that is urinary tract infection. Why quinolone? Because it is the most preferred antibiotic across the globe for treating urinary tract infection. So what does this quinolone target in bacteria? This actually targets two important enzymes involved in bacterial replication, that is DNA gyrase and topoisomerase 4. Interesting thing is that we are looking for mutations, and with mutations that is directed towards two important enzymes which are coding these DNA gyrase and topoisomerase 4. I want to bring your attention. They are gyrase A, B, PAR C, E. Why I am emphasizing here? Because later in my results, I will be showing you how mutations are detected in this particular segments of the genes which are responsible for being the target of the quinolones. Now you must be pondering what attributes to quinolone resistance. There are multiple modes by which quinolone resistance is imparted to the bacteria. Number one, chromosome mutations, where within the QRDR of the genes encoding these A-B subunits of the DNA gyrase, which I showed in the previous slide. Next, it can reduce the membrane permeability by altering the outer membrane organization or by reducing the porin expression. Number three, the major mode which has threatened the global health, and that is plasmids. The plasmids can be containing certain key genes like QNR, AAC, IBCR, and these enzymes are responsible for devoting quinolone resistance. Another mode is multi-drug efflux pump, which can contribute to quinolone resistance. So with this area of broad mechanisms which can be contributing to quinolone resistance, we wanted to see what is the current status of research in urinary tract infection. UTI is majorly treated with cephalosporins, fluoroquinolones, and trimethoprim. We did a literature survey to see the present investigations reported in PubMed. We found that most of the studies were devoted to PCR assays targeting 5 selected genes. Literature review revealed the following gaps. Chromosome mutations are predominant, but you can detect those mutations only by employing sequencing protocol. Most of the studies target PCR-based detection of plasmid-mediated quinolone resistance, so the investigation may miss out on the chromosome mutations. Limited studies on quantitative real-time PCR because that is the only mode which is simple to assess the expression of genes in the presence of antibiotics exposure. Most of the studies are province-specific, hospital-specific, or no global view existing on date. So what was the research question? We wanted to know if we can capture the complete genome to view both chromosome and plasmid and simultaneously get an insight of both, that is PMQR genes and chromosomal mutations in QRDR. I wish to tell again, quinolone analysis was the target of my team because quinolone is the main selective pressure exerted on urinary tract infection. With that, the aim was to whole genome sequence analysis of clinical E. coli isolates. The specialty of our study was that we didn't take region-specific or hospital-specific. We targeted the entire database available with E. coli genome. I wish to bring your attention that we took and screened all the complete genome. We didn't go ahead with contigs, draft genome, or scaffold. Whole set of complete genome was screened and find out what was the UTI cluster, and in that UTI cluster, we aimed to determine quinolone resistance in plasmids, chromosomes, and further for the resistant isolates, we wanted to check was the other multi-drug resistant potential hidden in those bacteria. With that, the methodology we used was highly recommended protocol from reputed journals, where we took 1,079 currently available whole genomes. We screened all 1,079 complete genome, and we arrived to 30 UTI E. coli cluster. And we found geographical location, date of submission, and sequencing platform, and we collected only those isolates which were submitted using PacBio platform, so that the advanced technology is not compromised for the quality of data. We went ahead with this pipeline where we make sure that we annotate the complete genome. We look for the mutations in QRDR, multi-locus sequencing typing, followed by characterization of MDR plasmids for conjugational potential. So the results of our pipeline observations and findings, as I show you the first table, which talks about the 30 UTI strands, showing that all were from PacBio sequencing and different parts of the country. We found 30 UTI E. coli from humans and 3 from canine. PacBio sequencing was the predominant sequencing mode. 21 E. coli harbored more than 2 plasmids. This is the snapshot of the genes we got to know from the annotation. On annotation, we found that more than 4,000 genes present in the clinical isolates. When we started digging out and comparative analysis, we arrived to the mutations in the QRDR, and it was interesting that majority of the isolates were showing co-occurrence of same mutations in gyrase A, PAR C, and PAR E. S83L, S80I, and 1529L were common patterns of mutations observed in isolates. These are the other isolates where the QRDR showed a similar pattern of mutations. Finally, we wanted to see and determine the other antibiotic-resistant genes present in the isolates. Oh my God, we were astonished to see for beta-lactam, there were array of resistant genes present. For aminoglycosides, again, a large weapons available in bacteria. Microlides, phenol, and others were seen to be extra powerful than we can ever anticipate. Next, we wanted to segregate, and contig separation we did and found that isolates were having big weapon of antibiotic resistance. I want to show you one snapshot where you can see this particular strain from Hong Kong has 3 plasmids, and each plasmid has 12, 9, and 2 antibiotic resistant genes, contributing to resistance to beta-lactam, aminoglycoside, and sulfonamide. The isolate was a matter of concern. Then multi-locus sequencing was done. We found there were 14 different types. But I want to bring your attention towards this yellow highlighted bar, which shows that ST131 was predominant E. coli colonial strain causing urinary tract infection across the globe. MLST shows a close relationship of the isolates. When we went ahead with the comparative genomic analysis with MAU, it was interesting to reveal another part of the understanding, that is horizontal transfer events revealed within the chromosome because of transposon. What was this transposon composed of? The transposon had 2 aminoglycoside modifying enzymes flanked by 2 insertion sequences that had increased the level of fat hidden in the chromosome and as well as the plasmid, making the isolate so powerful that none of arsenal of our antibiotics can effectively counterattack them. So comparative genomic events and all the complete assessment which we did in a systematic way, for the complete genome cluster of UTI, we found that they were all multi-drug resistant. ST131 was the predominant colonial strain, and there was clear-cut horizontal transfer events contributing to the growing rise in the antibiotic resistance gene pool and repertoire of these emerging pathogens. So the MDR power which was detected in the UTI cluster shows a set of genes responsible for destroying the beta-lactams, which we commonly use for urinary tract infection treatment. Second pool of genes, which the isolates were responsible for antibiotic modification. And finally, as I told you, that quinolone resistance was the predominant mode of mutations in chromosomes. So apart from chromosome mutations seen in the quinolone, these particular bacteria were also responsible for weapons to make other antibiotics also ineffective. So I want to show you a snapshot of the superbug combat 11I9, which had 3 plasmids beside the chromosome, and it had 23 resistant genes. Aminoglycoside treatment you want to give for this bacteria, it will not work. You want to treat it with the beta-lactam, it will not work. You want to treat it with quinolone, we will fail. If we want to treat it with sulfonamide, we cannot. When I started the journey, I aimed for fluoroquinolone, but when I finished the journey of my research study, I arrived at the conclusion that not only it is alarmic for fluoroquinolone therapy, but it is equally concerned for beta-lactam and aminoglycoside treatment, which are most preferred when it comes to urinary tract infection. So this shows superbug is a matter of concern. Think before prescribing antibiotics. If there is no cure today, there will be no cure tomorrow. The significance of the study is that data generation is easy in these days, but data analysis and make useful interpretation is need of an hour, which our team has devoted to find out significant information. We emphasize that there should be some molecular PCR testing which can aim for these mutations which can guide the correct use of antibiotics in the urinary tract infection. Conjugated plasmids are trying to be major mode of dissemination of antibiotic resistance. Existing database on superbug genome is a gold mine and can allow us the valuable unprecedented insights. One limitation of this study is we didn't have a PacBio complete genome of UTI isolates of India, which we think we need to contribute for our better understanding. And we think molecular diagnostic protocol need in the clinics before prescribing antibiotics, generation of whole genome sequence of Indian isolates. At the end, I would like to sincerely acknowledge Honorable Dean Sir of Dayana University, Bangalore, and all the teachers of Dayana University, and thanks to MST University Malaysia for providing me the great opportunity and providing me the great platform. I would like to conclude my presentation with these golden words. If we will not pay attention today, tomorrow it will be difficult for us to cure and treat simple bacterial infections. Thank you.
Thank you very much, Mr. Irshad, for a very energetic presentation. Thank you very much. It was very impressive, and you are doing a great work, Mr. Irshad. So let me check any questions from the audience. I don't see any question yeah, there is one question.
Yes, sir.
Mr. Irshad, the question is, why did not you study with nitrofuranation, as it is also a wide choice in UTI, urinary tract infection?
Sir, actually, we were not trying to treat the urinary tract infection. Actually, our aim was that we want to see that what is the trend of the resistance of the fluoroquinolone. When we started the journey, our aim was to check the causes of the resistance of the fluoroquinolone. But at the end of our study, we saw that there were many mechanisms harboring the bacteria, which was contributing for the resistance to other antibiotics like beta-lactam, aminoglycoside, and all. We were not actually treating the urinary tract infection.
Okay, thank you. There are no any other questions from floor, but I have a couple of questions, Mr. Irshad.
All right.
Because while talking, you said, you have screened, there were more than 4,000 clinical isolates of E. coli.
Yes, sir.
And some of them were having 2 plasmids or 3 plasmids.
Yes, sir.
So I'm just wondering, do you have any information these plasmids were stringent plasmids or relaxed plasmids?
Yes, sir. So the clinical isolates, the maximum number of plasmids which I have seen in these 30 UTI isolates, that by 10, and most of these UTI isolates, they were containing more than 2 plasmids, which were responsible for this antibiotic resistance. But so your question is that was these plasmids stringent or relaxed? Sorry, sir, I don't have much information about this, sir.
Okay. Another question is because you were saying there were antibiotic resistant genes in the this clinical isolates which you have screened.
Yes, sir.
So I'm just wondering, because in each and every country, people are working on genetic modification of microbes, and E. coli is used as a competent cells for the genetic manipulation. So do you have any information about from where these E. colis are coming in food supply chain or in contact with the humans?
Yes, sir. Actually, there are like different modes by which these pathogens, like E. coli, they are coming in contact with the humans. First one is that they are coming from the farmhouses. We consume the meat and we consume this vegetables and all. Those vegetables and all, those has been processed during the production. Antibiotics has been used. So pool of antibiotics is being used during the production of the vegetables or during this animal farmers. So by this, these pathogens, E. coli, which are resistant to the antibiotics, they come in contact with the humans and other animals.
Thank you very much. So
Welcome,
Mr.
sir.
Irshad, thank you very much for your presentation. Your presentation was really impressive, and your voice was very, energetic. Thank you very much for wonderful presentation.
Thank you so much, sir. Thank you, sir.
So you can unshare
Yes,
your screen, so we can move to next presenter. Okay, ladies and gentlemen, we have another 3 presenters. Then we are going to end this session. So the next presenter, for the introduction of next presenter, I would like to invite my co-chair, Associate Professor Dr. Siva Kumar. Dr. Siva Kumar, are you there? Dr. Siva Kumar, are you there? I think Dr. Siva Kumar may be some internet connectivity. So our next presenter is, I will introduce. Our next presenter is Mr. Shubham Ghosh. He's going to talk on design, molecular docking, and drug likeness studies of trioxin derivatives as novel anti-malarial agents. Mr. Shubham Ghosh is working at Department of Pharmaceutical Sciences, the Berg University of Assam, state of India, and he's presenting on this interesting topic. Mr. Shubham, are you there? Mr.
Yes,
Shubham?
sir, I'm there, sir.
So you can,
yes, sir. Hello, sir.
Yeah, you can share your presentation slides, and floor is yours. You may start presentation. Thank you.
Okay, sir. So is my slides visible?
Yes, yes.
Hello?
Yes, we can see your
Okay.
slides, please.
Okay.
Yeah, go ahead.
Thank you, sir. A very good evening to all the delegates, the respected judges, and all my dear friends who are present over this international conference on bioinformatics, biotechnology, and biomedicine. Myself, Shubham Ghosh from Department of Pharmaceutical Sciences, Dibrugarh University, Dibrugarh, Assam, India, and I'm going to give a presentation on design, molecular docking, and drug likeness studies of trioxin derivatives as novel antimalarial agents. So first, coming to the background part. So malaria, the term malaria is a very much familiar term with all of us, as it has been affecting millions of life worldwide across the globe. The deadliest parasite, which is mainly responsible for causing the disease, is Plasmodium falciparum. So as per the recommendation of WHO, the current available treatment for malaria are the ACTs, that is the artemisinin combined therapies. So the problem arises due to the widespread emergence of resistance of this particular deadliest parasite, P. falciparum. The clinical utility of existing drug therapies along with the artemisinin combined therapies has been increasingly limited. So in order to tackle this issue, we need to develop some novel antimalarial drugs which could be effective against this resistant P. falciparum malaria and hence become an essential treatment for the disease. So this is the structure of artemisinin, as we can see in the screen. And along with that, the trioxane, which is our main moiety of our study, is also present in this particular artemisinin structure. Now is the objective. So the main aim of our study was to develop a series of 1 to 4 trioxane derivatives which are novel, and which would also be effective against the deadliest parasite, P. falciparum. The trioxane derivatives were designed using different substituted acerophenols and substituted aromatic aldehyde. And moreover, 1 to 4 trioxane from the structure of artemisinin, which I have shown in the previous slide, is the main pharmacophore, or you can say the key pharmacophore or the scaffold of our interest. Now the materials and method. 15 number of trioxane derivatives were actually designed by the molecular modification of trioxane scaffold as possible antimalarial agent. So based on the computer drug design and Biovia Discovery Studio software, the molecular modeling studies of the derivatives were performed. The protein ligand docking was carried out against the target protein P. falciparum falciparum 2 or P. falciparum cysteine falciparum 2 with a PDB ID of 3 BPF, and it was used for simulation-based docking protocol. The flexible docking method was generally used for our docking study. Along with that, the assessment of drug likeness, ADMET properties, and toxicity were also investigated. And finally, redocking procedure was also performed so that the docking study can be a docking study and the accuracy of the docking method can be confirmed. So this is the 3D structure of our target protein, that is 3 BPF or cysteine falciparum 2. Which can be easily downloaded from the rcsborg.website by entering the PDB ID. This 3 BPF protein is already in this structure. This 3 BPF protein is binded with its co-crystal ligand, which is E64. Now the results and discussion part. So our study was mainly based on in silico screening or computer-based screening with special emphasis on the molecular docking studies. So what we get from our study is that after the redocking procedure, we have validated our docking study, and from there we concluded that the RMSD value of, that is the root mean square deviation value of less than 2 Armstrong was noted, which is in the acceptable limit. Moreover, docking results showed that among the series of 15 compounds, 3 trioxane derivatives were found to possess promising binding affinity, or they have shown the good docking with good LibDoc scores in the range between 117.16 to 116.7. So this is the LibDoc. This is the table for the LibDoc scores, where we can see that our the 3 compounds, which is the compound number 3, compound number 5, and compound number 8, shows the highest LibDoc score with number of hydrophobic and hydrogen bond. The molecular properties were also calculated for all the compounds, in which we found all the compounds possess a molecular weight of less than 500 with log P value of less than 5 and a drug likeness score of 0.55 for all the compounds. So this is the table for molecular properties, where we can see that the log P value for all the compounds were found to be less than 5, along with the molecular weight of all the compounds were also determined to be less than 500, with number of aromatic ring, number of hydrogen bond acceptor, number of hydrogen bond donor, and molar polar surface area, and a drug likeness score of 0.55. So the ADMET, that is the absorption, distribution, metabolism, excretion, toxicity properties for the derivatives were also determined and found to be satisfactory. So this is the table for ADMET data, where we can see that the solubility level of my 3 main compounds, that is compound number 3, compound number 5, and compound number 8. Sorry.
Mr. Shubham, we cannot see your slides.
Sir, is it visible now?
Slides are not visible. I think you have to share your PowerPoint presentation. Maybe by mistake.
I'm sharing, but so.
Now you try, Shubham.
Yes, sir, I'm trying, sir.
Now it should work. Now it should work. Shubham? I think Shubham have connectivity problem, so maybe we'll get back to him once he reconnect. Okay, ladies and gentlemen, sorry for the interruption. I think he's having connectivity problem. So let's move on to the next presenter. Our next presenter, we have another 2 presentations. So I would like to invite my co-chair, Dr. Sivakumar. Dr. Sivakumar, are you there? Dr. Sivakumar, you can unmute yourself and
Yes,
over to you. Over to you. Yeah, thank you.
So shall we move
Okay,
on
sir.
to
Yeah.
presentation, sir?
We can start oral presentation number 33 and 32. I think once he gets reconnected, some part is pending. That one will complete later on. So I think you can introduce next speaker and we can start. Thank you.
Right, sir. So we will move on to the next presentation. So the next presenter is Zubairu. So just give me a minute. Yeah, he's from Department of Soil Science, Faculty of Agriculture, and University of Maiduguri from Nigeria. And he will be presenting about distribution of available phosphorus in a sandy loam soil in Nigerian Sudan savanna. So you can start your presentation, Mr. Jubairu. So you can start your screen sharing and you can start your presentation now. Can you hear us? Are you there?
Hello.
Yeah, can you hear us?
Yes, sir. I can hear you, sir.
Okay,
Let
right.
me
You
share
can
my
start
screen.
your presentation. Yeah.
Yes, sir. Let me share my screen. Let me share computer. Okay, sir. Yes, sir. I am setting the PowerPoint. This will bear with me, sir.
Sure.
Hello. I
Yeah,
hope I'm
yeah.
ready to go,
Sure,
sir.
take your time.
Sir. Hello. Yes, sir.
Yeah, we can hear you. Yeah.
Yes, sir. Should I go on?
Yeah, sure, please.
Hello. Okay, okay, sir. My name is Abdurrahman. I'm presenting from University of Maiduguri. The title of the research is Distribution of Available Phosphorus in the Sandy Loam Soil of Nigeria and Sudan Savannah. The study was actually taken out within the research firm of University of Maiduguri in 2015. Let me go start with the introduction. The study was conducted with the aim of what? Determining the distribution available phosphorus in the sandy loam of University of Maiduguri, teaching a research firm. It is well known that phosphorus is one of the most important nutrients that are needed by plants in order to get optimum yield. While phosphorus is critical in nature, it is the most needed nutrient element by plants after nitrogen. In most of our tropical soils, the supply of phosphorus is low. Which can be due to the excess of calcium or totally leached soils. This can show
Jubairu, can you hear us? Jubairu?
I think again he's having some connection problems, sir. So they have mentioned in the chat that they're having some connection problems in Nigeria right now. So shall
So
we
the,
wait for him or?
So the previous speaker is on. So maybe he can start 32, number 32, Mr. Shubham.
Okay.
Sir, can you hear us? Hear me, sir? Hello?
Yes, yes, yes.
I hear you. Yeah.
Mr. Shubham, you can start from the slide.
Yeah.
Yeah,
Yes,
the
sir.
last
Yes, sir.
slide. Yeah, previous slide
Yes, sir.
where you left.
So, yes, sir. Due to technical error, I was left behind. So the toxicity, along with the ADMET properties, the toxicity profiling was also determined and found to be within the acceptable limit. So this is the toxicity table, where we can see the my three main compounds. That is the compound number 3, which was showed carcinogen against the topcat mouse male NTP, but in case of topcat rat female NTP, it was non-carcinogen. But in case of compound number 5, it showed non-carcinogen both in case of topcat mouse male and topcat rat female. And along with that, my compound number 8 also showed carcinogen in case of topcat mouse male NTP, but it was a non-carcinogen in case of topcat rat female NTP. So this is the 2D and 3D docking images. I have shown the docking images of my 3 best compound which showed good docking. So this is the 2D structure of compound 3, and this is the 3D structure of my compound 3, which shows different bonds. So this green color indicates the conventional hydrogen bond. This white color indicates the carbon hydrogen bond, and this yellow color color indicates the pi sulfur bond. Sulfur.
I think again he got disconnected.
Yeah. Sir, we are running out of time. So what we can do, this number 32, maybe tomorrow we can give chance for him to present. So we will move on. We'll try one more time to this presenter from the Nigeria. So
Abdul Rahman. Okay.
Yeah.
So is he here? I mean, can you hear us, Mr. Abdul Rahman?
I think he's not in virtual room, sir.
Not in Zoom.
So what we can do, because we are running out of time, so maybe these 2 presentations, we can push it to tomorrow's one of the session, because we cannot keep on waiting. It's already late. So maybe, yes, sir, you can go for the last presenter. You can introduce, and then we'll conclude the session.
Sure, sir.
Yeah.
Okay, so we will go on to the next presentation. This is the last presentation in this session. The presenter is Ayesha Siddiqui, Miss Ayesha Siddiqui, and she is from the Department of Biotechnology, Jinnah University for Women, and Karachi, Pakistan. And her title of presentation is Photosynthesis of Silver Nanoparticles Using Plant Extracts of Medicinal Importance. So I invite Miss Ayesha Siddiqui to give away to carry on with her presentation. So you can start your presentation, ma'am.
Thank you, sir. Can you hear me?
Yeah, we can hear you.
Okay. Good evening, all. First of all, I would like to thank you for the organizing committee of the IECBBB 2020 to provide me the platform to present my study. The title of my study is the phytosynthesis of silver nanoparticles using plant extracts of medicinal importance. So as we all know that the plants are the old friends of Homo sapiens, as they provide us the food, shelter, as well as there are so many components of the plants, and plants contain various compounds which provide the defense against various diseases. And the friendship between the Homo sapiens and plants is still sustained. And the reason behind that, the identification, synthesis, and development of antibiotics against various pathogenic organisms will provide the opportunity to the researchers to find the novel compounds from the plants and test it against the pathogenic isolates. As we know that there are a lot of microorganisms which are involved in the life-threatening disease. For example, the Klebsiella are the strains which are involved in the pneumonia infection. Shigellosis is caused by the Shigella, and it sometimes is caused the bloody diarrhea. Similarly, the family of the Enterococcus are involved in various diseases, including UTI, cellulitis, and other wound infections. In the same way, streptococci, the gram-positive aerobic organism, also cause the sinus problem as well as pneumonia and sepsis. Proteus species also involve the UTI infection, and they are usually affect the patients and persons working in the hospitals. Pseudomonas is also involved in the lungs infection, and they are life-threatening for the patients who are in the case of cystic fibrosis. So the nanotechnology, in fact, I must say the nanobiotechnology provide a green revolution in the past few decades, and it provides the safe methods for the drug delivery system. There are various methods, and the scientists face a lot of problems for the delivery of the drug. So nanotechnology bring us closer to the safe delivery, just to absorb the drug, and there are some other ways by which the particles or the drugs can easily be absorbed in the affected site. The development of metallic nanoparticles provide an important revolution in this era. There are a lot of metals, silver, gold, platinum, and other metals have been widely used for the propulsion of nanoparticles, but most of the researchers prefer the silver nanoparticles. And the silver is the metal of choice for researchers due to the comparative non-toxicity towards the human, and they are toxic for a wide range of pathogenic microorganisms. So aims and objective of the present study to prepare silver nanoparticles from various plant extracts, which has the medicinal importance, and to evaluate the antibacterial activity of those silver nanoparticles. I briefly discuss here the material and methods. We use the 6 clinical isolates: Enterococcus, Klebsiella, Proteus, Pseudomonas, Shigella, and Streptococcus. These clinical isolates were collected from the ESA laboratory of Karachi, Pakistan. We use 12 different plant sources to form and to prepare the silver nanoparticles, and then check the antimicrobial activity against the selected pathogens. Here I briefly describe the methodology layout. First of all, we plant the plant extract, the methanolic extracts of the plants, and then prepare the green synthesis of silver nanoparticle by mixing the silver nitrate solution to the extracts of plant in a various concentration. Then incubate this setup for 24 hours. And after the 24 hours, the characterization of silver nanoparticles were performed by color change as well as by the UV visible spectrum. Finally, we detect the formed silver nanoparticles against their antimicrobial activity against the selected pathogens. So results of the present study showed that among the 12 different plant extracts, 4 of them formed the silver nanoparticles, and after 24 hours of the incubation, these solutions showed the color change, and the further confirmation of silver nanoparticles were done by the spectrum. In this figure, you can see the on the right side is a control of the silver nitrate solution, and the center there is a mixture of the extract as well as silver nanoparticle at time 0. And on the right side, the tube is after the incubation of 24 hours. So we can easily see the color change. Although the color change is observed in various plant extracts, but UV visible spectrum not coincide that every plant extract form the silver nanoparticles. So in the next figure, we can see that. In this figure, when we characterize the nanoparticles by UV visible spectrum by taking spectra from 300 nanometer to 800 nanometer, so we found that the 4 plant extract form the silver nanoparticles: Cribia, Camellia, Archaea, and Funiculum. Actually, according to the literature, when the silver nanoparticles are formed, they give the peak around the 300 to 400, 450 nanometers. This is the preliminary study. Still, this is not the final characterization of the silver nanoparticles because it needs the some analysis in order to confirm the formation of silver nanoparticles. Finally, we have done the antimicrobial activity of those silver nanoparticles, and we found that they are effective against wide range of pathogenic species, and different zones of inhibition are observed against the Grevia, Camellia, Archaea, and Funiculum for the pathogenic strain Klebsiella, Shigella, Pseudomonas, Proteus, Enterococcus, Streptococcus by giving the zone of inhibition of 15 nanometers the highest, and lowest is the 8 nanometer, millimeter. 15 millimeter is the highest, and the lowest zone of inhibition is 8 millimeter. These are the results of the antimicrobial activity of various nanoparticles. Due to the shortage of time, I am unable to describe each and every figure. So if anyone is interested in discussing those results or the detailed methodology, you can email me. So I will discuss it. I overview here that in control we put the silver nitrate solution, and in the other wells we put the various concentrations of the plant extracts plus silver nitrate, or in fact the silver nanoparticles of those extracts, and then we incubated them on the at various temperature, 35 degrees centigrade, and on the other day we found the zone of inhibition against these plant extracts and silver nanoparticle extract of various species. So the conclusion of this study is that the plant could be the better source for synthesis of silver nanoparticles by providing the environmentally benign and environmentally friendly procedures, and this procedure can be easily be scaled up. As this study is the most important thing of this study is that it showed the inhibitory effect against various pathogens. But still, this is, as I said earlier, this is the preliminary studies. Future studies will focus on the isolation and purification of a specific compound that could be responsible for the inhibition. As you know that in plant extract there are hundreds and thousands of compounds are present, and our focus will be that which compound exactly inhibit these specific species. So the future studies will focus on that, and then we will go towards the further characterization of silver nanoparticles like size and surface area, etc. So these are in the process. These are the references. Those are welcome, and thank you very much.
Okay, ma'am. Thank you. Thank you for the wonderful presentation. So very informative presentation. We have a question from the audience. Is there any health implications with too much of exposed to the silver nanoparticles? That's what they want to know.
There are several research which involve the animal study before going to the scale up, and we use the 1 millimolar solution of the silver nitrate, and according to the literature 1 millimolar silver nitrate has no impact on the health. The first thing is that, and the second thing is that when the silver nanoparticles are formed, the silver has been reduced by the combination of various components of the plant extract, so the toxicity become reduced.
Right,
But before going to the final composition of any drug, we should have the clinical trials and animal trials, and as well as clinical trials.
Right, ma'am. And one more small query actually from my side. Actually, there have been a lot of articles published regarding the biosynthesis of silver nanoparticles from the plant extracts. Like in which way your study is different from those studies? Is it like you have used different plant extract, or in which way it is really different from those studies?
This is just we first we think that which strains are drug resistant in the Karachi, Pakistan. This is a part of our study. We also identify the drug resistance of these pathogens by placing them in the various concentration of the available drugs. Then we apply these extracts on them in order to compare the zone which is observed by those available drugs and by the silver nanoparticles of these extracts.
Okay. And there's one more question from the audience, ma'am. What are the challenges in using a plant for synthesis of silver nanoparticles?
The most important challenge is the environmental issues, especially in Pakistan, because if we found that the certain plant is really very helpful for the any drug discovery, so the most important challenge is to cultivate that plant. But somehow the plant tissue culture remove these challenges also.
Fine, ma'am. Thank you. Thank you for the wonderful, wonderful presentation. And so, shall we wind up the session, sir? Or because this is the last presentation, and but we have two more presenters. They had to leave in between because of the connectivity issues. So, are we going to wind up the session and giving them an opportunity to present tomorrow?
So we will pass the message to organizing committee. So I think they can embed their presentations in one of the sessions. I think tomorrow we can arrange. We can pass. Organizing committee can pass the message to them because we tried a few times.
Fine, sir.
But still they are having an internet connectivity problem, and it's 8 o'clock and 10 minutes Malaysian time. Even though participants are from different parts of the world and different continents, sorry for this technical problem. Dr. Shiva, you were saying something, sorry.
No, that's fine, sir. Maybe if this is the last presentation, so maybe you can wrap up the session.
Okay, thank you, Dr. Shiva. So altogether in this session, altogether there were 12 presentations. And in between, we have also completed one plenary talk, which was not completed because of technical reason in the previous session. So participants from different continents were listening, and those were all doing oral presentations. We are from India, Malaysia, Nigeria, and Pakistan. So just to summarize the session, I think this overall session was really wonderful and very impressive, because if you see the beginning, Ms. Sweta was excitingly presenting the possibilities of how the cellulose degrading bacteria from the termites' guts can be used to develop something, especially to develop the cellulose-based sustainable nutritious solutions in a resource-limited setting. So this type of research is, needed, because the global community is talking about the sustainability and dealing with the number of challenges, and this one is in line with the global sustainable development agenda. So if we see another presenter who was talking about the macro micro elements and heavy metals in the various polyherbal formulations, those are used to treat the diabetes. This case is from India, but this one gives the insights for everybody, because when we are using different types of formulations from the market to treat the different health elements, so these factors are also important, and Ms. Paul highlighted this one very nicely, and we got insights. The third presenter, Dr. Selvaraj, was deliberating the biosynthesis of copper oxide nanoparticles, and if we see the overall presentations, because many are on nanoparticles and some of the issues, and the ultimate aim is to contribute to improve the human health and to solve some challenges. Another speaker, number 4 speaker, he highlighted the importance of bioprospecting by capturing the bioactive compounds from mushroom and how it can be used to solve some of the problems. In addition to that one, another speaker highlighted the biosurfactant producing bacteria from the hot spring. So I think that one is also gave some insights about, even though it's a hot spring, there is something useful. So if we know the Taq polymerase, which is commonly used in all molecular biology laboratories, so basically that one is from a bacteria which can survive in a hot spring. So then another speaker who was talking about conversion of the waste from the bakeries. So this one is also in line with global trend. If you know the global sustainable development goals, so I think it's perfectly fitting in that one, and it is reflected that industries do have different types of problems to deal with the waste, and that's why they are. We as a scientific community and academicians in partnership with the industry, we need to find the solutions. In addition to that one, we also heard one of the speakers was talking about botanical insecticides, because now everybody is going for the back to the nature because synthetic insecticides do have many bad effects on the environment, especially the contamination of the river water as well as soil and food supply chain. So in that context, it was, a good initiative. Then another speaker was talking about global E. coli genomes, and very meticulously presented the data on E. coli genome, because there are more than 4,000 isolates genomes are available in the gene bank, and presented the antibiotic resistant genes. Those are available in the database. Then another 2 speakers, unfortunately, from the Nigeria and one from India, didn't manage to present because of internet connectivity. So we request organizing committee to look into it, and maybe they can consider tomorrow to give them a chance to complete their presentation. The last speaker from Pakistan was talking about the photosynthesis of silver nanoparticles using plant extracts of medicinal importance. So as a whole, if we look at all these presentations, so all this research is going on either to solve some problems in agriculture or healthcare sector, to improve the human health, and industrial challenges. For example, the bakery waste was one example. So everybody is, trying to solve some problems and come up with some solutions to make a human life better. So as a whole, if I just conclude, because all the oral presentations, very informative talks, and the overall scientific quality was very high, and it was appreciable. The oral presentations by budding scientists, we had very some young scientists, those who were talking about the science. So their presentations were also very impressive. And this session was, it was a compliment. Professor Balakrishnan delivered his plenary during this session. It was an honor to host him. It was a very informative talk, and he gave some wonderful insights about the HLA and from the molecular medicine perspective. So as a whole, the whole session was really informative and very good. And I can see the overall presentation and quality was very good. So I want to take this opportunity to thank all the speakers for presentation. Delivering your presentations, and I also want to thank Professor Balakrishnan for delivering plenary talk. Even though there were some difficulties because of connectivity, but we do appreciate his efforts and willingness to share his knowledge and wisdom with participants. I also want to thank my co-chair, Dr. Siva Kumar, for helping me to coordinate. And I also want to thank, last but not least, I want to thank the organizing committee for giving me this opportunity to chair this session. So thank you very much, all the participants, ladies and gentlemen. Maybe you are in different countries, different time zones. So thank you very much for staying with us. We started this morning at 9:30 Malaysian time, and it's almost 11 hours. So I mean, we are talking, and lunch break was also shortened. So thank you very much for staying with us. We do appreciate your participation, and once again, I thank all the participants, all the speakers, plenary speaker, and everybody who is playing from behind the curtains. So once again, thank you very much. I pass it to MC, Gerard. Gerard, over to you. Thank you. Gerard, are you there? MC?
You need to give permission, I think. That's Subhash.
Yeah, he's co-host, Gerard.
Okay.
Thank you,
Okay.
Dr. Subhash.
Yeah.
Thank you, Dr. Subhash and Dr. Siva, for co-hosting, hosting this session. So, well, ladies and gentlemen, it's been a long day. As Dr. Subhash told, it's been 11 hours worth of presentations and talks. So, after a long day, our session for this day has come to an end. I hope everyone has had a productive day. Thank you all for participating. I would especially like to thank the organizing committee and all the volunteers, especially the student volunteers who have worked tirelessly behind the curtains to give us a great e-conference experience. And I hope all of you all will be joining us back tomorrow for our last day for this conference. And we will be meeting back at 9:30 a.m. Malaysian time. So with that, I would like to wish a good day to everyone, and thank you.
Gerard, I will end the meeting.
Okay, sir.
Thank you.